Good morning, everyone. We're going to get started with MLCB 2024 edition. Thanks to my co-chair, David Knowles, and the organizing committee, Anshul, Suen, Gerald, and James, and UW for providing this beautiful space to us today. So for those of you that haven't been to MLCB before, I wanted to share a little bit of history with you. MLCB actually started back in 2004 as a workshop of NeurIPS. At that time, it was called NIPS. And it continued on for, I guess, over 10 years after that until 2016. And the participation grew. And at some point in 2016-ish, NeurIPS basically told us that MLCB is like a mature science now. Before then, it was kind of an emerging area, experimental between computer science, machine learning, and biology. And it was too mature to be a workshop at NIPS, which are supposed to be for emerging areas of machine learning. And at some point in 2017, we were thinking what to do. We can't be a NeurIPS workshop anymore. So we decided to do this experiment where we created MLCB as an independent conference. And the first time we did this, to take it easy on us, we made it co-located with NeurIPS, which worked out really nicely. And we had actually over 300 participants. But then the pandemic happened, but we still grew. And we were virtual for a few years until last year, where we created this hybrid mode where we have in-person and kind of virtual meeting. And the last 2 years have been in Seattle. Next year to be determined. At the end of the conference, we'll send a survey to see which location is most suitable for everyone here. Here is some information about the submissions that we get. So the submissions that we've been receiving are really high quality, has been growing over the years. Sorry for my graphs that I had to put together this morning. But this year we had about over 100 submissions. Small number of them were not very relevant, so the blue parts are rejections. We have 18 really strong oral presentations, a bunch of poster presentations, which you'll see today. And the poster presentation will be just the building across the street called the Allen Center in the atrium. And as of a few years ago, we started also having this PMLR proceeding where we accept 8-page full papers that get reviewed for our conference proceeding, and a small number of them are published in the special issue of PMLR. It's also fun to look at how the field has changed over the years. So this is the word cloud of the papers, or I should say titles of papers submitted to the first occurrence of MLCB in 2004. And maybe some of you were not even born yet. I was thinking about that today, and it's just amazing. So you might not realize that at some point, kernels were like the neural network of that world before you. And so kernels and proteins and learning, of course, and I think interactions were a really big thing. So now what has changed, like 14 years later? Well, first of all, this was generated by ChatGPT, so I think it's like probably a more accurate representation of what has been submitted. And I think there's a lot more regulatory sequence models, RNA, DNA. Back then, it was mostly proteins, it seems. Well, today, proteins are big, too, but I think regulatory sequences are the next big thing, plus RNA. And they're still learning, and they're still multitasking. But there's also fine-tuning. You can see the fine-tuning in the bottom. Well, this was the orals, so it's a small select number. But what about the entire MLCB submissions? So I think the regulatory seems to be mainly in the oral, so that's an emerging, really important area. But the whole submissions, we have DNA, we have predictions, data. Data is a big thing now. I think back in 2004, people didn't really care about data that much. It was more about the models, maybe. So that's the fun part. So we're going to have an exciting program ahead of us for the next 2 days. We have 5 keynote speakers. You hear from 2 today and 3 tomorrow. We have 17 oral presentations that are about 20 minutes each. We've asked the presenter to make sure they have a 15-minute talk to leave some time for question and answers. We have 18 spotlight talks, which are 3-minute long, and really they're like an advertisement for the posters that got the top scores under our review system. And I mentioned the poster session will be later today across the street in the Allen Center. And today we also have this fun panel, and the topic is foundation models for biology, when it's useful or not useful. So that should be fun to talk about. And finally, I'll end with thanking our sponsors. So we have very generous sponsors this year and the last few years. This year, our sponsors are Recursion, Deep Genomics, Shape Therapeutics, and InstaDeep. So thank you so much. And the generous donation from, or funds from these sponsors allow us to actually have this free conference, as you all can enjoy. All right, so now I'm going to hand it over to David, who's going to introduce the first keynote.
And do you want to start setting up? Yeah, so it's my great pleasure to introduce Jian Zhou. So Jian's really been a leader in the field of using deep learning to represent sequence-to-function relationships, kind of from the beginning of this becoming like a mainstream area in computational biology. So did really like groundbreaking work with Olga Troyanskaya, developing DeepSea, one of the early applications of convolutional neural networks to predict transcription factor binding, represented by ChIP-seq, and lots of kind of really great work in that sort of direction. So predicting RBP binding, which is an area close to my heart as well. And then in his group at UT Southwestern, they've been doing really great work looking at these really challenging tasks like predicting chromatin conformation as represented by Hi-C, with this ORCA method, some really amazing engineering efforts there, and most recently understanding transcriptional regulation using interpretable deep learning models, which I think is what you'll be talking about today. So excited for your talk, and thank you for coming.
Yeah, thanks, David, for the kind introduction. And I would like to thank the organizers for the invitation. Today I'll share with you a story about discovering how the sequence controls the transcription initiation. So to start, I will start with a probably somewhat provocative statement, right? So genomic sequence might be the most underexploited resource in genomics, right? So what do I mean here, right? So of course, that genomic sequence is being used every day in all kinds of research. However, I know with the exception of many of you here, but most of the time, when genomic sequences were used, it's often used as a way to make a measurement, right? So we use the reference genome to do all kinds of measurement about the genome, and it's used to detect mutations. But the actual sequence that's encoded in the genomic sequence, right, the biology that's encoded in the sequence, is still relatively rare to be extracted as a way to drive those biological discoveries, right? So most of the discoveries were not made from extracting this information from the sequence. So compared to how much biology is already encoded in our genome, so there's a huge gap between that and how much that we can use that for discoveries, right? So I think there's a huge gap here, and also like a great opportunity to, by reading more information out of the genome and use that to help us gain better understanding of the biology. Right? And to bridge that gap, of course, I think the challenge is that it's still hard to understand the genomic sequence. Right? So I think understanding the genomic sequence is a problem that is it's quite hard for traditional approach, but it's almost perfectly set up for machine learning approaches. Right? So reason for that is that first we know, of course, our entire genomic sequence in the human genome, the 3 billion base pair. And at the same time, we also have tens of thousands of different genomic data sets that's measuring various kinds of activities about the genome. Right? Let's put some random ones here, from the transcription factor binding to the 3D genome organization to the transcription in increasingly more cell types. Right? However, just having those genomic data, it won't directly tell us about how are those activities determined, or at least depend on the genomic sequence. Right? So those are the information that we cannot directly measure, but we can infer through a modeling approach, right, by building machine learning models. Right? So in the space of capturing the dependencies between the genomic sequence and its activities, I think of it as 3 main directions that we explore. So the first direction is, of course, prediction. So we want to go from the genomic sequence to predict its activities, right? So it's also where I started from, and in the past we've done a lot of work in developing sequence-based models to predict various kind of genomic regulatory activities, right? And as we build those models, we think they are great tools for us to understand the underlying mechanisms of genome regulation. But I also gradually realized that just having a predicting model, so being able to predict something does not equal to being able to understand that, right? So a lot of time, I think we certainly get the feeling that our model understands it. It can certainly understand something that we haven't understood about the underlying biology, but we don't know what it is, right? So I think it is still a non-trivial task to be able to extract that understanding from the predicting models. So it is a challenging task on its own, and also I think it requires development of methods specifically for understanding on its own. So I put it as a separate direction here. It's also one direction that I will be mostly focusing on today. And the third direction is kind of the flip side of prediction, right? So instead of being able to go from the sequence to predict its activities, we also want to be able to do it the other way around. So we want certain kind of regulatory activities. Can we design sequences that give us that activity? So I think there is a synergizing effect among all three of those directions. So today I'll be focusing on a study of transcription, and more specifically transcription initiation. And I'll be mostly touching upon the direction of prediction and understanding, but it's also one direction that we have done a bit of work on all three of those directions. So we started from asking a simple question. So by the way, this is work with my brilliant graduate student, Sinia. And so we started by asking this question of so what is a promoter, right? So what defines a promoter at the sequence level? So of course, we know that promoters are essential part of a gene, right? So you can have a gene that does not encode the protein, or you can have a gene that does not have an intron, but all genes have to have promoter to be transcribed, right? So it is one of the most essential elements in the genome. But perhaps surprisingly, we think of what should a promoter look like at the sequence level in the human genome. So our understanding to that was still quite incomplete. So in the past few decades, there have been quite a bunch of those called promoter motifs have been identified from many different organisms, right? But if we look at the human promoter, so even a strong promoter, it might have none of those motifs, right? So like the Tata motif, depending on our threshold, we think it's present in no more than 10% of the human promoters. So for most of the human promoter, you look at the sequence, it's still hard to tell why is it a promoter and why transcription starts at the exact position that it starts at, right? So it's still something that we didn't have a full understanding about. So to tackle this question, we want to just first formulate it as a machine learning problem, right? So what we want to do is that we want to build a model that can start from the genomic sequence and predict the transcription initiation activities at the base pair resolution. So those are one examples of the transcription initiation site measurement by phantom cage. So those measurements give you the precise 5' end of the transcript. So you can count how many transcript start sites are detected for each base pair position. Right? So you plot that on the axis of genomic positions, you can obtain a plot like this. So the y-axis is on the log scale. So you see this wiggling of those activities across different positions, right? So those wigglings are not noise. So it's actually highly reproducible across samples, and even reproducible across cell types, right? So we think those curves, those wiggling shape here actually reflects the underlying mechanism of the transcription initiation at this promoter. Right? So if we are able to capture how are those shapes determined at the sequence level, we might have captured something about the mechanism of the transcription initiation. Right? So we first built a deep learning model to be able to predict that from a 100 kilo base pair of sequence. And also, it turned out that we are not the only one who had this idea. So Zhou's lab at Stanford, also Charles Dunkel's lab at Cornell, also independently develops those methods that does similar kind of prediction. And you should also check out those work. And I think the author of ClipNet, Adam, is also here in the conference, so you should talk to him as well. So what we did a little differently in our approach is that we are able to just study this deep learning model that makes this prediction very well and summarize what we learned from the model and use it to develop a simple model that can also do a very good prediction of the shape of the transcription initiation, but only using 600 base pairs of input and also a very small amount of parameters. And we can interpret pretty much every one of those parameters. Right? So this small model that we call puffing is the focus of this talk. So before I go into how does this model work, just show you the prediction performance of this model. So this example of a promoter and its transcription initiation activity profile predicted from the simple model, the deep model, and the experiment. Right? And just two examples of that. And of course, we can quantify this performance by looking at the correlations of the model prediction on a holdout chromosome that the model has never been trained on. Right? So we summarize those correlations. We see that for the most expressed promoters, where we have the most measurement, the prediction correlation can get to up to 0.9. Right? And with the promoter that has lower transcriptional activities, the correlation is lower, but it's also partially due to the noise in the measurement because we look at the correlations between two different experimental measurements of phantom cage and encode cage. We also see a decrease in correlation with the lower activity promoters. Right? So overall, the puffing, even though it's not as good as a deep model, it is still doing pretty well in capturing the shape of the transcription initiation. And also, both of them are actually able to achieve higher correlation compared to when you compare the two different but most correlated experimental techniques. Right? So which says that the model should have captured quite a lot about what determines the transcription initiation. So even though it is a very simple model. Right? So how does this model work? So we summarize it in this diagram. And actually, it's very simple, just doing 2 steps of computation, mainly. So I just go through that step by step. So the first step is that we start from the genomic sequence, and we scan for motifs that are present in the sequence. Right? So how it's done is through there's a convolution layer with the soft flux activation function, and you get this motif activation profile. So we use different color to indicate different motifs, right? Just detecting the motif positions. And next, the second step, the only other step of computation that's needed is that we convert these individual motif activations into its corresponding effect, like this red curve, which is Tata activation is converted to this red effect, right? And this NFY motif activation is converted to this curve of NFY effect. So that is, this effect are applied through another convolution layer. And then we only have to sum up the effects at each individual location. And surprisingly, that is sufficient to make a good prediction for most of the promoters, right? So what I didn't tell you is that, so in addition to the motifs, there are also 2 other simpler types of sequence patterns that we detect, which effects were also added to the motifs effect to obtain the final prediction. So the two other sequence patterns are initiators, which are only affecting very local ±7 base pair window to fine-tune the base pair resolution transcription initiation signal. And the other one is a trinucleotide, which is just the residual dependency that we couldn't explain, but we can capture that with just a 3-nucleotide effect. So as we sum up all these sequence pattern effect per position, it gives us our final prediction already. So which is, you can interpret it as the log of the transcription activities, and we just need to convert it into the log +1 scale to compare with the experiment. Right? So I would like to also show you that we can have this model working live in a web server that we made. Right? So this is you go to like puffin.zhou.io. So this is a server that actually loaded up this model that you can look at, not just look at the sequence, and you can also edit them. Right? So if I remove the Tata, you see the prediction decrease. Right? And if I can remove the other motif, the prediction will decrease even further. Right? So this is a way that you can actually just interact with the model live on the website. And you click on demos, it shows you the predictions for many different promoters. Right? So I'm just showing this to give you a sense of how diverse these promoters are. Right? So almost every one of them have distinct combinations of motifs. But following the simple rule, we can predict quite well about the transcription initiation activity in all of those promoters. Right? So going back to the model, so how does it make this prediction? Right? So the core of it are the motifs that it has learned from this task of predicting from sequence to the transcription initiation activities. Right? So all of those motifs were learned from scratch. We didn't give it the motif. But after we have found those motifs that has been repeatedly learned as we train multiple rounds of those models, we figure that most of them actually match known motif. Right? So that's why we can give it the names here. But with the exception of Tata, actually for most of those motifs, we were not known. It's not known previously, like whether and how it contribute to transcription initiation, right? Which was captured in the motif effects that are learned here, right? So those are probably the most key information that has been captured by the model, right? So how to read this graph is that if we position the motif at the center, we place the Tata at the center here, it will activate transcription at almost exactly 30 base pair downstream of that motif, right? So if you put the YR1 motif at the center, it's going to activate the transcription at almost immediate upstream of that motif, and also has a weaker effect, about 200 base pair upstream of that. Right? So we can also resolve the effects on the two different strands. Right? So as you can see here, both Tata and YR1 has pretty strand-specific effect. Right? So it has different effects on the forward strand transcription and the reverse complement transcription. Right? But there are also a group of motifs which have almost symmetric effects on both strands. So you activate transcription on both strands simultaneously with almost identical effects. So we call those bidirectional motifs. Right? So those bidirectional motifs, they seem to share some similarities in their effect curve. But still, each one of them has a distinct shape. Like for example, for NFY, you see this about 10.5 base pair periodicity, which matches the helical periodicity of DNA, which rotate every 10.5 base pair to the origin, right? So we think that reflects that NFY might have a more rigid interaction with the POL2 pre-initiation complex, right? So it has to bind at a certain surface relative to the POL2 pre-initiation complex, right? So perhaps each one of those curves are actually reflecting the underlying molecular mechanism of how does those motif and the transcription factor activate transcription initiation. Right? So there's a special motif here that we included, but we know that it does not affect transcription initiation, or have very little effect on transcription initiation. So the reason that we can know this is that we built models for different types of experimental techniques. So including those that measure mature transcript, like CAGE and Rampage, and also those that measure only nascent transcript, like ProCap and GrowCap. So this motif, the UNSNRMP, also known as the 5' splice site, it only has this effect when we are looking at the mature transcript. But it has no effect if we're looking at nascent transcript level. So, which means that it likely affects the amount of transcript after transcription initiation happened, right? Which is also consistent with recent studies which show that it has an effect in promoting the transcription elongation. And finally, there was one motif that are highly directional, and initially we didn't recognize it. But later we realized that it is this very long motif. It's actually a combination of several previously identified co-promoter motifs, including the initiator, which is where the transcription starts at, and the several downstream motifs here. Right? So all of those motifs were considered to reflect the preference of PF2D, part of the pre-initiation complex. And I also think that this long initiator might be reflecting the overall preference of TF2D. Right? So, with those effects, so now we also want to be able to just more directly test or verify the effects that the model have captured. Just beyond the fact that these motifs are reproducible and able to make good predictions on the holdout chromosome. Right? So one thing this model can allow us to do, because it is a simple and interpreted model, which we know that how each element in the model do, is that we can now predict the effect of removing a transcription factor from the system. Right? Because we know which kernel corresponds to NFY motif activation and motif effect, we can just remove that NFY activation and see what are the predictions of the transcription initiation signal after we have removed NFY. Right? So because NFY will activate transcription downstream and slightly repress transcription upstream, if we remove them, generally the effect is a decrease in the downstream and a slight increase in the upstream. Right? So we can summarize the prediction here. Right? So depending on the NFY position and all the other motifs in the promoter, we see different effects at different promoters. Right? So here it is sorted by the activation effect to be able to more easily see the trend. And there is a data set that has been generated that did an NFI knockdown. Right? So we compare with that data set and show that experimentally we can see a very similar pattern as predicted by the model, right? Which is the repression downstream and activation upstream. And we're also able to test that for another motif that we predicted to have an effect on transcription initiation, which is YY1. Right? So this is a pretty effect of YY1 knockout, and this is the experimental effect of depleting YY1. Right? So we also see that the predicted effects were indeed observed in the experiment, and even the secondary, like the less strong effect of YY1, about 200 base pair upstream, can also be observed in the experimental measurement, right? So this effect was actually not originally reported in this paper, but when we digging into the data set, we found that actually there is an effect of YY1 knockdown, YY1 depletion on the transcription initiation. Right? So we can also test it in the waves of mutating motifs. Right? I'm not going to go too deep into this, but I just show that the model can also reproduce the experimental effect of inserting and deleting those transcription factor motifs. And after our preprint is published, there have also been a couple of other studies that have been consistent with our prediction. So for example, one of the studies that tried to look into the ZNF143, which is actually one of the surprising factors that we were not expecting when we were building the model. They were looking into it with the expectation that it might be a looping factor, but it ended up finding that it is a transcriptional regulator, which can, if you mutate it, it's going to significantly decrease the transcription signal. Right? So another study that also verified that those motifs that we also captured in the model will have an activating effect on the downstream of this motif and the repressive effect if it is the transcription start site is on the upstream of those motifs. Right? So beyond those experimental validations, we can also look at the evolutionary conservation. And somewhat to my surprise, you can also see this trend quite clearly just by looking at the conservation signal. So what is showing here is that if we look at the conservation of motifs, it actually depends on where it locates relative to the transcription start site. So the motif will be more conserved across species if it is located at the position where it has the maximal effect on the transcription start site, right? So like Tata has the maximum conservation when it is located about 30 base pair upstream, and YY1 has the maximum conservation when it is just downstream of transcription initial start site. And even this periodicity of NFY can be seen through looking at the evolutionary conservation and analyze its dependency on the distance to the transcription start site. Right? So all of those are supporting what we have learned from the model. So now we're quite confident about what the model has learned about this, how does those motifs contribute to transcription initiation. And now we want to ask, like, what does it mean for the gene, right? If we know which motifs are contributing to the promoter, right? So we can now summarize the contribution of each motif to every promoter into a score, right? So here it is an example of the contribution scores for all of those motifs to top 100 most expressed promoters in the human genome, right? So in the highest expression promoters, you see an overrepresentation of Tata motif, but still the contribution is quite diverse, right? You see many different combinations of those motifs, which is generally what we see for those promoters. We can do this analysis for all the top 40,000 promoters, and we just sorted by the motif that has the maximum contribution to this promoter. So SPs are the motifs that contribute to the most promoters, but each one of these co-promoter motifs are contributing, is the dominant motif for a fraction of these promoters, right? And overall, we see when we look at the correlation between the any pair of motifs, it's never very strong. It's always below 0.1 correlation. So we really think that you can very flexibly combine those motifs to make a promoter. Right? Yeah, but what does those motif contribution mean for the gene? Right? So all of those motif bind transcription factors that are ubiquitously expressed, even though the expression level can be a little varying. So I don't think any one of them will have a strong effect preference to a certain tissue, even though ETS is slightly have slightly higher effect in blood lineage instead, especially T cells. Most of them are having similar effects across tissues. However, we do see a pretty strong correlation between the motif contribution and the expression variation across cell types. So here, we're looking at the expression level and its dispersion across cell types. So each dot here represents a gene, and the x-axis represents the expression level of that gene, and the y-axis represents the dispersion of that gene. So I should say promoter, right? So it's actually quantified at the promoter level. And the color here indicates the contribution of Tata motif to this promoter, right? So a clear trend that you can see here is that with higher contribution from Tata, the promoter is more likely to be variable across cell types, right? Even at the same expression level. And in contrast, if the promoter has very high YL1 contribution, it tends to be more ubiquitously expressed and less variable across cell type. So all of them, each one of these motifs actually has its own preference in this spectrum of preferring this more tissue-specific expression pattern versus the more ubiquitous expression pattern. And the U1 SNRMP, the 5' splice site, as we mentioned that it should not affect the transcription initiation. It's kind of neutral in its preference to the tissue-specific versus ubiquitous expression pattern. So why is that? So why are those motifs preferring different tissue specificity level? So we have a hypothesis about that. So I'll just first tell you what's the hypothesis, and then show you why we think that is the way. So the hypothesis is that the motif composition will tune the responsiveness of those promoters to the transcription regulatory signals like enhancers. So the Tata promoters might have a higher dynamic range than the Y1 promoters, which may have a lower dynamic range and less responsive to the regulatory signals. So the reason we think of it that way is also because we look at the promoter activities in an almost orthogonal way, and we see a similar order of motifs as the tissue specificity. So here, we just insert a lot of promoter sequences into different genomic locations and test, and we predict its activity from the deep learning model, right? So what we found is that if we insert a promoter which have a strong Tata contribution, then this promoter is more selective. It is expressed in fewer positions relative to the max expression level, whereas Y1 promoters are less selective. It is more likely to be expressed in more positions where we insert it into. And this general order of motifs is quite consistent with what we see in terms of the tissue specificity. So it's still a hypothesis, but we think that that might be what explains the preference of those motifs for different levels of tissue specificity. And finally, what we can explain with this model, you might notice from introducing those effect curves and bidirectional motifs, is that why those most human promoters we see transcription initiation on both strands, on both directions, if we look at it at the nascent transcript level, right? So most of the human promoters are like that. If you look at the nascent transcript, you most likely see transcription on both directions, right? If you look at the mature transcript, it is much more unidirectional, right? So we think that bidirectional transcription is due to the bidirectional motifs which activate the transcription initiation on both strands, right? And for the mature transcript, you have more contribution from factors like the 5' splice site, which only affect the transcription, mature transcript level on one strand, right, which leads to more directional output of the transcript. So we can also do this quantitatively. So you can also quantitatively analyze what are the contribution to each strand at base pair resolution. So here is an example where this is an experimental measurement, and it's our prediction. And we can quantify how much each base pair contribute to the forward strand, and how much each base pair contribute to the reverse strand, and compare how much they are shared, right? So this is a contribution to the forward strand. We can also decompose it into contribution through different motifs, right? And this is the contribution to the reverse strand. And as you can see that it is very similar to the forward strand. And when we summarize this for all of those forward and reverse transition star side pairs, we find that if the distance is smaller than 300 base pairs, more than 90% of those contribution scores has more than 0.75 correlation, which means that they're quite shared. They have both the forward and reverse strand transcription has very similar sequence contribution. So finally, we also want to know how conserved are these, what's the model captured across different species, right? So we can train this model for both human and mouse because both of them have very similar experiment done through the Phantom Kit project. So we can train separate models on human and mouse data and make predictions on the other species, right? So to summarize, what we see is that the model that's trained on human and mouse are basically giving us identical predictions. So the correlation is very high, very close to 1 when we compare it. And moreover, if we compare the human model and mouse model prediction, and we only evaluate on the mouse experiment, actually you don't see a human model having a lower performance compared to the model that is trained on mouse data, right? So I think it's really that it is almost identical in terms of how does transcription initiation depend on the sequence between human and mouse. And we also want to look more diversely across mammalian genomes, right? Which is hard because we don't have those transcription initiation measurement for all those other species, right? So we did what we can do. So we try to look at the evolutionary conservation. So previously, I've shown you that if we look at it from the human genome perspective, the motifs are more conserved when it is located at position where it has stronger contribution to transcription star site. So now we do it from the viewpoint of each of the 240 mammalian genomes, right? So we are just starting from each one of those genomes. We do have to take assumption that the transcription star site is conserved from the human to that species. With that assumption, you see that, so here actually there are 240 curves here, each represent the conservation in one species. They all share a very similar trend of the position dependency, right? Which seems to suggest that it is likely that this sequence dependencies are also conserved across mammalian genomes. So to summarize, that we are able to build a simple model that can predict from the sequence to the transcription initiation signals. And this has been able to allow us to understand something about the promoters, including like what are the sequence dependencies and how does those motifs contribute to the promoters. And we are able to use that to stratify promoters by motif contribution and found that certain motifs have different preference for tissue specificity. And we can use that to explain the directionality of those promoters. And we can look at the conservations across different species. And just finally, I think to look ahead, I want to just revisit this okay, triangles that I posed at the beginning, right? I think here, I think mostly just looking at the synergies between the predicting models and the understanding, right? So we're looking at how predicting model can help us gain a better understanding about the underlying process. I think there are still a lot more synergies here that has not been explored. For example, if we gain a better understanding, can we use that to design better predictive models, right? So can we use that to design new promoter sequences? And if we design some sequences and we measure its activities, can we use that to verify or refine our understanding, right? And I think there's really a lot of synergy between all three of those directions, which can hopefully improve our ability to do each one of those tasks. And hopefully, I think it can help us get to the question that I posed at the beginning, so we can use that to extract more of the biology through studying the genomic sequence. So yeah, finally, I'd like to thank my group who have contributed to this study, which is Sinia, who is the first author, who works with me mostly on the computational side, and the Donghong and Jian from Singhal did the experimental validations of this study, and Chen Aishi developed the website. So thank you for listening, and I'm happy to take questions.
Yeah, we definitely have time for some questions.
Hey, so I have a technical and a conceptual question. So for the motifs that appear symmetric in terms of activation, they're not exactly the same.
Right.
Do you know if that's like up to statistical error or there's something meaningful there? And my second question is, how much do you have to worry about dynamics, right? Because some RNAs are initiated, but they're immediately degraded, and then you don't observe them.
So the first question, I think, is from the reproducibility, we do know that this slight asymmetry from the forward and reverse strand are actually reproducible. So if we train the model multiple times, we consistently see these differences. So I do think there is some slight asymmetry, even though that is almost symmetric. And for the question of the dynamics, so I'm not sure I completely get your question. So there might be some of the intermediate transcripts that were not able to be detected through the existing technique. Is that?
I mean, it's not like a flaw in your approach. I was just wondering if you've thought about this, that in theory there's a bunch of transcripts that are initiated, but they're degraded, and you don't observe them even in the cage data, and like how that factors into how you think about the process.
Right. I do think that the current model is kind of, as you said, I think kind of a static way. I think the way that I understand the Patel model is that we have those motifs, and each of them have can probably alter the binding energy of the pre-initiation complex, right? And that sum up to the energy profile of the pre-initiation complex binding at every position, which leads to the transcription, right? But I think it is. This is probably not the full picture still, right? So it's a first order approximation, probably not, there's still probably a lot of other things that are missing. And one thing that I do nowadays try to think about is like, how does a model these dynamic processes like happening in the genome? Like, how does the order of things affect the outcome, right? So if a transcript factor binds first and another transcript factor binds second, or like if there is some order of transcription, minor burst of transcription event, and it might affect the later transcription level, right? So how would those dynamics affect how we should be building our models? I just don't have an answer to that, but I think it's certainly questions that we should be thinking about in terms of building those interpretable models. Yeah.
Hi, beautiful talk. I wonder how it would look like if you distill all this to a set of rules, basically describing what makes a promoter. Is it simply 8 or 9 exclusive groups of promoter classes, or can you go more granular and define in terms of different combinations?
I mean, use that to group the promoters into different classes, right? So I think first of all, what we tried here is to look at the promoters sort of classes based on which motif contributes the most to that promoters. Which does show different properties of those promoters by the most contributing motif. But on the other hand, do I really think that these promoters are well clustered into different clusters? think not necessarily, because when we look at the correlations between different motifs, the contribution and correlation are usually pretty low. So there is a quite flexible way of combining those different motifs into different promoters. So I think it's more of a continuum rather than having different classes, even though having different motif contribution will likely still affect the property of those motifs, of those promoters.
Thank you for the great talk. So I think in your one of the last slides, you were showing that the multiple models that you have developed over the time, and for example, I think there was Expecto and Puffin, I think, grouped together. So when these different models are in the literature, and there are different groups developing these models, do you think we should come up with on consensus on the folds to train them on, validate them on, and test them on? For example, for Expecto, I think you proposed removing the paralogous regions from the test set. So, and did you follow the same procedure for Puffin test sets to remove probably the paralogous genes or the homologous promoters? Thank you.
Yeah, I think that's a very practical question, I think, for those of us who develop those sequence models, right? I think it would be very helpful if we can all compare with each other's models on the same holdout set, right? So, our models, we do always try to be consistent in the holdout sets so that it's comparable across all the models that we develop. But certainly, I think different groups can use different holdouts. So if they are holding out different chromosomes and different regions, then it is harder to find just the intersection to evaluate the models. I think it will be indeed very helpful if I think the field can agree on a certain way of doing the holdout, or as David Kelly's group have been recently doing, that you actually train models using just many folds, many cross-validation folds, so that you can actually have different models that are holding out all parts of the genome, right, in different models, so that also allows people to compare with each other more easily. So I think both of those will be very helpful for the development of models in the field.
Great talk, Jian, and very interesting study. It's very interesting that some of the motifs, like Tada warnings, has more cell type specific, like driving ability in promoters. I wonder, have you thought about, let's say, design a synthetic promoter that, well, usually we think that the cell type specificity or tissue specificity came from enhancers. Have you thought about design a specific promoter that can boost the expression of enhancers and maintain the specificity of those enhancers?
Yeah, I think that's certainly something that we are interested in exploring. So following what we think, I think we build a strong Tada promoter that it should be more responsive to enhancers compared to we build a YYM promoters, right? But how do we balance the expression level versus specificity? I think that is still something that we don't know, and I think it will be a great question to ask in the future.
I think, unfortunately for time, we probably need to wrap up there. Let's thank Jian again for a great talk. So next up, we have Thomas Pierrot, who will be telling us about the nucleotide transformer. There are professionals if we have any struggles with setting things up. So.
Take that one. Okay, sounds good.
Okay.
Okay.
I'll try and signal when you have like 5 minutes left.
Sounds good. Okay. Hi everyone. Can you hear me? It's good? Yeah. Thank you so much for the opportunity to present today. So I present the research that we're doing at InstaDeep and BioNTech in our group on building foundation models for genomics. And kind of to start this talk, I wanted to start with what is for us, at least, the definition of foundation models. Okay, I think there are still many definitions out there, and not everyone agree. And so I actually asked Gemini, because why not asking a foundation model for what is the definition for what it is? And what's fun is that it's kind of also the definition we had on our team and that you can find on Wikipedia and others. And so for us, we're going to go like foundation models, any large AI model that's been trained on lots of data and that can be used to like perform lots of different tasks. And so with that definition, we can take all the models trained with unsupervised learning or self-supervised learning that I present today, but also all the models that have been trained with supervised learning, for instance, to predict lots of different tasks, such as Borzoi or Informer, are going to satisfy this definition. And so I've put on that side lots of different sources of data that one can use to train these models, and lots of different models that have been trained so far, some of them that I covered today. And obviously, the list is much, much longer than that, as the field is kind of growing a lot right now. And in our group, the question we're asking is, how can we use all of this data, and even more, to build these models that eventually one day will maybe teach us new things about biology. And to start that journey, we started with these nucleotide transfer models, where the idea was to first run from genomes and use all these techniques from NAP and unsupervised learning, saying, how can we take these genomes and run in a free unsupervised way? So we have no labels, we just have genomes from individuals, genomes from species, and we want to train a model that can run something out of that and be useful. And the first idea we had, and that's now been done also in lots of other models and other fields in science, was say, okay, let's take these genomes. We can cut them into pieces. So for instance, in that case, we can take sequences of 12,000 kbps and then can kind of tokenize these sequences. So in our case, we decided to group the nucleotides by 6 to have 6 mers. There are lots of other choices that are possible. And we use a technique called mask language modeling or random masking to learn. So we mask 6 mers in the sequence, 15% for instance, and we train the model to reconstruct these mask positions. And we did that a lot on lots of data and investigate many effects. What happens if you just take the human reference genome? What happens if you take genomes from lots of different people? What happens if you add species? What happens if you make the model very big, smaller, etc. And kind of one of the first very cool results that kind of comforted us in the fact that it's interesting to try these approaches was if you, so in that slide I'm showing is the biggest model we trained on genomes from enough of different species. And after playing this game of reconstructing mask position in the sequence for a very long time, we took sequences for which we have labels. So our model has never been showed any label data. So it's just been shown sequences and need to reconstruct. We took sequences for which we have labels, so we know that some of them correspond to intergenic sequences, other to genes, UTRs, intron, exons, and so forth. We fed these sequences to the model. We looked at the activations at lots of different layers, and then we use UMAP or TSNE, for instance, to visualize. And what was very interesting for us is that we observed that actually the model, despite the fact it's never been seen any labels during training, actually got these concepts of some of the genomics elements. But even more interestingly, if you look across the layers of the model, you can see that it learned these concepts at different levels of granularity through the layers. So at the very beginning of the network, it can make the difference between instance coding and non-coding regions. And if you go further, like it learns much more granular representation and can learn, for instance, to distinguish also UTRs. And we've done in the paper lots of these different experiments trying to understand what has been run by the model. And we show that, for instance, looking at activation, but also attention map, you can capture these patterns. And then the second set of experiments we did was say, can we now use these models as starting points to solve lots of downstream tasks? So can we fine-tune them? And this thing has also been done now a lot in the field. And in other fields, kind of what was important for us in that study was to try to find kind of a systematic way to evaluate design decisions. So how can we know, is it useful? So for instance, if we care about human data and predicting tasks in humans, is it useful to add the genomes of lots of people? Is it useful to add the genomes of other species, even if we care about humans? And so in our paper that's going to be published in Nature Methods in the coming weeks, we decided to build this data set where the idea was to cover lots of different tasks. So maybe not the harder tasks in genomics or the most important ones for a field, but trying really to cover lots of different aspects of the field and having enough tasks so that we can compute statistics and do robust analysis. And so by doing that, we were able to validate lots of design choices. And so, for instance, we show that, as in other fields, increasing the size of the model actually helps in terms of performance, but also adding lots of different species, for instance, in the training helps a lot. And so what's interesting also with that methodology is that actually now with the final version of the paper, we had to change all the data from all the tasks. So we still kept 18 tasks. We're still trying to cover the same effect, but we completely changed the assays and the sources of the data we used to compute this analysis. And despite that, we got the exact same ranking between all the methods and the exact same conclusions, which also show the robustness of the analysis. And since then, there's been many groups, including our group, but others also who run benchmark on these models and kind of kept showing the same types of conclusions. Also, a cool fact I wanted to mention to kind of conclude this section about the nucleotide transformer was we also asked, so in that idea of building models that can understand biology and not only DNA, we also asked the question, can we use these models to understand proteins? So in making the assumption that you have the coding sequence of the protein. It has been done already by people who are trying to build models based on codons. In that case, people are trained data set of CDS or of mRNAs. But in our case, we're like, OK, our models have been trained on full genomes. So for instance, for humans, it's only 2% of coding sequences. There are introns between the exons, so the data distribution is pretty different from coding sequences. So we just asked the question, how these models are going to perform, and we compared, so we took hours or so DNA birth, and we compared to some models for proteins, including ASM. And we saw that on some tasks, as we could expect, protein language models are much better, especially on tasks where you need structural information. But what's interesting is that actually, and as it's been showed for codon models, on some other tasks, the genomics language model actually even outperform the protein language model. But more interestingly, is that we show that these two models are actually additive. So if for a protein task you have access to the true CDS and the amino acid sequence, you can take a model trained on genomes, can take a model trained on amino acid sequences, and combine them, even in a very simple way, and just fine-tune this more end-to-end. And you show that then on all of the tasks of the study, at least you get the best performance of both worlds, but in some cases you even get additive transfer. And so, for instance, for protein stability prediction, you even kind of get state-of-the-art performance by combining the two types of approaches. So now for a second part of this presentation, we'll show how we started from building these models with self-supervisioning and what are the types of things we can do with it. And so I first present quickly our segment approach, and then show also ChatNT, which is a very different type of idea. And in that segmented work, kind of the motivation to start this study, we say, OK, for now, we mainly looked at this problem of understanding genomics as an NLP problem. So, OK, DNA is a language, so we could use techniques that have been developed for language to understand it. But why not also looking at this problem as a computer vision problem? So in computer vision, people now also build very powerful foundation models. I'm thinking, for instance, about SAM, a segment and listing model from Facebook that's kind of understanding the world by segmenting objects inside. And so when you look at genomics, so it's not going to be a genomic sequence, not going to be a 2D world, but a 1D world, but kind of similar to a computer vision problem. You have a background, and on top of this background, you have elements. Some are bigger, some are smaller, and they're going to interact together at different levels. And so it made us ask the question, could we build, similar to what's being done in computer vision, a segmentation model that's going to run to segment these DNA sequences and kind of teach us genomics through that lens. So that's what we did in that study. So we decided to start to focus on 14 elements, some regulatory elements, some genic elements, and we've built this simple system where we take our nucleotide transformer that has been pre-trained using self-supervised learning, and on top we simply plug a UNet, which is kind of the state-of-the-art architecture for segmentation. It's been used for a while now in many applications, and we train that model end-to-end on all these annotations. So for one DNA sequence as input, we're going to predict at each nucleotide in the sequence 14 numbers, which correspond to 14 probabilities of belonging to the elements. In that case, they don't need to add to 1. Each number is going to be a probability between 0 and 1, so you can belong to several elements at the same time. The first interesting result was actually this task is not easy to predict. So if you just try the UNet directly, you can get to some performance, but actually there are many elements you don't manage to predict. Then you can make it bigger. You can also take the architecture with lots of transformer layer and stuff and try to predict directly. But what we show is that if you actually do this self-supervised learning on lots of genomes, the performance gets much better. So you already get some transfer from learning on genomes and then transferring to this annotation. Another interesting fact was there's been many models in the nucleotide transfer, in DNA BERT, in other papers developed to predict if elements, for instance, are unanswered or promoters as a classification task. So you give the sequence and it tells you yes, no. What we show is that if you actually take these models also and slide them over the genome, the performance degrades a lot. And it actually works much better to actually directly treat the problem as segmentation and predict it this way. Another interesting fact was you can also get transfer learning on the sequence lengths. So we did this curriculum learning where we first trained on sequences only of 3 kbp. So we took the genome with all this annotation, chopped it in 3 kbp sequences, trained, and then we started the training again on 10 kbp, 20, 30. We stopped at 30 for compute reasons, because as we use transformer, they scale quadratically with the sequence length, so you don't want to go too big. And we showed that kind of you can get this improvement in performance as the sequence input grows. But also what was interesting for us is that at inference time, if you take a model trained only on 30 kbp sequences, but you actually try on bigger sequences, you keep getting an improvement up to 50 kbp. So here is what it looks like if you do prediction as this 50 kbp resolution, where we predict like these 14 elements over a sequence of 50 kbp. So you have 700,000 predictions here. You could use all these probabilities and draw bounding boxes by using thresholds or other heuristic. We thought it would be interesting kind of to show the raw probabilities. And so here you can see, for instance, that the model can capture the genes, the different regulatory elements, and so forth of that sequence. And a final experiment before going to this chat-NT approach was we also tried to see the generalization capabilities of our model to different species. And interestingly, if you take the model trained only on human, it can actually get good transfer on species that are related. For some of them with almost no degradation. But you can also start training on more and more species, and the more species you add to the mix, the better the performance gets everywhere. Now to kind of conclude this talk, I would like to quickly touch down on a very different idea that you had with the group, and so hopefully one day we're going to mix the two, which was, can we give a voice to these models? So there's been now lots of work that looked at, for instance, models pre-trained in computer vision, and where can we kind of connect them to models trained in NLP, and for instance, get settings where you put an image and you start asking questions. Like you feed the image to the computer vision model, and then you ask questions to the NLP model, and it should be able to answer somehow. So why not doing that with genomics? So in that case, could we take a DNA sequence, put it through the nucleotide transformer, get some representation, and then pass that representation to Llama, for instance, and ask Llama questions. So in that case, we could do all the tasks that I showed previously, but in English. So you could say, is there a promoter in this sequence? Is there an enhancer? Is there a splice site? And you would train the model all the time to answer yes, no, or give you more details. You could even do regression tasks by training the model to output some numbers. There are two main motivations behind doing that. The first was try to make these models as accessible as possible. So now you go from a setting where you need to know how to code, or to import a model in a notebook, to fine-tune it, to analyze the output of the neural net, to transform that into predictions and so forth, where now you just have a chatbot. One day you could even directly discuss with that bot and ask all your questions. But the second main motivation for that work was also to say, can we get actually transfer also at the downstream level? So in what I showed before is that you get some transfer between this pre-training phase and this fine-tuning phase. But in what we've done so far is that we fine-tune on lots of other tasks, but there's zero transfer between all this fine-tuning. Like you get one fine-tuned model per task. And so if you have 5 tasks about promoters, you're going to get 5 promoters models, but they're not going to learn from each other. And what we showed here is that instead of building, for instance, 18 models for the 18 different tasks of the NT paper, you can build a single model that's going to be able to perform all the tasks by asking the questions in English. But not only you can retrieve the same performance with that single model and being interactive and so forth, but you can also get transfer. And what was interesting for us is that if you look at all these tasks on the right, they actually correspond to like detecting histone marks in the model. So in all these questions, there is the name histone that appears. And we showed that somehow the model managed to connect this concept of the histone world in what is actually an histone in the model. And thanks to that, we managed to improve even further the performance of the model while being interactive. We didn't stop with this 18 tasks. We also wanted to see if we can get further. And so we increased like, the pool to 30 tasks. So actually, the 30 do not include all the 18 for the first round. We kind of just really try to cover as much diversity and species as we can. We also wanted to look in that idea of building like models that can understand all the biology. We also looked at RNA tasks, also at protein tasks, making the assumption that you have the CDS. And we showed that the same model can do classification, but also regression, not only on DNA sequence, but also on protein sequence. So same model can give the CDS of a protein and ask, for instance, what is going to be the stability, what is going to be the melting point. You expect the model to reply some sentence and give you a number. And what I show is that we did this experiment 10,000 times. Each time we just retrieve the number with a regex, and we plot these values and look at the correlation with experimental data on the test set, and actually the model is very accurate. And kind of, yeah, to finish this talk, I just wanted to show this animation. So it's looking so far, and now we're trying building an interface that looks like ChatGPT, so you can just come and drop a DNA sequence or even several DNA sequences and ask some question that's related to that sequence, and the model should answer, and then you can retrieve the value from that model. So to finish this talk, I would like to thank all the people that made it possible, so at InstaDeep and BioNTech, but also our colleagues from Cornell Tech University, MIT, NVIDIA, and others. Right? Thank you for your attention.
All right, probably just one question so that we're not too behind on coffee.
Thank you for the great talk and a lot of great ideas. I just have a question. In an earlier slide, you mentioned that nuclear transformer has learned genomic knowledge without, in an unsupervised manner. I was wondering if there's a way to like reverse engineer and see like how the nuclear transformer has distinguished one group of sequences from another sequences.
Yeah, that's a great question. So I would say I see Amber smiling because now it's the kind of questions we're also asking. So I think there are lots of interpretability methods. So we don't have results so far, but yeah, that's kind of on the roadmap of the group trying to use interpretability methods, gradient-based, science maps to try actually to understand how the network can run these things. Yeah.
Okay, so we have, that was fantastic. Let's thank Thomas again. Do you want to say, if we have a 10-minute break, so I think that means we should be back. Oh wait, no half-hour break. So we can cut. Okay, so we're back again at 10:40. So yes, enjoy coffee and pastries. Okay, everyone, if you could take your seats, we're going to get started again. Okay, so we got a talk from three folks from Anshul's lab. So they're going to introduce themselves since there are three of them, but this is going to be telling us about how to evaluate regulatory genomics models. So thank you guys.
Hi, we're Austin, Aman, and Arpita, and we are presenting Dart Eval, which is a set of DNA language model evaluations that focus on regulatory DNA. Yeah, so self-supervised models, they've proven extremely useful at learning complex and contextual relationships, first for text, and also more recently for proteins as widely adopted protein language models. And this begs the question, can this approach also be applied to genomics? Perhaps a DNA language model or a DNA LM? Well, despite how recent the concept is, a lot of DNA LMs have come out. And in our study, we specifically look at whole genome DNA language models that try to capture a universal representation of all genomic information by pre-training on all of the DNA from one or more species. However, learning this universal representation is actually very challenging. So far, most of our success has been in protein coding regions, which are relatively dense and homogeneous and encode a coherent biological function, which is protein structure. However, these regions form only a small fraction of the overall genome, which contains several different types of functional elements, each representing distinct biological processes operating under a completely different set of rules. And this is further complicated by the predominance of neutral DNA, whose sequence is not relevant to biological function. And what's worse, all of these different types of elements are interdispersed as a big messy soup. But for Dart Eval, we don't really attempt to evaluate every type of functional element, but rather we focus specifically on non-coding regulatory elements, which encode the programs controlling gene activity. And a hallmark of non-coding regulatory DNA is its cell state specificity, where the same stretch of DNA can be read in very different ways depending on cell type due to the presence of different transcription factors and cofactors. And this context specificity makes regulatory DNA amazingly expressive, but it also poses a unique challenge to any attempt to model it. But unfortunately, the rate of model development has greatly outpaced the availability of robust evaluations, especially for regulatory DNA. First, there aren't that many benchmarks that examine context-specific regulatory activity, and moreover, they often only involve surrogate predictive tasks that don't address key downstream applications. For example, oftentimes we see that a DNA LM is fine-tuned to classify the type of regulatory element and solely evaluate it on the same task. But this misses the practical application, which is not to classify elements, but rather to reveal sequence motifs or design synthetic sequences. And lastly, they suffer from dataset design issues, such as the lack of compositionally controlled negatives or not accounting for LD when scoring variants. And to fill this gap, we introduced Dart Eval, which is an adaptable benchmarking framework consisting of a series of tasks that assess a wide range of regulatory DNA use cases for DNA language models.
All right, so now we'll go into some more detail about our modeling approach. And we apply these evaluations on 4 prominent and recently published DNA language models. And these are DNA BERT2, Jenna LM, Hyena DNA, and nucleotide transformer, who you just heard about in the last talk. And then for each task, we have some combination of zero-shot and supervised evaluations, along with rigorous baseline models. And so in the zero-shot regime, we look at the last layer embeddings of the models, along with their predicted sequence likelihoods for the sequences in question. And in the supervised setting, we do probing, which in our case entails freezing the pre-trained model and then training a small CNN on the last layer embeddings. And we also do fine-tuning using LoRa. And LoRa is a parameter-efficient fine-tuning method intended for large models where fine-tuning the entire model might be infeasible. And then for each task, we also have baseline models that are ab initio, or trained from scratch. And these are typically dilated convolutional architectures. So now let's go into each of the tasks and the results. So starting with our first task, which again is the easiest, and involves testing whether the models can detect any kind of regulatory DNA. And on a high level, this task involves comparing regulatory sequences to controls and determining whether the models can tell those apart in zero-shot and supervised settings. So going into this in more detail, the positive examples in our dataset for this task consist of approximately 2.5 million candidate cis regulatory regions, CCREs, as defined by the ENCODE consortium. And these are basically high-confidence regulatory regions. And our negative set consists of these sequences, but shuffled in a way such that dinucleotide frequencies are preserved. And this kind of shuffling means that the positive and negative sequences are compositionally equivalent and are not affected by confounders such as GC content or differences in dinucleotide frequencies, which has been a big problem with previous benchmarks. And the only difference then between the two categories of sequences is the presence of regulatory motifs, which is what we expect the models to detect. So we test this in two ways. In the zero-shot setting, we look at model likelihoods, and we expect the models to assign a higher likelihood to the regulatory sequences than to their negatives. And in the supervised setting, we train classifiers to differentiate the two classes. So first of all, our ab initio CNN baseline proves pretty effective at discriminating the two categories. And then if we look at the zero-shot setting first, we see that DNA BERT2, for example, is pretty competitive with the baseline, albeit a little inferior. And all the other models generally show competitive performance as well. And then when we get into the supervised settings, the DNA LMs even start outperforming the ab initio models. So overall, what we're seeing is strong accuracies across the board for this task. And I would argue that these results generally reflect the lack of difficulty of this task. And we see this one as kind of the preliminary bar that we have to clear before getting into the more complex evaluations. So speaking of which, our next task is intended to see if the models can graduate from recognizing any type of regulatory DNA to actually recognizing specific regulatory motifs. And we do this in a zero-shot setting with testing both likelihoods and last layer embeddings. So in more detail, we use a list of 1,443 transcription factor binding motifs as produced by the Hokomoko database. And we also have 100 of the shuffled background sequences from the previous task. And then for our positive set, what we do is we insert each of the motifs into each one of the background sequences. And then for the negative set, we do the same thing, except with shuffled versions of these motifs. So let's take a look at DNA BERT again, specifically in the likelihood setting. And here, we define an accurate prediction as one in which the model assigns a higher likelihood to insertion of the true motif than to the shuffled one. And then we see that model performance is generally effective, but not perfect. So the plot here shows the CDF for the accuracy of each motif, and the line for convenience is at 70% accuracy. And we see that around 20% of motifs exceed that threshold. So the model seems to identify a good number of motifs pretty well, but not all, and there is a large number for which the accuracy is pretty poor. And this trend carries over to the other models as well. Some are better than others, but all of them show pretty reasonable performance for a large number of motifs, with poor performance on some of them. And the numbers again show the proportion of motifs with over 70% accuracy. However, the last layer embeddings, on the other hand, seem to show very little information, if at all. So here, we define an accurate prediction as one in which the embedding distance from the neutral background to the positive is greater than the distance from the neutral to the negative. And all accuracies seem to be around 50%, which essentially indicates random predictions and is not really informative at all. So overall on this task, we see very mixed results.
Moving on from identifying specific transcription factor motifs, it is also important to understand cell type specificity. As we said earlier, a hallmark of non-coding regulatory DNA is its cell state specificity. So it's important to evaluate whether models can truly learn cell type specific regulatory vocabulary. To achieve this task, we use chromatin accessibility peaks measured by a TAC-seq for 5 cell types that come from very different regions of the body and have differential accessibility patterns. Moreover, each of these sets of differentially accessible peaks contain distinct regulatory DNA, including specific motifs that characterize them. We first evaluate models using zero-shot embeddings, where the model embeddings are produced on these differentially accessible peaks in each cell type. The baseline embeddings are produced by featurizing each peak as a vector of motif counts, and then these embeddings are clustered. We evaluate the embeddings in 2 ways, visually and quantitatively. Visually, we look at the UMAP of the embeddings of these sequences, and quantitatively, we perform k-means clustering and use mutual information to evaluate the clustering quality with respect to the true labels. Here, we can see that the bag of motifs baseline approach is able to identify modules that can separate by cell type. On the other hand, let's take a look at DNA BERT2. The model embedding clusters are not separable at all by cell type compared to the embeddings. And the mutual information scores further support what we see, with the DNA BERT embeddings having a much lower score compared to the baseline embeddings. The other DNA language models follow suit and are unable to classify cell types similarly to DNA BERT2 and perform much worse than the baseline both visually and quantitatively. So the models did not perform on par with the baseline in zero-shot space, but what if we help the models using supervision? This time, we will train a 5-way classifier on chromatin accessibility peak sequences coming from each of the 5 cell types. We measure the performance of the classifier using AUROC, and as we can see here, the ab initio baseline is able to classify cell types well with an AUROC of little more than 0.9. So what about the DNA LMs? Probe DNA BERT2 performs at a much lower AUROC than the ab initio, and fine-tuning we see improves accuracy but does not beat the ab initio. And similar to DNA BERT2, other supervised DNA LMs also perform worse than the ab initio at classifying cell types. And this is across all cell types. So overall, for this task, we see that most models barely perform on par with the supervised baselines, even after these much larger models are fine-tuned. So moving on, we want to evaluate whether models can actually quantitatively predict regulatory activity, which is an even more difficult task. Chromatin accessibility is a strong quantitative indicator of a sequence element's regulatory activity. If a model can successfully provide a quantitative measure of accessibility from sequence, the model can be used to provide a more nuanced understanding of non-coding cell type-specific regulatory syntax. This is a common classical task for which many supervised models already exist, but can we do better with DNA LMs? We evaluate for this using chromatin accessibility peaks from the same 5 cell lines as the previous tasks, along with their GC-matched inactive sequences. We benchmark these models using supervision, where we train models on the peak sets and corresponding negatives to predict the log DNase read counts over the input regions. And we evaluate performance using Spearman correlation between the predicted and true peak counts. The supervised sequence to activity ab initio model is represented by the dashed line here. The sequence to activity DNA BERT2 underperforms the ab initio in both probed and fine-tuned settings at quantitative regulatory activity prediction. And so do the other sequence to activity DNA LM-based models with both probed and fine-tuned performance well below the ab initio. And this happens again across all cell types for all DNA LMs. So most evaluations of models end at this surrogate task, but we cannot just rely on observational performance when evaluating models. It is necessary to evaluate models on true counterfactual tasks, such as our next one. For task 5, we benchmark models on this counterfactual task where the models are evaluated on sequence they have never seen before in their training set. This task tests whether models can predict effects of genetic variants in regulatory activity. This is the most difficult and biologically meaningful task in our benchmark. If models can accurately predict variant effects, we can determine how a single allele is influencing the regulatory readout. To test our models, we use a dataset of chromatin QTL variants present in African and European LCLs and utilize our sequence to activity models from the previous task trained specifically on the GM12878 LCL regulatory activity and not any variant datasets. To benchmark these models, we use rigorously curated variant sets. The causal QTLs are defined as those that are statistically significant and located within peak regions, thus drastically reducing the confounding effects of linkage disequilibrium. The control set consists of statistically insignificant QTLs that also fall within peaks. We first evaluate models in the zero-shot setting, where we measure the cosine distance between the embeddings of the reference and alternate alleles, with the causal variance expected to produce a higher embedding distance. So our ab initio model, again, is a sequence to activity baseline model from the previous quantitative prediction task. And it's important to note that it was not trained on any variants. We measure performance using AUROC, and as we can see here, the baseline performs quite strongly on both data sets. DNA BERT2 zero-shot AURC is well below ab initio at about 0.5, and other models perform similarly across both data sets. So next, we try to help the DNA LMs out by using the sequence to activity DNA LMs trained on the GM12878 LCL chromatin activity from the previous task to predict variant effects, again, not trained on any variants, and we compute the absolute predicted accessibility difference between the two variant alleles and use that as our metric. Unfortunately, supervision on chromatin accessibility does not help DNA LM performance either, and we can see that the DNA LMs display significantly inferior performance to the ab initio models across all settings. So this task demonstrates the necessity of real counterfactual tasks to determine true performance of the models.
Yeah. So to recap, we evaluate state-of-the-art DNA language models in zero-shot, probed, and fine-tuned settings across increasingly difficult regulatory DNA evaluations. And we find that although DNA LMs excel at simpler tasks, their performance degrades with increasing complexity relative to more resource-efficient ab initio baseline models of regulatory DNA. And all of these analyses we package into our adaptable DNA LM benchmarking framework, which provides 5 families of biologically motivated tasks and datasets that evaluate diverse aspects of regulatory DNA. And that being said, there is a whole world of other aspects of regulatory activity that we have not touched. These include long context effects, which we can infer from histone ChIP-seq or Hi-C. It also comparisons of regulation across species. And lastly, other types of functional elements such as UTRs and splice sites. And finally, it is clear from our experiments that DNA LMs have to get better before being practically useful. So we argue that the unreliable performance comes from the noisy and imbalanced whole genome pre-training data. So the lowest hanging fruit, even before optimizing for architecture, is to improve data curation and data training. And these can include training local short context models on specific classes of functional elements, such as promoters, 3' UTRs, and coding sequences, and adaptive sampling of elements to account for class imbalance, and explicitly conditioning on cell state context. But note that all of these strategies depend on some sort of annotation beyond just a whole genome sequence. And it is an open question whether universal DNA LMs can be built without the help of these annotations. Yeah, so thank you so much.
Do we have time for a few questions?
Hi, thanks for that great talk. I had a question on the choice of baseline. There was a paper here last year doing some benchmarking on regulatory elements, and it showed that non-neural models, so like support vector regressions or other types, can be competitive or even outperform things like deep-C or ab initio models. Do you have any further plans for additional baselining or any thoughts on ab initio models there?
Yeah, that's a great question. I think there are a lot of potential baseline models that can be used, but I think one important thing to note is that a lot of the models that have been used for baseline benchmarks in the past, like deep-C, for example, are trained on data that is not quite at the state of the art, because now we have really rich quantitative data for a lot of these regulatory readouts, and so I think the most competitive baselines are models that are directly trained on that. So some of the models that even have been published a few years ago now are not quite at the forefront, and it's important to make sure that we use supervised models trained on the most recent data. So I think similar to the dilated convolutional architectures we have here, and I would wager, we can totally try, but I would wager that those would beat more SVM type models on this type of quantitative data.
So I had a question for you guys that all the DNA language models, when they're pre-trained, they use some folds to train and test. But when they're compared with all other sequence expression models, for the downstream tasks, they don't use the same folds as the pre-training. So do you think a lot of their comparison could be leaked completely and they should be retrained using the same folds as the sequence to expression models?
Yeah, that is a great question. We definitely agree with that. For our purposes, we don't bother retraining all of the pre-trained models on a rigorous howed-out test data set, which does give them a little boost. So there is a possibility that the DNA language models here have the additional help of some amount of data leakage from the training set to the from the pre-training set to the test set. And we believe that this is probably a bigger issue for the easier tasks, like the CCRE classification, and probably less of an issue for the more of the counterfactual tasks. Yeah, and I guess in fact, for the variant prediction task, that is the only task where we guarantee to not have data leakage because none of the variants are present in the reference genome. Yeah, thank you.
Great. Let's thank Aman, Arpita, and Austin again. So next up, we have Anirban Sarkar, who's going to be telling us about designing DNA with tunable regulatory activity.
Thank you. Yep. Hello. I'm Anirban. I'm a postdoc from Peter Ku's lab at Cold Spring Harbor Laboratory. I'm going to present the paper titled Designing DNA with Tunable Regulatory Activity Using Discrete Diffusion. This is a joint work with Amber, Chris, and Peter. Transcriptional regulation requires the complex coordination of many proteins. This simplified diagram shows proteins interacting with DNA at cis-regulatory elements, also referred to as CREs, such as enhancers and promoters. And within a CRE, short sequence elements called motifs coordinate the binding of transcription factors to regulate gene expression. The rules governing this coordination are known as the cis-regulatory code. Decoding this complex language of the regulatory genome, that is the cis-regulatory code, is a major goal in genomics. And understanding the regulatory code is crucial for many downstream applications, including designing synthetic sequences with desirable properties. Supervised models have become the standard for regulatory genomic sequence analysis. High-performing supervised models can be used to design DNA sequences by treating them as a scoring function. In this way, designing sequences is reframed as a sampling problem. There have been a few approaches that have been proposed to sample local sequence space based on in silico evolution, gradient ascent, and simulated annealing. By contrast, generative modeling of DNA sequences offers the chance to globally sample sequence space, but it is still in its infancy. There have been attempts such as GANs, deep exploration networks, and more recently with DNA language model. Yeah. Generative modeling for regulatory DNA sequences is quite challenging. So while VAs, GANs, and LLMs work so well for protein sequences, they often struggle with regulatory genomics. That's because regulatory sequences are characterized by high variability in their positions, their lengths, and compositions. Evolutionary constraints are not imposed at the sequence level, but rather at the functional level. Diverse arrangements of strong and weak motifs can produce the same functional outcome, which means that regulatory DNA can drift quite a bit, so they are quite diverged across species. Second, the challenges of modeling non-coding DNA are further compounded by a significant class imbalance. Important regulatory sites are relatively sparse and scattered among seemingly random nucleotides. So when training generative models using reconstruction-based losses or even language modeling objectives, this extreme class imbalance of information means that the model is tasked with predicting uninformative nucleotides at most positions, which can only occur by definition via memorization. Third, the relationship between DNA sequence and cellular behavior is highly complex. While every cell in a multicellular organism shares the same DNA, cell types exhibit widely varying behaviors due to differential regulation encoded in the genome. And this encoding is complex and activated based on spatiotemporal cues. So generative models that focus solely on DNA for their training are unlikely to capture the full complexity of cell type specific cis-regulatory codes in regulatory DNA. Recently, diffusion models have emerged as a promising approach for generative modeling of DNA sequences. These approaches span standard denoising diffusion probabilistic models, latent diffusion, to modified diffusion models that better accommodate the categorical nature of DNA sequences. And the latest work in this direction is Dirichlet flow matching. However, the evaluation of these models have been on the lighter side, leaving open questions of whether these models are a significant advance in the generative modeling of regulatory DNA. In a standard diffusion model, there are two processes, a forward process that adds noise to an input sample and a reverse process that serves to denoise the data and generate a new sample. In denoising diffusion probabilistic models, the noise is given by an isotropic Gaussian distribution. In each step, a small amount of Gaussian noise is added to the image, ultimately leading to pure random noise. The reverse diffusion process aims to learn how to denoise the data, and the typical architectures are based on variational autoencoders. Score matching is an alternative formulation where the goal is to use a neural network we call S theta to approximate the score, which is the gradient of the log probability density function. Adding some tricks to this enable getting around the pesky normalization constant of probability distributions, which is typically intractable to calculate. Intuitively, score matching is the vector field that points to the higher density region. Score matching can be formulated in a similar manner to diffusion, where Gaussian noise is added, and there is a denoising process that uses the trained neural network S theta. Score matching has been shown to work really well for continuous data like images. Recently, score matching has been extended to discrete data by using concrete score. For a data point x in discrete space and another neighboring point y, the density ratios qy over qx are collectively known as the concrete score, which also can directly be modeled by a neural network. Again, we name it S theta, similar. This has been shown to work well for text data, and so we thought it would be suitable to explore for DNA sequences. I'm going to skip over the details of this, as it is quite technical, but you can find me to chat about them throughout the conference if you are interested. To assess the quality of the generated sequences, we come up with a wide variety of in silico evaluations. For functional similarity, we want to assess whether the generated synthetic sequences exhibit the same distribution of functional activity as natural genomic sequences. Conditional generation fidelity uses a supervised oracle to provide functional labels for synthetic sequences and matched genomic sequences, and then calculate the mean squared error. We match sequences by using the functional activity of the genomic sequences as a conditional value for the generated sequences. The Frechet distance is similar, but instead of predictions, it measures the distance of the distribution of penultimate representations from a supervised oracle, assuming a multivariate Gaussian distribution. Another way to calculate functional similarity is through the predictive distribution shift, which measures the largest gap in the cumulative distribution function of functional levels given by a supervised oracle for matched genomic and synthetic sequences. This is essentially the Kolmogorov-Smirnov test statistic. For sequence similarity, we use the percent identity, which measures the extent of memorization as well as the diversity of the synthetic sequences. But since percent identity alone isn't useful for regulatory sequences, we also consider the K-MER spectrum shift, which is the Jensen-Shannon distance between the K-MER distributions. And discriminatability assesses how well a supervised model can distinguish between synthetic and genomic sequences. This method is analogous to a GAN discriminator, but applied in a post-hoc manner. Ideally, we would like to see low percent identity and low K-MER spectrum shift, which indicates similar K-MERs within the regulatory DNA, but not through explicit memorization. Also, we want the discriminatability to have an AURC of 0.5, which means that the synthetic DNA sequences are similar to the genomic sequences. Compositional similarity aims to evaluate whether synthetic sequences contain key elements of regulatory DNA, particularly motifs and their grammars. First, we conduct standard motif enrichment analysis using a database of known motifs, and we also examine their co-occurrences. We also analyze functional motifs identified through attribution analysis, such as gradient shape, that allows us to assess the composition of functional motifs that might not be present in existing databases. Furthermore, we also deployed an attribution consistency metric to quantify the consistency of patterns within a population of attribution maps. We benchmarked D3 using an established dataset for diffusion models, which is based on the conditional generation of human promoter sequences. The conditioning is based on case-seq data, an experimental technique that measures transcription start site activity, which serves as a proxy for expression levels. Here, the sequences are 1 KB long, and the case-seq track is also 1 KB long. In this benchmark, previous diffusion models have been evaluated using conditional generation fidelity. The original study was Dirichlet diffusion score matching, in short DDSM, by the Zhou lab, and they incorporated other approaches including bit diffusion and D3PM. More recently, Dirichlet flow matching improved upon DDSM. Here, we further expanded the benchmark using D3 models, using CNN used by previous methods, and a transformer to see if we could do even better by using a more expressive model. Our results demonstrate that D3 with transformers produces sequences that are not only diverse, but also functionally similar to natural promoters with a lower MSE and similar predictive distribution shift. These synthetic sequences also exhibit motif distributions that closely resemble those found in natural promoters, but the sequences are not being memorized. This suggests that D3 effectively captures the complex patterns and functional characteristics of human promoters, at least through the lens of the supervised oracle. Next, we benchmarked the diffusion models using a fly enhancer dataset, which is based on the StarSeq assay from the Stark lab. It consists of 249 nucleotide long sequences, each associated with 2 scalar enhancer activity values corresponding to a developmental promoter and a housekeeping promoter. We trained both our D3 model and the Dirichlet flow matching, in short DFM, model using the same 2 base architectures, CNN and a transformer. In our approach, we simultaneously conditioned the models on both activity values. Our evaluation on the fly enhancer dataset yielded consistent values, consistent results with our previous findings. D3 generated sequences exhibited a higher efficacy in their function. They exhibited the same composition as natural sequences, and even a downstream discriminative model could not tell them apart, as indicated by an AURC close to 0.5 in the discriminability test. Together, this reinforces the effectiveness of D3 in capturing and generating regulatory genomic sequences. Next, we investigated whether the synthetic sequences provide additional information that can benefit a supervised DNN as a data augmentation. To test this, we systematically reduced the size of DeepStar training set and then added synthetic sequences generated by different diffusion models. The results were encouraging. Even when using the full training set, adding synthetic sequences from D3 improved the performance of a freshly trained DeepStar model. This suggests that D3 synthetic sequences provide greater genetic variability while still encoding meaningful cis-regulatory information. Moreover, the benefits of augmentation with the D3 generated synthetic sequences were even more pronounced in the small training data regime. Next, we investigated the model's potential to generate sequences with task-specific activity levels. We tested their ability to generate sequences with predetermined activity levels for 2 different tasks, developmental and housekeeping promoters. We looked at different cases with same activity levels and high activity in one task and low in the other. The results show that our D3 TRAN excels at generating sequences that closely match the intended activity levels, irrespective of whether the activities are similar or different between tasks. This indicates that D3 has captured meaningful biological patterns and can apply them to create synthetic sequences with desired properties. In this study, we extended score entropy discrete diffusion to DNA sequences and performed extended evaluations of the generated sequences. However, many of our evaluations rely on in silico oracles to provide levels of generated sequences. This just evaluates how well the generative models align with the supervised oracles. So if generative models learn cis-regulatory codes that are better than supervised oracles, this cannot be properly assessed. Also, we are largely interpreting the cis-regulatory codes from synthetic sequences through either existing bioinformatics tools or attribution analysis using a supervised oracle. If the generative model is learning better or more expansive cis-regulatory codes, we cannot assess this. Another limitation is that biology often lives in a small data regime, and it's unclear to what extent diffusion models can work well in such regimes. We are actively exploring this as well. Moving forward, we have several open questions. What benefits, if any, will generative models for DNA provide over the more prevalent supervised models that have already been proven to be quite useful? What new applications can generative DNA models help accelerate? We don't necessarily have good answers for these open questions yet, so if anyone in the audience has any good ideas, I would love to hear what you think. I would like to thank everyone in the lab for short and long technical and non-technical discussions at some point that improved my understanding on diverse topics. A special thanks to Amber and Chris for their great help in this project. Finally, I thank Peter for constant support, guidance, and encouragement during my time at the lab. My fellow postdoc, Jessica, will present a spotlight talk on genomic deep learning with knowledge distillation this afternoon with poster number 6 and ID 60, I guess. Please check them out. Check that out if you like. Yeah, thank you.
And we have time for a few questions.
Could you expand on how you measure memorization?
Okay. Let me. So actually, we calculated. We actually measured memorization through percent identity, and this is basically dot product between the generated sequences and the true sequences. And while this is not very good representation of like quality measurement of the generated sequences, but this is something. Yeah.
Hello? If you shift something by one nucleotide, would that be captured?
Come again, please.
If I shift a sequence by one nucleotide, would it be captured in this metric?
Yeah. It should be captured. So it captures. So it doesn't have the biological insight. This particular measurement doesn't have the biological insight, like what should be, what is actually changed compared to the true sequence. But this actually captures even one nucleotide change.
Sorry, I meant if I shift the whole thing, like is your metric alignment based, or is it more accurately described as a product based?
It is product based, yeah.
I guess my question was going to be, how much have you compared to the other way of doing the generation, which would be the more NLP way with autoregressive models, and do you see those being competitive with diffusion?
Yeah, so actually this particular method was applied for, or shown to give good results for text. And this particular, so for text, this particular method was compared with GPT-2. So I would say diffusion-based models are still lagging behind compared to the DNA language models. But this is a different way to understand the sequences and generate new sequences. So, people are working on that. And recently, people started working with flow matching. And I would, personally, I would be very interested to see what is coming up. Yeah.
Thanks for the talk. Very interesting. I had a question regarding whether you tried, I guess you tried this for conditional generation too, right?
Yes.
So I'm trying to understand when this kind of approach goes out of distribution without being easy to detect. So often, if you ask these kinds of models to generate sequences that are simply impossible, right? Like very, very high activity that's effectively not possible at all, or not at least in distribution in the data you trained. Have you noticed that these models also do that? And if so, is there an easy way to detect when they go out of distribution? Because that is, I think, more important. It's not that everything will go out of distribution at some point, but is there an easy way to detect if that happens?
Thank you for the question. This is really interesting question. And actually, we tried to see with very high activity levels, and tried to see the quality of the generated sequences. But probably the generator sequences they have like some motifs. They have like too many occurrences of very important motifs, which might not represent the original sequences. So that kind of stuff we kind of noticed, but we actually tried to understand, like by removing some motifs and all, like how they interact and what actually matters to give you that kind of high activity. So, but this is something we also want to explore. Yeah, thank you.
Great. Let's thank Anirban again.
Thank you.
So next up we have Akshat, Singhal from UC San Diego. This is HDMI, I guess, is it? Yeah.
Okay. Hi everyone, my name is Akshat Singhal. I'm a PhD candidate in Dr. Triadica's lab in the Computer Science Department at UC San Diego. And today I'm going to present our work titled A Deep Learning Model of Tumor Cell Architecture Elucidates Response and Resistance to CDK4/6 Inhibitors. I'll start by defining the class of models we use in our work. Cancer drug response prediction models aim to improve cancer treatment outcomes by customizing therapeutic decisions for each patient based on all available information, genetic, molecular, cellular, and clinical. The three basic components of a model, the input, output, and the method, in the context of drug response prediction models are as follows. So the first is input, which is tumor samples here, and we have, in terms of the data types used, we use genomic data such as gene alteration profiles, transcriptomic data, histology images, radiology images, and even other types. The sources are generally cell lines, patient-derived organoids or xenografts, or patient data itself. The method is typically a supervised learning approach. Earlier, simpler methods were used. The simultaneous advancement in high-throughput drug screens and machine learning allowed for more complex modeling techniques. These days, deep learning models are the most common, and maybe in the very near future, we'll see foundation models being added as the first step. The output is some phenotype associated with drug response. It could be IC50 or area under the dose response curve, progression-free survival or overall survival in patient data, or the RECIST criteria for PDXs or PDOs. Even in our models, we use all these three output types based on the data used. So our primary aim in building these models is to make them clinically applicable. But what's needed to make a model clinically applicable? As I understand, there are four core properties, such as, so first is robustness, which is accuracy on diverse data sets, regardless of what the model was trained on. Translation, which is accuracy on clinical data. It's a separate point from robustness because the clinical data is specifically very difficult to predict. The patient data has a lot of heterogeneity. It's not easily available. We don't have the same features, or even the output labels are different. The third is interpretation. The model should be able to explain the underlying factors of the predictions. I will expand on this later. Validation, which is rigorous validation on both predictive and mechanistic accuracy. Okay, so interpretability in a drug response model adds a lot of utility because if a clinician is using the model, they can better understand what's happening inside the model. Now, there are several ways to make a model interpretable. I will go over the method that we have used in our work. And to start off, let's suppose we have a deep neural network without any additional information. It's a black box. A simple change is to make neurons represent some biological entity, such as a gene or a complex. And that's what we do. We map a neuron to some biological entity. For our models, we use hierarchical acyclic graphs of protein assemblies. I'll explain what hierarchy means here. Essentially, we are guiding the neural network architecture through a biologically informed structure. So a hierarchy of protein assemblies is a tree-like structure. I don't know if you can see the, maybe 1 second. Okay. So a hierarchy of protein assemblies is a tree-like structure where each node is a cluster of interacting proteins which integrate at multiple levels to create the hierarchy. These genes or proteins cluster together to form small assemblies, which merge to form bigger assemblies all the way up to the root of a hierarchy, which represents a cell, in our case, a cancer cell. In particular, we used a hierarchy called NEST, short for nested systems in tumors, which contains about 400 assemblies, integrating about 19,000 proteins. So using NEST as the underlying structure, we created visible neural networks, or VNNs, for predicting tumor response to a drug. The concept of VNNs was introduced in a previous work from our lab called DCell, and it was also used previously for drug response prediction in DrugCell, but in a different kind of architecture. So since the aim of the models is to be clinically actionable, we curated gene list from clinical panels like Tempus and Foundation One, because it's what's most commonly available in patient data. We collected training data from publicly available resources such as CCLE and high-throughput drug screens such as CTRP and GDSA. Next, I will go over the architecture of the VNN. So the genes, gene mutations, and copy number alterations form the feature layer of the model. Each feature is in the binary format, where 1 means alteration and 0 means wild type. These genomic features combine to form the gene layer, where each gene is represented by 1 neuron. Genes merge together to form several assembly layers, which finally end at the root, representing a tumor cell. Each assembly is represented by multiple neurons to allow the model to capture complex interactions within any assembly. The number of neurons here is a hyperparameter, so it can change. Okay, so having designed the models, we next trained several VNNs. All the models were trained and tested using nested cross-validation, where the cell lines used for testing were never seen by the model during training. The figure shows a performance comparison of 41 such drug models against 2 methods, random forest and a black box neural network. The black box neural networks have the same number of neurons and layers as the VNNs, and the random forest was used with standard configuration. We observed that most drug VNNs outperform both benchmarks, with random forest being a very close second. Next, I will go over the model for palbociclib, or as I'll call it, palbo-VNN, which is the focus of this paper. So palbociclib, that's the correct pronunciation. Palbociclib is a targeted therapy which stops cancer cell proliferation by inhibiting CDK4 and CDK6. So CDK4 and 6 bind to cyclin D to phosphorylate RB, which ultimately makes the cell line or cell cycle progress from G1 phase to S phase. In many cancers, this mechanism is uncontrolled, which is prevented by the CDK4/6 inhibitors. So palbociclib is currently a standard of care in ER-positive, HER2-negative advanced breast cancer, along with endocrine therapy, and has markedly improved treatment outcomes. However, overall response rate hovers around 50%, and the development of resistance is nearly universal, which underlies the need to better understand the drug's mechanism of action. So onto the results. The first analysis was to measure the predictive performance of the palbo-VNN model on held-out cell lines. The model was highly accurate on confidence samples, which are like outside of the first standard deviation, and it achieved a performance accuracy of odds ratio of 40.1. Next, we applied the model to ER-positive, HER2-negative advanced breast cancer cohort from the Gini database. The patients were separated into 2 groups. The first group of 67 patients were treated with endocrine therapy and a CDK4/6 inhibitor, and the second group was of 159 patients with only endocrine therapy. We observed that the model is able to stratify patients in the first group, but not in the second group. Notice the difference in the blue and the orange curves. So we see a stratification here, but not so much in the second group, which means the model is specifically predictive of CDK4/6 inhibitor response. Motivated by these performance results, we interpreted the model to identify important assemblies. The model highlighted 33 assemblies covering diverse mechanisms such as cytoplasm, nucleus, and signaling pathways. Overall, there were two major observations. The first, PARBO-VNN captures known drug targets. So 8 out of 9 assemblies that contain CDK4 or CDK6 were detected to be important by the model. Second, 25 assemblies that do not contain CDK4 or 6 were also captured by the model. Several of these, such as histone-mediated transcription regulation, adherence junction, EGF-FGF simulation of cell proliferation, and even immune system, are not commonly associated with palbociclib response. In a subsequent analysis, we focused on one assembly that governs palbociclib resistance. So NEST85, or histone-mediated transcription regulation, is a densely connected assembly of histone acetylases, deacetylases, and transcription factors. PARBO-VNN predicts that copy number amplification in KAT6A, TBL1XR1, and several other genes leads to resistance in palbociclib. The resistance odds ratio is about 2, which indicates that cells harboring these amplifications are twice as likely to exhibit resistance to palbociclib than cells without these amplifications. To validate, we measured the number of cells entering the S phase or cycling under various conditions. So in the non-targeting control or the base setting, we see a decent number of green, which are EDU positive cells, which means the cells are cycling. In the overexpressed setting for each of the genes, we still see a lot of green, which means the cells continue to cycle. But when we add palbociclib to the control setting, we see almost no green, which means cells stop cycling. On the other hand, the overexpressed cell lines or the media in with addition of palbociclib retains ability to cycle. Overall, we observe that KAT6A and TBL1XR1 produce significant increases in the proportion of cells entering the S phase under palbociclib treatment compared to the untreated group. So my key takeaways from the work are interpretable drug response models can provide deep insight into the mechanism of a drug. The VNNs do not lose predictive accuracy. PARBO-VNN accurately translates to clinical data, and the model can capture both known and novel assemblies of drug response. There are several next steps that we are working on, but I would like to highlight one that we are currently working on a real-world analysis of 139 ER-positive, HER2-negative advanced breast cancer patients treated with palbociclib at UCSD. And the preliminary results have come out to be very promising. According to us, the model was able to stratify the 75 patients treated with palbociclib in the first-line treatment with a difference of 30 months in PFS at median survival, so 50% survival, and it gave a hazard ratio of 0.63. A lot more results to be analyzed, but so far it looks good. Okay, so I would like to thank my PI, Dr. Treidekar, first co-authors, Dr. Sungjin Park, and Dr. Erika Silva, all the co-authors, the funding sources, and last but not the least, the Idekar Lab. And I'm also presenting a poster later today titled The Hallmarks of Predictive Oncology, which are guidelines and standards for building predictive oncology models. This is the code repo for the PARBO model and all other VNNs, the single drug VNNs, and QR code for the paper, and the media to reach out. Always happy to take feedback. Thank you.
Thanks, that was a great talk. I think these seem like really exciting models, but I guess one reason deep models haven't been used as much is because it's hard to get sufficient data to train them. Can you comment on roughly like how much data went into your model compared to the number of parameters, but then also what are the prospects for this field to really get massive amounts of data for this kind of problem?
Open your own company. So yeah, so for cell lines, we had about 1,244 cell lines, which is all we could get from publicly available resources. This is the biggest public cohort that's available. GDSC CTRP, so CTRP is by Broad, GDSC is by Sanger. There are high throughput drug screens that's available to us. Other than that, the real, I guess, validation data sets that come from patients is very limited. Genie provides one of the biggest resources, but it's not the Genie with 100,000 samples because they don't have matched outcome. The matched outcome is like 200 patients, 70 patients, something like that. Although all the companies like Tempest, Foundation, Caris have more than 100,000 patients, but this is a very valuable data set, so they're not publicly released. But yeah, we're trying to see how we can leverage. Hospitals are much more happy to share their data with research folks, so we're trying those angles as well.
Hi, very quick question. So I was just wondering about your comparisons of the VNN to the black box ANN. And you mentioned they have the same number of neurons and the same layers.
Yeah.
But do they have the same connectivity? So the way I see it is that the VNN is sort of effectively regularized because you are removing a lot of edges. Now, what if you kept the edge density the same also? Because that's kind of you just randomize the edges, but you keep the density the same.
Right. So the evaluation here was to actually check if a fully connected network would be better than this regularized version, right? Is the sparsity helping or not? Because, so we think because the data is so sparse and so less, the sparsity is actually helping the model perform better. But we also did shuffled versions, but we did shuffled versions only for the palbociclib because we had to shuffle 500 times, so which is very intensive. So for the one drug, we did 500 shuffles, and we see that the model is better, but we also did that more for our interpretation validation, so we found out some statistically significant assemblies. But yeah, we shuffled the gene connections randomly.
Yeah, I guess the reason I'm not surprised that the VNN does better. The question is whether you can believe the flow of information through the VNN, right?
Right.
That's the only question.
Right. Yeah. I guess an ideal approach would be to do, some kind of ablation study, but it was turning out to be too heavy for us, so we didn't. So we tried a simpler approach where we just shuffle the genes connection to assemblies, and we found that our version was better.
I guess, is it true that a given gene or protein could be represented multiple times if it's in multiple assemblies? And I guess if that's true, then there's some proteins that are very multifunctional and they'll be overrepresented. I guess, do you have a sense that are those then multifunctional proteins driving the results? Or, okay.
So, yeah, so for example, I showed this result here. We see CDK4 or CDK6 available in multiple assemblies, also P53. P53 is very common across multiple, but very surprisingly, P53 didn't turn out to be important for our model. And it has been shown before that P53 is not a driver, or P53 mutations are not a driver of response or resistance in palbociclib early on. Later on, it becomes important, but the data doesn't have that, so we don't see that.
Okay, I had two questions. One question was asked by Anshu. The other one is about the just a high-level idea of using this biologically interpretable, inherently interpretable architecture. You're using prior information.
Yes.
How much does it depend on that? I mean, you may be reducing the chance that you will find, new pathways or new organizations of these, genes or proteins.
Yeah. So the first model, drug cell, used gene ontology Go, which is much bigger, and we also compared it with Go. But Go is manually curated and not specific to cancer. So we came to NEST, which is specific to cancer and built in a data-driven fashion. And we observed slightly better performance. It was not statistically significant. The other is Reactome. Reactome is also very good. Of course, I think the better or the best way is to make models for everything. But then I don't know how to actually interpret it, and it becomes too complicated. So we kept to nest, and so far we have done only one in our paper. Yeah.
Great. Let's thank Akshat again.
Thank you.
So I think lunch is ready. Thank you to all the speakers for keeping very precisely to time. Very appreciated. So we actually get lunch on time.
Hi, everyone. So we're going to get started with the spotlight session soon. There's, I think, 8 of you-ish that are doing the... It's not on. Mic is not on. You're a spotlight?
Yes.
You're the first one. I'm going to ask everyone first to line up, and then once everyone's there, then we'll Hi, everyone. We're going to get started with the spotlight session. So I think 8-ish talks that are 3-minute long. If you're presenting a spotlight in this first session, please have a seat on the front row so you can all come when your turn is up. So maybe I can get the first presenter to come, and the slide is here.
Okay, great. So, okay. Should I just start? Okay. So, good afternoon. My name is Francesco, and I'm here together with my colleague Vamsi to present our work on full-length ribosome density prediction from sequence and graph-based representations. So, the ribosome density prediction task involved using some AI model on sequences, and we saw today already many of those, to predict, starting from an mRNA sequence split in codons, the codon-wise ribosome density. So the ribosome density is telling us where the ribosome is stalling in the sequence. And knowing this is very useful because it leads us to better understand translation elongation. And in turn, this can help us in doing, for example, codon optimization, in which we make the ribosome stall at the position that we want. So our work distinguishes from the previous one, since we are trying to consider both the mRNA sequence and the mRNA secondary structure. So if we consider our codon-wise sequence, essentially we are adding structural neighbors, or jumps, between the sequence corresponding to the edges in the secondary structure. So our method is named RiboGL, and is a combination of a graph and recurrent neural network. So we learn both graph embeddings and embeddings through the recurrent neural network. And our method performs best on our dataset of mouse liver ribosome density, ribosome profiling data against the current state of the art that is named RiboMimo, that is just a recurrent neural network, so based only on the sequence. And our ablation study showed that still the sequence is giving most of the information. And this, I think, is a very interesting task since we have essentially a graph overlapping a sequence. So these kind of tasks are very interesting, I think, widely represented in biology. So I think our work can lead to similar results in other tasks. And so while our method performs similar to a sequence-based model, we can see from the interpretation extracted from Cap2 that we can get longer interactions, so longer interactions. And so if you consider, for example, the sequence above, where the A site is AAG, so the ribosome stalling we are considering, and we can get where is the most important codon leading to this prediction, we can check the distance. And when we do this on a BiLSTM against our model, we can see that the BiLSTM is not able to capture longer age dependencies as our RiboGL is able to do. So we provide also a visualization tool to see the result at the mRNA sequence level, and that is available on our GitHub repo. And we are waiting for you at the poster 1 to discuss some future direction on graph-based modeling for ribosequencing data. Thank you.
Okay, so hello everyone. Our group worked on investigating investigating data leakage when using protein language models. So I'm sure you've already heard of data leakage, but I will give you a quick recap. So data leakage happens, for example, when you use a pre-trained model, for example, a protein language model for a downstream task. And then the pre-trained data set and your data set might share samples. If these samples, which the protein language model was trained on, are also in your test set, which you might not know about, this could result in unrealistic performance in the evaluation later. We decided to test this on thermostability prediction from ESM2 protein embeddings on the Meltome Atlas dataset, which contains proteins and their respective melting points. And we tested this on 2 different splits. One was the flip split, which doesn't consider the protein language model pre-training. You can see right here that the protein cluster C is contained in the ESM2 pre-training set. So that could lead to to improved results, which are due to data leakage, not due to your model being generalizing better. Then we also created our own split, which is called the EPA split, which removed all proteins of clusters that ESM2 was trained on from the test set. So that split is data leakage free. And we tried this out for a number of variants of ESM2 and also ESM1B. And we see a significant increase of 11% on average. And the experiments on the flip split outperformed the experiments on the EPA split on all architectures. And what we conclude from this is that data leakage seriously distorts the measured performance when you don't think of it when constructing your dataset splits. And we also think that other downstream tasks are affected, but this probably needs to be proven with further research. So I hope this reminds everyone to check their dataset splits when using pre-trained protein language models, because this problem doesn't really get noticed if you don't know about it. And if you want to discuss this further and are interested in additional information, you can look at our preprint or at our poster number 2. Thank you.
Hello, everyone. Let's meet PathoLM, the model we built for identifying pathogenicity from DNA sequence using the Genome Foundation models. So first, what is pathogen? So pathogens are organisms like virus, bacteria, fungi, those spread disease among human and animals. So, sorry. Okay, so by identifying pathogens, so there are species for whose some strains are pathogenic and some strains are not pathogenic. So our objective was to build a model that can well distinguish the pathogens from the non-pathogenic sequences. So our key focus are limited to the most clinically important escape pathogens, including E. coli, Enterococcus, and some other species. And for virus, we took coronavirus, norovirus, influenza virus, and those from NCBI and Petri datasets. Okay, so what are the prior works on pathogen identification can be categorized in 2 parts, alignment-based methods and ML-based methods. So the alignment-based methods are computationally very heavy because they relies on the known database and sequence search. The ML-based methods resolves this problem by a large margin, but they need manual data curation and feature extraction. So what we did, we used pre-trained genomic large language models to fine-tune for pathogen identification task. And one of the important part of our project was to manually curate the pathogen data sets because it's limited and not very well distinguished because which strains are pathogenic and non-pathogenic. This decision was very important. And we fine-tuned nucleotide transformer version 2, 50 million parameter version, and solved the problems of minimal level data sets. using the pre-trained knowledge. So why nucleated transformer over other DNA language models? So for answering that questions, we experimented with zero-shot and few-shot learning. So as you can see that nucleated version 2, transformer version 2 performs moderate, but DNA-BERT-S performs very good on zero-shot learning, but the learning curve for nucleated transformer version 2 was higher than the other DNA language models. So that was one reason, and another thing was it is computationally less extensive than the DNA-BERT-S. So one of the goal was to build a lightweight model for pathogen identification. And we benchmarked with other models, DCI Patho, and we outperformed using in a large margin for all the evaluation metrics. And we also extended our PathoLM for species level classification, and as there is no model built dedicatedly for species classification, we compared with traditional machine learning models and deep learning models and showed that our model outperformed all of them. And also, we also showed the performance variation for varied sequence length, and the blue line, you can see PathoLM-SP outperformed for short read and both long read sequences, and outperformed all other methods. Yeah, for more details, please visit our poster presentation. Thank you.
Okay, hi everyone. I'm Adam. I'm a PhD student with Charles Danko over at Cornell, and I'll be giving you a quick introduction to some of our work on training deep learning models on personalized genomic sequences. So I'm sure all of you are aware, there have been some pretty spectacular advances in sequence-to-function models over the last several years. But one of the main challenges, I think, to these models is that predicting the effects of non-coding variants remains quite challenging. And this is highlighted by these two recent papers benchmarking the Informer model. And one potential solution to this problem is to incorporate genetic variation into the training data sets. This strategy has been used by a number of different groups, many of which are represented in the audience today. And so our sort of approach to this was to train the ClipNet model, which I think you all heard about this morning. So what this does is it's a CNN-based model that predicts the transcription initiation signal around enhancers and promoters at single nucleotide resolution. And the way that we trained this model is we used matched genomic and ProCap data sets from about 60 genetically distinct LCLs. And in our paper, or in our preprint, we showed that this model does reasonably well at predicting both across loci and across individuals. And so this result sort of naturally raises the question of how many N do we actually need? What's the relationship between the number of individuals in the training data and the performance, particularly on variant effect prediction? And so what we did was we subsampled the number of individuals that we use in the training data set and then evaluated both the cross loci and the cross individual performance. And to cut a long story short, what we found is that the more individuals you feed into the model, the better it performs at both tasks, which seems pretty sensible. Another issue that we wanted to tackle was whether these sort of benefits that we gained from training in this sort of variant-aware fashion can be retained after fine-tuning to a novel cellular context. So what we did is we took our ClipNet models that were trained on all 60 individuals and then fine-tuned it to a ProCap data set that was collected in K562. And then focusing specifically on the variant effect prediction tasks, we then benchmarked our model as well as ProCapNet, which is a similar sort of CNN-based model, as well as Informer, on predicting the effects of variants in a K562 MPRA. And what we found is that our model outperformed both of these two models, which I'll note were natively trained in K562. And so I'll finish by arguing that training models on genetically diverse data sets can improve variant effect prediction. Thanks, and I think based off the order, I'm poster number 4, but not entirely sure of that yet.
Sorry, we don't really have a strict poster ordering order. We'll see, but hopefully people can find each other. It's not that big of a poster session.
Thank you. Hi, everyone. My name is Ray. I'm a PhD student from Christian and Leslie's lab from MSK. Today, we're going to introduce Hi-C2Self, a self-supervised Hi-C contact map denoising model. So this is the general framework. It's very simple. There are 3 different components. First, since this is a self-supervised model, we have the input matrix as this low-coverage input matrix. They're used as both the input and the training target. And then the second, we use a mask to achieve the self-supervision training. And then at the end, we design this 2-channel output, and then so that we can use the negative binomial loss so that the model can directly train and run on the row count matrices. We explored the model in 4 different use case scenarios, 2 on the bulk and then 2 on the single cell. First, on the bulk library denoising, we can see on the top is the low coverage input data of the Hi-C, and then this is the Hi-C2Self recovered results, and then this is the deeply sequenced model that the deeply sequenced library that the model has never seen. And then the second, we also explored our model onto micro-C data at 1 KB resolution. And then we can see on the top, this is a very lowly sequenced micro-C library. And then in the middle, we can reconstruct some of the structures. And then at the bottom is a deeply sequenced library that the model has never seen. We also cause significant interactions, and then showing the ROC curve over here. In addition, we also tried the model on trying to reconstruct some of the structures for the single cell Hi-C. First, for many of the single cell Hi-C, they actually have the co-assay which has for example, they have a a single cell attack-seq and a single cell Hi-C. We can first bin kind of cluster the cells based on the attack-seq, and then try to explore and reconstruct the structures on the pseudobulk of the Hi-C. So here, for example, we show a 50-cell pseudobulk at 10 KB resolution around the local gene, and then we can successfully reconstruct this unique structure in the cell stage. And then at the end, we also try on the single cell data. And then here we are showing at 50 KB resolution. Here are 2 random cells from 1 cell from a lowly methylated region from a gene, and then this is a highly methylated cell. And then we can see the model can reconstruct the different structures. We also try to cluster cells based on their local structure around this gene, and then color by the methylation level. The 3D structure reconstruction shows consistent pattern with the methylation. And then here is the model. We have 4 different applications, and then for more, please go to our poster number 5. Thank you.
Hi, everyone. My name is Aniket, and along with Daniel and our advisor, Nyla, we have been working on, what's going on? Okay, we've been working on conditional diffusion models for generating stable mRNA sequences. So as we all know, mRNA therapeutics have been a very promising field of research, but currently they're held back by certain biophysical properties such as stability. And current approaches to optimize for stability mostly concentrate on optimizing individual components of the mRNA sequence, a common example of this kind of approach being codon optimization. But they seldom consider the full sequence during this optimization. So to alleviate this sort of problem, we use diffusion models, which can actually look at the full sequence during sampling. So what we do is we train conditional diffusion models, which take in the full mRNA sequence as an input, but we condition on 2 types of stability quantifications, half-life and minimum free energy. Half-life quantifies the rate of decay of an mRNA sequence, and minimum free energy, or MFE over here, is like a thermodynamic measurement of the mRNA sequence's stability. By conditioning on these properties during training, during sampling, we're able to provide a desired value for each of these properties and generate corresponding mRNA sequences.
So generally in mRNA sequence design, we're interested in actually fixing the amino acid sequence for a desired function. So we reflect this in our analyses. This is showing 65 randomly sampled genes from our training data set, and then we fix the coding sequence and then use our diffusion model to optimize the 5' and 3' untranslated regions to make more stable mRNAs. On the left, we have on the x-axis the original sequence predicted minimum free energy, and on the y-axis is the improvement or change from the original MFE. Here, lower is better, and the blue dots are showing the predictions for our diffusion sampled sequences, and generally we see improved minimum free energy for these mRNAs. On the right, we have a very similar plot, but now for half-life, and we see a similar result where our diffusion sampled sequences are able to improve the predicted half-life. Further experiments have shown that we're not able to modulate the half-life with as much granularity as we are with minimum free energy. And if you're interested in this, definitely come check out our poster. And we're actively looking for interesting data sets that might help us with this task and collaborators.
Hi, everyone. My name is David Lau. I'm a PhD student at UC San Diego. Oh, you're ahead of schedule here. OK. I'm here to talk to you about my tool called GenVar Loader to apply sequence models to personalized genomes and tracks. For anyone who's not familiar with sequence models, they can, for example, be used to take DNA sequences input and then predict the read depth corresponding to RNA-seq or attack-seq experiments. Generally, they have only been trained on sequences from the reference genome, but recent work, many of which has even been talked about a few minutes ago, has shown that this approach really fails to capture the effects of genomic variants, and furthermore, that training on personalized genomes can really help. All right. So in order to do this, we'll need to change the reference genome and read depth to reflect those genomic variants. Unfortunately, existing methods to do this generally, they were not thinking about training neural networks when they designed them. So for example, BCF tools requires writing individual genomes to disk. And if we scale this up to like thousands of individuals, we're looking at terabytes or petabytes of storage required just to generate this data. Instead, GenVar Loader is going to write those variants to a sparse memory mapped format and reconstruct those personalized genomes on the fly while also still including the indels. So as a result, GenVar Loader requires hundreds to thousands of times less storage than those FASTA files and enables training on thousands of individuals or more. So for example, the 1,000 Genomes project, there's 3,202 individuals in that. Writing all those FASTAs out would require over 6 terabytes of storage, whereas GenVar Loader requires just 3 gigabytes. And this is all while still maintaining a lot of performance. So that GenVar Loader is over 1,000 times faster than reading the FASTA files, regardless of the size of the data set. In fact, it's so fast, it even exceeds the CPU to GPU transfer rate for state-of-the-art GPUs like NVIDIA A100s. And by also memory mapping track data, like from bigwigs, we get similar performance there as well. So what does this all mean is that during training, you can get really fast training speed-ups because you've eliminated all the data loading bottlenecks, right? So in this example, we're training a BPNet style architecture to predict personalized attack-seq, and we're seeing a speed-up of about 3x. We've also put a lot of effort into making GenVar Loader as easy to use as it is performant. It only requires one new major step before you can get a PyTorch data loader ready to use. If you're interested in using personalized genomes or tracks in your research, come say hi at my poster. Thank you.
You're number 8? 9. OK, we'll do you, and then you're the last one for this session.
Recovering GRNs or gene directory networks using machine learning is like glorified curve fitting that doesn't actually fit. That was a joke to get your attention, but it does actually fit to some extent. I'm Zach. I'm working on Perturb ODE and Interpretable Neural ODE for GRN discovery. It is well established through experiments in yeast and E. coli that such regulatory systems are governed by nonlinear dynamical systems. In the simplest example, suppose gene X upregulates gene Y, then we can model the dynamics of gene Y as an ODE with a nonlinear activation of X and a linear decay of Y. By most other examples, the dynamics are much more complicated. That's why we're presenting our model, Perturb ODE. Our objective is to recover GRN from interventional RNA-seq data and predict trajectories under unseen interventions. So in our data sets, we have hundreds of perturbations and a distribution corresponding to each intervention. Let's first look at our ODE formulation. So suppose we are given a gene expression vector. We first map it down linearly to a lower dimensional latent cofactor space with some additional shift and scaling. And then we apply a layer of nonlinear activation of a sigmoid function, and then we map it back up linearly using the B1 matrix. And our GRN is basically encoded by multiplying the B1 matrix and the B0 matrix with some additional scaling. And we also have a linear decay term. And we also have an intervention term that we set manually. If you look at the underlying parts of the ODE, you can basically think of it as a base vector field that is tunable like a neural network. And then we manually set the intervention so that this will lead to a different distribution of cells given different interventions. For training, for each intervention, we basically push forward some initial distributions to our prediction, and then we compare the Wasserstein 2 distance to our target, and then we backprop. Some additional comments: our model can manage both perfect and imperfect interventions, including both overexpression and knockout scenarios. And we also train with some diffused target densities to promote local stability by contraction mapping theorem. And we can handle dimensions as high as 1,000 with 32 gigabytes of memory. And this is our result. We outperforms DCDI, DCDFG, no-tears, no-tears LR in both real data and simulated data. And we are also capable of making accurate predictions for unseen interventions. So in conclusion, Perturb ODE is a biologically realistic and scalable alternative to traditional SCMs. And if you're interested, please check out poster number 9. Thank you.
Okay, now I'm going to hand it over to Anshul, who's going to introduce the second keynote.
Hello. Yeah, great spotlight talks. It's my privilege to introduce Sergey. He has been, in many ways, a pioneer in developing next generation models for protein sequence, structure, and function. And today he's going to help us venture into interpreting these models, which, as we've seen, is quite important to understand what they're actually learning. And so look forward to his talk. Thanks, Sergey.
Thank you. Thanks, you guys, for inviting me to be here. It's really great to be back. I think first time I came to this conference actually was still part of NeurIPS, and so it's been interesting to see the evolution. Okay, let's see. Slideshow. Oh, there we go. Yep, so today I'll tell you guys about protein language models, but since a lot of us care about genomes, I threw a little bit of genomics in there as well. So I'll tell you guys a little bit about some of the investigation that we've been doing trying to understand what different protein language models are doing. Some of these slides may be familiar to you guys because some of my past collaborators have actually presented at this conference before. So, but maybe this will be a refresher for those of you who've seen it before. Maybe for new people, also as well. All right, so the big picture and the roadmap that I'll follow today is quickly introduce you guys what are sequence models, at least the ones that we use, how these relate to protein language models, and how different are they. And then towards the end, describing what's also been learned about genomic language models, and potentially how these two things are connected, and how tokenization takes a role in what these models end up learning at the end of the day. So big picture, let's say if we have a sequence, and the sequence could be DNA sequence, RNA sequence, or protein sequence, and we have one sequence, there's not too much we can say about it. But where things become really, really powerful is when you actually have a collection of sequences, right? So you have one sequence and you collect a whole bunch of sequences that are related, and now this becomes really, really powerful because now you can say, hey, this is like a very conserved position, these may be covariant positions, maybe this position is completely random. So it's through power of comparison that things become really, really powerful. But how do you actually make these comparisons? Like what kind of model could do these comparisons for us? And so some of these comparisons and some of the things that you could learn from these models is like, for example, like as I mentioned, conservation, phylogeny, coevolution. And one example that I'll be focusing on today is this idea that one really, really powerful thing that you can extract from these kind of data are these coevolution signals. And the reason why it's quite important to get the signal is because let's say if you want to predict the structure, using this coevolution signal, you could potentially tell you like these two positions are close together in the structure. And so you guys may have all heard about AlphaFold. At the end of the day, it looks like AlphaFold is essentially still using this exact same information to make the predictions. It's extracting these covariance patterns from a collection of related sequences, and that's how it works. And what I'll try to argue today is that even these new language models, protein language models have been trained, are essentially still doing this same thing, but less implicitly, less explicitly, more implicitly. Okay, so how do you actually model these constraints? So I'll just walk you through a toy example. So let's imagine we have a multiple sequence alignment, a bunch of sequences. Two positions are covariant with each other, and one position is completely conserved. I'm going to kind of take that one sequence, let's say from human, one hot encode that sequence. I just need to describe it somehow in order to pass it to a computer program. So like for blue is 1, 0, 0, and so on. Then I could come up with some kind of a neural network or a layer that describes the relationships. You can think of it as like training some kind of graph model, or you could just think of it as a dense layer that connects everything to everything. So for example, like position 1 is connected to position 2 is connected to position 1, and so you could essentially use a very simple matrix to describe these relationships. And then finally, I know that some positions are completely conserved, like sort of in my toy example, like this guy's conserved, it's not dependent on anybody, so I can add some kind of bias term to describe that at the very end. And now finally, let's say if I wanted to train a model, essentially like an autoencoder that goes input to output, I have to make sure their outputs sum up to 1. And so here I could add some kind of softmax activation to make sure every position sums up to probability of 1. And then finally, I could try to train this model to essentially recover the inputs on the other side. And if you imagine training such a model, you'll probably learn something very, very simple. Like in this case, it could just learn an identity mapping. And so to get around this problem, what people in practice do is employ pseudo-likelihood. And so in this case, pseudo-likelihood would essentially be saying, let's just mask the diagonal, make sure that there's no self-connections. So the idea is like, let's say if I want to know how 1 depends on 2 and how 2 depends on 1, if I remove connections with yourself, then instead of the only way to explain that would be to pass through another residue through a different position. And so now we can actually learn these weights would represent those connections. So this is just history of how we typically used to do this. And one particular example of this model is a method called Gremlin, and this is the method I'll be comparing to as far as comparing language models to these traditional methods. And the reason why it's called Gremlin is because you're regularizing it, and it turns out when you do name your algorithm, it doesn't always have to be the first letter of each word. It turns out you can use any of the letters. Just heads up if you want to be more creative in your names. Yeah, so, but the main thing here is that it's essentially the same model, just what I described to you, but sometimes it's easier to think of it as a neural network as opposed to some kind of graph model or Markov random field or a POTS model. It's essentially just a single layer, and you're just doing softmax and then minimizing the cross entropy between the inputs and the outputs. But once you've got these parameters of this model, like what do you do with these parameters? How do you even extract these contacts that I just told you about? So what you could do now is you could take these weights and you get back some kind of tensor that's length by length, in this case alphabet by alphabet. And then you could take each of these little boxes here that's alphabet by alphabet and reduce it to a single value. So usually you take like L2 norm or Frobenius norm, and you get back a contact map. And so now the question is like, okay, that looks pretty cool. And just to summarize, so now we train it on this data, and now we have essentially a contact map that represents the connectivity or how the different positions are dependent on each other. So this is just for a quick toy example, and just to show you that I'm not making things up. Here's an actual protein. So here you can see in gray are actual contacts coming from the crystal structure. And this is one particular example of a protein. This is a ribosomal protein. And let's say if I wanted to show how these these cyan contacts interact with these orange contacts, I could just look it up here. So cyan, orange, that's they are. So that's how you interpret contact map. I'll be showing quite a few of these, so I thought I'd spend a little time showing you how to read these guys. But now what I could do is I can overlay these contacts that I get back from this essentially a covariance matrix of this multiple sequence alignment, and you could see they actually overlay really nicely. So this was actually a really, really exciting result back in around 2009, 2011, where people started realizing, hey, these are actually really, really powerful information just by simply mapping this very, very simple model, or training this very, very simple model. And this sort of allows you to imagine a scenario where, let's say if you didn't know what the structure was, let's say if it was just some long extended chain and you put like rubber bands everywhere, you could really start to snap things into place. Like all these white lines are positions that are covariant with each other, and now you could potentially build a structure that maps those contacts. All right, so how about protein language models? How are those connected, and is there any difference? Are they the same? And so what I'll try to argue today is essentially it's almost doing the same thing. And what I mean by that is you still have an input sequence that you're one-hot encoding, you're passing it through some kind of layer, and you're returning probabilities at every position. The probability of that amino acid condition on everybody else. And in this case, and let me just break this down a little bit further, just to make it more interesting. But before I do that, just to quickly remind you guys the big difference between the two ways of training. So imagine with methods like Gremlin, what we usually do is we train one model per protein family. So for example, you have a ATP synthase, you have a separate model. Beta-lactamase, you have a separate model. With methods like protein language model, you usually train one model for all sequences. And usually if you do have to do that, then you might have to also think about positional encoding, because every protein might have some kind of different meaning at every position. You maybe have to do some kind of relative positional encoding, because maybe you want to allow for things to move around, like have insertions, deletions. And so, like in this case, it's a very, very simple model, but in this case, you do really have to pay attention to how you actually encode the positional information into the model, so it actually learns something useful. Okay, so let's break it down a bit. So let's say we have a sequence, which I'm just going to show as a gray box for now. I could pass it through a very simple linear layer and also add some kind of positional embedding. And so this is my new inputs. Then I could pass it through 3 more layers to get query keys and values. And if I take the outer product of these keys and values, I can get essentially how each key attends to each query. This is also referred to as an attention matrix, for those of you familiar. And then once you have this attention matrix, you now have to populate those attention with some kind of values. And so you multiply that by the values, and you get a new output. Now, once you have this new output, you could pass it through another layer, which in this case I'm just going to call a decoder layer, to get back the sequence. And then you could take the softmax of that sequence and minimize the difference between the input and output. So essentially, it's the exact same objective as the Gremlin model that I just told you about. But in reality, what happens is, here I'm just showing it for like a single layer, a single head. But in reality, you could go a bit further. Like you could say, hey, there could be multiple heads. But one thing that I should probably emphasize here is, in the case of Gremlin, it was really, really easy to fix the problem of information just coming right through the model, right? So like one question you might wonder is like, why can't the model just learn an identity matrix once again, right? And so to get around this problem, often people, what they do is to train these models, they would train it with a masked language modeling objective. And so what I mean by that is take your sequence, mask some positions, and then in this case, you only want to predict those positions that you masked. And so what I'm trying to argue in this particular slide here is that the pseudo-likelihood objective is actually very, very similar to the masked language modeling objective. In that case, you're masking some positions and recreating them. And in fact, what you could do technically is you can go to every position one by one and mask them one by one, but that would be really, really slow. And so in practice, people mask like 15% of the sequence. But in some ways, the two functions are approximate each other. So just based on this information alone, one could probably argue like, okay, maybe they're learning something similar, right? Okay. All right. Oh, I don't know if anybody has any questions, but if anybody does, feel free to ask during. I don't know if we have to wait till the end. Yeah. Any questions before I keep going? Or concerns? Otherwise, I think I might be going a little too fast. So I feel like somebody has to slow me down a bit. OK, so all right. So next, oh yeah, go ahead.
So 1 second, let me bring it up.
I guess you mentioned briefly that the one-hot encodings were dependent. They were different length depending on the length of the protein sequence. But somewhere we got around that. I missed it. At some point, they all become the same length. Is that correct or no?
Great question. So here, when I'm showing these little arrows going from sequence to input, or even from input to keys, you can almost think of it as just doing like a 1D convolution. You essentially apply the same weights to every position the same way. And so you might imagine, like, oh yeah, that seems kind of silly. If you apply the same weights to every position, it would probably don't matter how you rotate your sequence, how you shuffle your sequence, right? And so to get around that problem, you add some kind of positional embedding. And this positional embedding could be like some kind of sine wave, or it could be a combination of sine and cosine waves. And essentially what you want to do is essentially give every position some kind of unique representation. It's like position 1 has this representation, position 2 has this representation. In practice, for protein language models, what's found to be the best is to do something called rope embedding. What rope embedding essentially at the end of the day does is learn some kind of just relative representation. So I guess you could say, like position 1 and position 2 are going to be really different from each other, but position 2 and 3 are going to also be relatively similar to each other. And so you essentially just encode some kind of relative embedding instead. And so then what this allows you to do is now if you give it a protein of different lengths that is the same, you essentially still have similar relative encoding of that sequence. And so then you can get away from the problem having to align those sequences. Now, if they're all aligned, your positional embedding could literally just be an identity matrix that literally says this is position 1, this is position 2, this is position 3. But if they're not aligned, then you have to provide some kind of relative information. Some of the earlier models, like the tape model, actually had a problem where they didn't do that. They essentially had some absolute positional embedding. And in that case, what happened is like you literally shift the sequence by 1 position, and it just crashes, no longer works. So it literally learned the perfect embedding for every sequence, but then it was not able to generalize as soon as you added like 1 position anywhere in the sequence. So this becomes really, really important on how that works. Yep. All right, so, all right, okay, in that case, I'll keep going for now. All right, so I just described to you guys how you go about training such a model. And I guess the argument I was trying to make is that it's the same inputs and the same outputs. Even the loss function is essentially approximating the same thing. And so maybe a spoiler, it may be potentially doing something very, very similar. But how do we actually demonstrate that it's doing something similar? And so one approach we took is that, well, what if we just train a protein language model from scratch, and on a single protein family that's already, all the sequences are aligned to each other? What will happen if we just train a single model on a single protein family? Will it actually learn something interesting? Will it learn the same thing or not? Because here we have full control over the inputs we give it, and that's the only thing that it sees is the same exact inputs. And so what we found is if you train it on a single protein family, you actually only need a single layer to essentially return the same results. The other thing that's kind of interesting is because if the sequences are aligned, like I mentioned earlier to Jeff, is that you could essentially put in the positional information could literally just be a one-hot matrix, which specify, or I guess you say identity matrix, which specifies position 1 is 1, position 2 is 2, position 3 is 3, and so on. And so it actually becomes really, really, really simple model. But now the really remarkable thing, which probably is not too surprising in hindsight, is if you extract the querying keys, like the actual parameters of the model that represent the queries and keys, and take the outer product of those guys, you actually get back on contact map. And so within the parameters themselves of this very, very simple protein language model are already embedded all the same parameters that you get back from, let's say, like a gremlin model. And just to make things more confusing, it turns out you can, I guess you could say, refactor a simple attention model into different components that essentially connects all the parts. And so the reason why I'm showing this is, like, for example, in a gremlin model, we have a weights matrix. And that weights matrix could essentially be decomposed into 2 matrices where you say, hey, I'm going to model all the interactions and have some kind of heads parameter. And then each heads might be representing some 20 by 20 matrix describing how those parameters interact with each other. You could further decompose that into, like this parameter could be decomposed into queries and keys. This could be continue going on. And eventually you could see the values and the final feed-forward network are actually going to tell you all the information about how they're interacting with each other. And then the queries and keys are essentially the decomposition of this length-by-length matrix. And so for a single protein family, you essentially could go from beginning to end, like fairly completely decomposing it into the query keys and values and the final feed-forward network. Or you could just keep bringing it all together back into a single linear model that just has the length by alphabet matrix. Now, if we actually go through the process, because now that we know how the relationship between this and the queries and keys and values, we can actually now take the queries and keys and values and compare it back to this matrix and see, are they actually learning the same thing, right? And so I guess the argument here is technically if you make this H, which is like the heads, large enough, it should actually be able to reconstruct this matrix exactly. And that's essentially what we see. So here I'm comparing this W matrix that we get from a gremlin, but also this W matrix that we could pull out of a protein language model. And what we find is as you increase the number of heads, those parameters become more and more equal, which I guess mathematically kind of makes sense, but it's kind of cool to see that it's actually, essentially, if you make heads the largest possible amount, which I guess in this case would be like 400 values, then it essentially should start equaling each other. Okay, so but this is just for a single protein, single protein model. For a single protein family. What if you actually train this model on all the sequences, right? And so this is actually in practice what people do. People don't train a separate protein language model for every protein family. They train one model for all proteins. And so when you do that, often a single layer is not enough. Like you have to actually have multiple layers in order to get a meaningful pseudo-likelihoods from the model. And so in practice, you have multiple layers. And in fact, often you have to have up to like 33 of these layers. And so just to give you an example, and one way we could potentially interpret these models is if you take a sequence, pass it through many of these layers, you can start looking at these individual layers one by one. And it turns out if you start looking at these layers, they actually capture some really interesting features. Like some of the earlier layers, very similar to some of the early work on convolutional neural networks, are kind of capturing some low-level features. But as you keep going to the end, it turns out often the last layers are beginning to capture these contact maps. And so some of the hypotheses are maybe like some of the first layers maybe picking up some local structure, and then as you keep going further, you end up building up the entire contact map. And this is an analysis of across all layers, across all heads, and what I'm showing here is the data points that are showing how accurate these heads correlate to contacts. So the higher you are, the higher correlation you get. And so you should see, the interesting thing you see is often towards the end is when you start to recover all the contacts in the model. Besides us, there's been other people that showed similar things. One paper being Biotology Meets Biology. So if you guys are curious, you can check out some of that more work. But one thing that I wanted to sort of highlight is if you start to actually compare the performance of these models, like the performance of how well these contact maps correlate to ground truth, they depend highly on the number of training examples. And what I mean by that is if you take a protein family and you ask the question, or you take a protein sequence and you ask the question, how many examples are there in the training set that are very similar to that protein? And there are some proteins that don't have that many examples, and there are other proteins that have a lot of examples. And so it turns out with gremlin models, we saw that before, where the correlation is really rapid or striking, where like the more data you have, the more accurate these models are fit. But the interesting thing was even with these language models, you see the same kind of correlations, where the more examples there are in the training set, the more accurate these models become. But one thing I should emphasize when I say training set is like we're not training the models to return any contacts, it's purely unsupervised learning, but just the parameters themselves have better correlations to contacts. And ESM2 recently did the same experiment again, but one thing they had access to, which we didn't at the time, was this ability to say, well, we could train models larger and larger and larger, right? So in this case, I'm comparing it to one of the initial tape models, ProteBERT, and so on. But what the ESM team group did, and I believe Alex Rivas may have talked about this last year at this conference, is that the idea is that as you increase the number of parameters, you actually start getting better and better contact accuracy. But one thing I wanted to highlight here is this orange line here. So this orange line represents examples of proteins that don't have that many examples in the training set. And as you see, as you start to increase the number of parameters, it does indeed start to get better and better and better, but it has not yet reached this level here. And I think at some point I was trying to extrapolate how many parameters you would actually need to get there. And I think the estimate right now is 10 to the power 25. So you need to burn a lot of trees to get there, I guess. But eventually we'll get there. But that's assuming that it's linear. Who knows? It's probably not going to be actually like that. Yeah. Oh, yeah. Question? Yes. Yeah. Uh-huh.
So is it the amount of training data, or is it actually the unbalanced sampling? So like what would happen, is it really that you just need more training data, or is it that you're seeing more of one type of family versus, so what if you upsampled or did some kind of balanced sampling of families? Would that help, or does it not help at all?
That's a great question. So in some ways, there has been some data balancing in terms of, like during training, often what people do is they first cluster all the sequence data, and then they pick one member per cluster when during training. So there is some degree of data balancing going on. But one other hypothesis was like, maybe we just don't have enough capacity, right? So one of the things that Alex Rivas' team says, this is why they're calling it evolutionary scaling. They're like, okay, we just need to scale, and if we scale, we should potentially be able to, even for the very, very low protein families that don't have that many sequences, eventually they'll be able to also capture those parameters as well. But I guess what I would argue is even if you have infinitely large model, eventually things start to plateau, and that's essentially what we're seeing with this orange line here. So those very small protein families, they are now getting better and better in terms of their, the model seems to understand those small protein families really well, but even at 15 billion parameter model, it's not getting much better. This is just like wishful thinking, say, well, if you make the model infinitely large, maybe eventually it'll get there, but in reality, I think it will essentially just plateau. Now, there is some hope saying, well, with all the metagenomic sequencing out there, maybe we'll start to sample some of those families better and better, but I don't think that will solve the problems for like eukaryotic protein families, for example. Like eukaryotes on average are very, very similar on the protein level, and so I don't think it's going to be any kind of, one thing sort of related to this plot, sometimes people ask me, like, how do you know that the model has now had some kind of a breakthrough? I don't know if AGI would be the right word for this, but I would say if indeed you see like this line is flat, then I would say, okay, maybe the model has actually learned some kind of protein folding, maybe the model has learned physics, because the idea is like you give it a sequence it's never seen before, it should be able to understand the structure of that sequence really well. But in reality, what we see is this really, really strong correlation. And we do think it's one of those things where it's not because of data balancing, it's more like there's just, I mean, it's probably a little bit of both, because in some ways, it's just there's not that many examples for that, and there's no reason for it to even store that information in that family. But even if it had the ability to store it, there's like nothing to store, because there's essentially all those sequences are very similar, just a few of those guys. Yeah. All right. Okay. Any other questions? Yeah. Oh, I think there's one all the way in the back.
Hey, so do you already see that double descent slope in these models at that plateau, or is it like all dependent on protein families?
So this is showing an average across all protein families. So you can think of it like this line represents protein families that don't have that many sequences, and this represents protein families that have a lot of sequences. And if we just focus on protein families that have very low number of sequences, you see that it just sort of plateaus. And I mean, I guess there is some hope. Like I guess this is where I was trying to draw this line here. But I don't think it's gonna. I don't know if that was the question, but I don't think it's gonna get any better for those families, unless it does somehow snap out of just the effect of learning conservation coevolution about these guys. Yep.
Maybe protein structure might not be enough. So maybe there's some interacting proteins that help to stabilize the structure. What are some thoughts about that? And if you fold in some information about interacting, like known protein interactions, could you improve performance?
It's a great question. So in this case, the model never sees any protein structure at all. It only sees sequences. One thing I'll talk about a little later today is there are other models that you can give it pairs of proteins or more than just a single protein. And there, I guess, I mean, we would expect maybe to see improvement in prediction between the proteins, but maybe not so much within the proteins. But if it does improve something within, that would be actually kind of cool. But that's not something we looked at yet.
So one more question along the previous lines. So if you take a look at these proteins that have very few members, I mean, the performance is not zero. It is better than random. So what's your interpretation? Is it actually learning some physics, or are we underestimating the number of real neighbors?
It's a great question. So I don't have a line here, but it turns out if you actually just make a very, very simple model, say, given the amino acid identity, what's the probability that it'll be in contact? It's actually around 20% or so. So it's actually not too bad. And so I would say, I mean, Gremlin, this model knows nothing about proteins, right? There's no priors about proteins. So at bare minimum, maybe some of these jumps, for example, above 1 might just be coming from the fact that you say, hey, for example, one thing that you're adding is positional information. Gremlin knows nothing about position. And so you might say, hey, if you're 5 residues apart, you're more likely to be in contact than if you're 30 residues apart. So a lot of this is actually just coming from those very simple statistics that you could have probably added yourself. Yep. OK. All right, so just make sure I still have time. I think I have like 20 minutes, right? All right, so given all of the results, what this sort of tells us or leads us to suspect is at the end of the day, protein language models are essentially just learning a library of Gremlin models. So this is a visual here saying, given a sequence, maybe somewhere inside the parameters of this model, it's looking up the Gremlin model, and then based on that Gremlin model, it's returning to you the probabilities and statistics for that protein family. And so another way to maybe reframe protein language models is some kind of fancy lookup and compression algorithm. But that being said, there's a chance it might do better because of shared parameters. Because right now, when we do fit these individual models, we're only providing information for that one single protein family. But maybe there's multiple protein families that have similar kind of motifs, and those motifs could potentially help each other in some ways. So we do expect that they will do better than just training separate models. But at the end of the day, it's still essentially compressing, or at least this is what we believe they're doing. And so I guess let's say the 3 hypotheses about how the protein language models are storing structure is protein folding, which I already told you is probably not the case. But the 2 cases that I'll talk about for the rest of the talk are these: is it looking up entire proteins, or is it maybe looking up little motifs? Because instead of storing individual little models for each protein family, you can imagine it's storing like common motifs that are shared across proteins instead of storing a separate model for each protein family. And so I guess I'm just going to rule out this one for now. Say, okay, it's not doing protein folding, because if it was, then we would have expected this line to be flat. Like, it shouldn't be dependent on the number of sequences, right? So that are already telling us that's probably not the case. Maybe with a 100 billion parameter model, maybe we'll get there, but who knows? We'll have to see once we have enough GPU power. Okay, so we're ruling out this for now. So now that leaves us with 2 different hypotheses, that it's looking up entire proteins or it's just looking up motifs. But before we can even go about answering that question, how do we actually go about extracting information from these models? So I told you guys about extracting contacts themselves through the attention matrices, but sometimes this is not possible, like especially some of these newer models that use like these hyena layers. You don't actually have explicit attention layers that you could look at. But in other cases, like if we actually want to argue that it's learning coevolution or storing coevolution, we want to actually pull out the exact dependencies, not just some approximation of dependencies. So how do you pull out dependencies from these models? It turns out one way, a really relatively straightforward way, is to say, well, what if we just map every input to every output? Like just essentially create a matrix that shows how every perturbation of the input affects the perturbation of the outputs. And in one particular example of this is to compute like a Jacobian matrix. So like, for example, if you take a model like Gremlin, which is a linear model, and you compute a Jacobian, which essentially is a matrix of partial derivatives. It's essentially telling you how every input, how perturbation of the input affects the outputs. You get actually back a really nice contact map. If you do the same exact thing to protein language models, it becomes really, really almost bland or somewhat flat. And so we were kind of wondering, like, why is that? Like, why does it look so flat? Does that mean it's not learning coevolution? And so this is where you might wonder why. And so And so what I try to argue here, but before I go into more arguments, let me just explain what these concepts I'm referring to mean. So in a very, very simple model, what exactly is this input-output value that I'm plotting? What is this Jacobian is? And so essentially, you could think of it as essentially some perturbation in the input and checking the perturbation in the output. And a Jacobian is essentially when you get to this limit of 0. So once you get to the limit of 0, it's whatever the slope of this line is, tells you what that gradient is and how the inputs to the output map. But what if you have a scenario where this model that you're evaluating doesn't actually have that nice linear effect or a nice curve, but instead it has a lot of flat areas, right? So like what if I just made some input here, like a small little change here, and the output is exactly the same because you're in, so essentially this linear space, right? And so when we're computing a Jacobian over a protein language model, maybe the reason why it looks all flat because small little changes do not affect anything on the output. That's why it kind of looks flat. And so one thing we try to do is say, well, what if we actually make the step size larger? Like, what if we actually make a larger perturbation? So let's say if we want to see, like this right here doesn't change anything, but if we make this input a little bit larger, then maybe we'll actually start seeing differences in the outputs. And so we actually tried to do a scan of different step sizes, and probably not too surprisingly, we found the optimal step size was 1. And in hindsight, it kind of makes sense because 1 is roughly enough to maybe almost switch the category. In fact, if you maybe take a little bit larger step, you might completely flip the category within the model. And in practice, we find that actually just directly just changing the step altogether, like instead of just adding a 1, just replacing that with the 1 hot encoding of the other amino acid, it actually gives us better performance. And so this is, every dot here is a different protein family, and I'm evaluating the contact accuracy. And so computing Jacobian, which is essentially just a step size as a very, very small step size versus a step size of 1, got a huge boost. Now you actually see a very, very clear contact map. And finally, a step size of 1 versus actually just switching the category gives you a slight boost, but not too much of a boost. So this is now going to be our default protocol that we'll be following, and this is what we're going to essentially be evaluating for the rest of the talk is this 1 hot switch. So just to summarize this part of the talk, so the idea here is that you could take an input sequence, you could pass it to the model, get some kind of logits. These are the probabilities at every position for that amino acid. You can make a mutation at every position to all the other possible amino acids, and you get differences in the output logits. Now you can take the differences in the outputs and you get back this, what we for now are going to refer to as a categorical Jacobian. This is just a term that we made up, but if somebody has actually done this and gave it a different name, we're happy to change it as well. So anyway, so we're just calling it that for now. But the idea is like you could do all possible perturbations and get back a length by alphabet matrix. And just what I showed you guys with the gremlin, I could also do the norm for every 20 by 20 matrix to get back a single value. And what this allows us to do, which is kind of cool, is to start to actually compare these values directly between the models. So we could take a multiple sequence alignment, in this case compute an inverse covariance, which is an approximation of gremlin, then take the Jacobian over a protein language model and directly compare all the parameters to all the parameters. And one thing we see, the contact maps are very similar, but even the 20 by 20 matrices are actually very similar to each other. It's capturing similar probabilities for how the amino acids change to each other. And this is actually showing all the values, length by alphabet, and you can see that there's a reasonable correlation between the two. So this essentially tells us that it's not only that the contacts look similar, but actually the parameters themselves that it's learning are actually almost identical between getting a multiple sequence. Now, the reason why they're not perfectly the same partially might be because maybe it's the MSA, like the implicit MSA that the language model is using maybe includes more or less sequences. Like we don't really know what the actual MSA it's implicitly learning. And also potentially you could fill in information. Like there's a bunch of 0 values here that may be potentially filled in from other protein families. And so maybe you could do a better job at learning these parameters. Yep. Yep, go ahead.
Can I ask a question? Okay.
Sure.
I just want to know if I correctly understood. So the Jacobian essentially is telling which output features are more sensitive essentially to change because it's where like the contact is happening. So this is like the intuition. Like I could use this to understand, for example, importance. Like these are the important. If it would be like an autoencoder or something, I could get the importance because that is where is the most important point in the output because a slight perturbation is affecting it the most.
That's a good question. So I guess you could call it importance, but I'm not sure if that's completely the way, at least the way, maybe the simpler way to think about it is like, let's say if position 1 is dependent on position 3.
Yes.
So if I mutate position 1, then I might have differences in position 3,
yeah,
and vice versa. And so you sort of get at least what, I guess you could say, the dependencies.
Yeah.
But whether or not that means importance is not always true. And what I mean, the reason why I'm saying that is because sometimes the most important things do not co-evolve. Like for example, let's say if you have a metal binding site, it might be completely conserved, and in fact, you won't see that in the matrix. And so I think the interesting thing about coevolution is that things that are actually co-evolving are actually not the most important things. They're sort of things that are medium important, because then they can tolerate substitutions.
And a related question was like, I think I might be wrong, but on computer vision, I read something about like the norm of the Jacobian is related to how much a point is out of distribution. So I don't know if the population you were mentioning before, like the rare proteins, have like a higher Jacobian norm, because whatever you change is like you get something in the output that changes because the model is not getting what is happening, or something related. So their on vision was like if you are on the manifold of the images, then like it's challenging to get which point to change. But if you are out of it, like everything is like affected. So I'm going back to my, because I come from different intuitions, I come back to this importance concept, but I don't know if this might be happening on.
I'm not sure. I'll think about it some more. Maybe we'll get back to you.
Okay.
Yeah. But that's it. It's an interesting connection. I haven't thought about it that way. Thank you. All right. So, just to make sure we're on time, because I think people want to hear about genomics as well. I'll just keep going far. Okay. So we're almost done with this section, but essentially, to summarize this is like the contexts are similar, which is pretty cool, but also the parameters that we could extract from the models are similar to the ones that you could get from just training a simple model and that same protein family. And so this gives us a lot of confidence that, in fact, maybe the models are just indeed storing these coevolution matrices. And in fact, when you give it a sequence, it's essentially just extracting those same parameters. But there's still the possibility that maybe it's not doing a full fold lookup, right? Maybe it's sort of, you can imagine, like, if you were to compress all this data, like if you were to do it by hand potentially, you wouldn't go around storing a separate model for every protein family. You say, hey, these families both have helices. Maybe there's some shared set of parameters between these protein families. In fact, maybe you'd share like motifs, like how this interacts, and you store like mini gremlin models, right? And so Zidian in my group, who was visiting at the time, thought we would do a quick experiment to test for this. And so one idea is say, well, let's just say this is a little motif here. This is a local motif. We have a beta strand interacting with the helix. And what we could do is we could just mask everything except for that motif and ask the question, does the model understand that motif? And what understand means, does it have contacts that represent this interaction? And also for motifs that are separated from each other. For example, this helix separated by this helix, we can mask everything in between and on both sides and ask the question, how much contact is actually needed to recover those motifs? And so if it turns out you need the whole sequence, then we say, okay, the model does essentially a whole sequence lookup. But if it only requires that single small motif, then that tells us, okay, the model probably only needs that one segment in order to understand that interaction. And so here's a quick animation. And so here, what I'm showing here is literally taking this whole protein and masking everything except for that motif. And I think one kind of cool thing about this animation is essentially you could almost get rid of the whole protein, and there's only a certain part where essentially it starts to disappear. And so you could almost get rid of everything except for that motif, and you can see that it's still there. Now for the pair interaction, this one's actually kind of cool. It's like essentially you can almost get rid of everything except for that helix, and you could still see everything. So essentially all I'm doing here is removing everything from the sequence except for that motif, and then asking the question, does it understand the interactions between these two parts of the sequence? And it still does understand them. And so to us, this suggests that the model is indeed maybe learning not whole proteins, but learning motifs. And this could be a good thing for protein design. For example, let's say if you want to design a new protein, and you're like, hey, I want to recombine a whole bunch of motifs in different creative ways. This suggests maybe one way to be able to do that. That being said, it's also possible because it's just recombining motifs. The question is, like, is it really novel, or is it just doing the same thing? So that's something up to debate. Okay, so this sort of leaves us with this hypothesis that it's essentially storing these motifs here. And so just to summarize this section, Jacobians are very useful because it allows you to analyze models, especially if you don't want to sort of think about what's going on in these black boxes. And we do have some links here for analyzing ESM models, for example, for analyzing protein and PNN models to extract contacts. But on top of that, you could also do this on every single layer. So this little animation here I'm showing here is computing Jacobian from input to layer 1 to layer 2 to layer 3. And you could sort of, the interesting thing is you could sort of see as you go through the layers, you start to build up more and more of the contacts, and eventually you get all the contacts on at the very end. So if you guys want to analyze this for your own model, check out the code here. And then finally for I think I only have 10 minutes left, right? Am I doing the math right? Here, okay. So finally for the last 10 minutes, I'll tell you guys about genomic language models, but not going to introduce any new concepts here. We're going to apply the exact same thing that I just told you about, but now to genomic language models. And one genomic model that you guys may have heard of already that was recently put out there is this new model called EVO. To me, this was actually an interesting model because they didn't use explicit attention layers. It was not one of those things that you could just look at the attention maps and ask the question, is learning contacts? You essentially were kind of forced to do this kind of Jacobian perturbation that I just told you about. So the idea here is that you have an actual DNA sequence, and the output is also a DNA sequence, and you can now perturb the input of the DNA sequence and ask the question, how do the probabilities of that sequence changes? So the exact same analysis, and there's a lot of crazy stuff happening in the middle, and I guess what I would say is like, let's just ignore what's happening in the middle, just focus how the inputs map to the outputs. And one exciting thing is if you give it an RNA sequence, you could actually recover all the stem pairings. And so you can get local contacts. At some point, I gave this talk at a different conference, and I said it didn't learn long-range contacts. Turns out there was a bug in the EVO code, and the authors emailed me and said they fixed the code. It should now recover long-range contacts, and it does indeed. So so it turns out that they had some bug in how they, I guess, tile the hyena layers or something. But yeah, so now it's actually picking up these long-range contacts, which is quite exciting. But one thing, and also I just want to do a quick plug-in. There's another group that actually did something very, very similar to what we did, but more across all different DNA-based language models. And so if you guys want to read more about other models, you should check out this work as well. But for our purposes, we don't really care too much about the RNA. We care more about the protein side. So the question is, like, how about proteins? Do these models actually work on proteins? And so here is one particular protein family that has a huge amount of sequences. And so we don't doubt that it hasn't seen these proteins many, many times. And we see with protein language models, it actually captures the context really, really well. But genomic language model, where you essentially perturb the nucleotides and see how the changes in the outputs change, we see that it's actually very, very, once again, looks somewhat fuzzy. The reason why I'm showing only half of the matrix is because it is an autoregressive model. So essentially, when you perturb something in the input, everything changes downstream, but what you change downstream does not change what happens upstream. So so that creates essentially a half matrix. But for visualization purposes, you could always just drop it on the other side. One thing we try to do is say, well, maybe it doesn't understand proteins, but maybe it understands interaction of proteins, or maybe how the proteins are arranged in the genome. So if you give it, like, for example, an operon, in this case, 3 different proteins, it looks like it's learning some kind of splitting between them. There's these streaks that I think might be maybe related, like the start and stop codons. So maybe if you mess with the start codon, you probably start losing some information. But it doesn't seem to be understanding contacts in the same way that we have with proteins. And so this sort of leads to maybe 2 hypotheses about why it's not learning protein-protein side chain interactions. One, of course, scenario is like, well, now the task is much larger, right? You're trying to sort of store all the evolution of the genome. Maybe your capacity of your model is not large enough. You maybe just need to make these models larger, and that's what we saw with ESM models. Like, essentially, as you scale them, eventually they stored better and better contacts. Another potential scenario is maybe it's just the alphabet is wrong. And what I mean by that is often, especially for protein reasons, if you mutate one of the nucleotides, like, for example, like the 3rd codon or the 3rd nucleotide of a codon, you're really easy to recover what that amino acid is without even thinking about what the rest of the sequence is. So maybe the way the model is trained by just masking individual nucleotides, the model never had an incentive to learn these long-range protein-protein interactions. Luckily for us, in collaboration with Yuna and Andre from the Tata Bio company, they actually said, well, you know what if we actually train a new language model But in this case, we purposely use just amino acid encoding for the amino acid positions, but then use this BPE encoding for the rest of the DNA parts. So the idea here is that you could take a chunk of the genome, in this case a metagenomic context. For parts that are protein coding, you actually encode with an actual amino acid. So you have 20 tokens. And parts that are DNA or non-coding, you can just encode with these little segments, and these segments could be of variable lengths. And so then during training, you can actually mask entire, effectively, codons instead of masking individual nucleotides. Maybe another way of we could have done this is to say, well, let's actually encode like the 64 codon tokens. That could have been maybe a better way to do it. But in this case, encoding the individual amino acids as essentially, I guess every codon to an amino acid token. And so when you do that, and then you analyze, for example, protein-protein interactions. So in this case, this is one protein ABC transporter interacting with another protein. If you run these through Gremlin, we get contacts. That's not too surprising. These things are co-evolving with each other. And we see the same thing with genome language models. Like we can do mutations in protein A, and we see that the logits in protein B actually change as well. So you actually see the interactions appearing in those guys. Just to contrast that with the two other models. So this is the EVO model. We see nothing there. It's like just blank, not learning anything about the interaction. And the same thing with ESM2. Like you give it the two proteins, and it doesn't give you back any contacts between the two proteins. I mean, it's never seen those two proteins together, and so that's not too surprising. But I think it does definitely, like I guess the summary here is that the tokenization does definitely probably play a big role. Maybe not necessarily tokenization, but how you mask. So maybe like for protein regions, maybe it makes sense to mask like 3 nucleotides at a time that are close together. Maybe for non-coding regions, you want to mask individual nucleotides. Otherwise, you might not get enough resolution for DNA strands and so on. So I think it's something to keep in mind when we're training these models. OK, so with that, I think I'll stop. Some people to think. So I think I described a couple of projects here today, and so there's a lot of people that were involved throughout this whole thing. But if anybody's interested in collaborating, maybe interested in joining my group, I just started up at MIT in January, so we're relatively new. And so if anybody's curious, feel free to stop by, say hi. All right, well, thank you very much. I'll stop there. Yep.
Thank you for this wonderful talk. So I had a question. The way I understood the categorical Jacobian method is that it's sort of learning a general covariance matrix. And I guess something I was wondering about, do you think there is something special that these PLMs learn so much about protein structure and that it correlates so well to this categorical Jacobian that you compute? Like, could it be learning that there are some other properties that the categorical Jacobian is capturing? Like maybe a toy example is that you have 2 complementary, some sort of active sites that have common function, and they don't necessarily interact with one another, but the model learns that they're codependent. Would it be captured in this Jacobian, but maybe not necessarily be reflective of structure?
I mean, in some ways, we are perturbing pairs of interactions. And so I guess if one wanted to go crazy, you can compute like a Hessian or something, where you can maybe get some higher order stuff going on. So in this case, we're explicitly perturbing pairs of things. And we do see the highly correlation with structure. But I guess one thing I should mention is the correlation is not 100%. It ranges from like 50 to 90% across different proteins. And it might actually be interesting to look at those examples where it's like 50% accurate. Maybe it is actually picking up on things that isn't just protein.
Maybe these like genomic models, they're learning something else. That's something that's beside.
Great question. So like in this case, like it does feel like it's learning maybe the starts and ends of genes. But the reason why it's not learning protein contacts, I suspect it's mostly just because of how it was trained. Just because they mask individual nucleotides instead of trying to mask individual codons. So I think if they retrain with just masking of codons, I think it would start to pick up on interactions. Unless that's actually what you want. Maybe you purposely want it to have 2 models. One learns protein, one learns gene structure or something, and then maybe that's a good way to disentangle those signals. Yeah.
When you put up the example contact matrices learned by Gremlin and the protein language model, and there's correlation was 0.6 or something. I am really curious which one is more accurate just on this particular example.
That's a great question. So the accuracy of the contacts are actually more accurate for the protein language model. So like if I compare the ground truth contacts coming from the crystal structure, they are more accurate. So I guess what I'm saying is like we're not expecting it to be one because we expect the protein language models to get better, just because they're able to maybe use more information or maybe filter information in different ways. One of the sort of still an unsolved problem in the MSA space, so people who work on multiple sequence alignments, is like questions like which sequence should I include and not include? And then also like how do I align these sequences to each other? And so we would expect a model that was not explicitly forced to do either of those tasks may have learned a better version of that to some extent.
And I have another question that's maybe related, but why, what's your intuition for why in the protein language model, it only learns the contact map in the final layer, whereas in the other model, it's in the first and only layer of the model? Like, why does it take so many layers?
Oh, which other model? Maybe
like just the Gremlin model, right?
Oh yeah. Well, I think the key part here is that if you have if you're just training on a single protein family, you don't really need that many layers. Like we actually tried many layers, and we found that you could just go to one layer and still get similar pseudo likelihoods. And so I think the reason why that's true is because to go back a little bit to the slide that I had. Essentially, all the parameters of the coevolution could be literally just stored in the queries and keys. And just to go back to this picture here, like for example, ultimately this is what we're trying to learn. And a single layer already has those sets of parameters. Like both the queries and keys could be already represented in just a single layer. Now, of course, if you're training across many, many different protein families, then maybe you need to have multiple layers for like some kind of lookup algorithm. So I guess if we believe what I'm saying, that ultimately protein language models are some kind of lookup algorithm, then maybe what's happening is like the first few layers are saying, okay, which motifs do I see here? I need to look them up and stitch them all together. And by the time you get to the end, you finally put them all together. One little side note here, the genome language model folks also found that sometimes like the middle layers are much better for homology search versus the last layers. Like so for example, if you take the representations at different layers and ask the question, which one will find the homolog best, it turns out some of the middle layers actually do better job at that. And the reason I'm bringing this up is because I feel like that adds some evidence that maybe, I guess you could say the beginning and middle layers are essentially looking things up. And so if you're also want to look up things, this is probably the best representation to be using. But if you're interested in getting contacts, maybe the last representation is what you should be using. And so I guess different parts of this, yeah. Mm-hmm. Oh, I guess.
Thanks, Sergey, for the great talk. I had a question related to your 3 panels where you're showing folding versus lookup versus motifs. So there are many cases, I think, where we want to learn folding. We want those scaling laws across protein families to be straight lines because that allows us to generalize to more de novo settings and explore a broader part of the protein space. And so I'm wondering if you've had any ideas of how to factor out this information from the models so that the model factor out coevolutionary information so that models learn more physics-based folding. Just yeah. Do you have any ideas of like basically how to avoid cheating on coevolutionary information?
Yeah, I'm not sure if there's an easy way to do that. I mean, one thing we assume is happening is like as you start training the model. So let's say you take a sequence, you mask a position, and you want to recover that position. Like the simplest thing you could do is say, what does this sequence belong to? Like what family does it belong to? So there's probably some version where it's like learning some kind of k-means or some kind of clustering, and you can return that probability. Then as your model gets larger and larger, you say, well, besides just learning like the means of each matrix, you could also start to learn the covariance of each matrix. And maybe as your model gets larger and larger, you could probably learn some trivariance. I don't know if that's a thing, but you could essentially start to maybe learn higher order things. But I don't see at what point will it decide, hey, it's time to not just store the statistics of these families, and when do I start to try to reason why those statistics exist? Yeah, I don't have a good idea of how to make it go beyond that. And does anybody else have any ideas how to push the models to snap out of conservation covolution?
If you add like the mean and the covariance as input to the model as well, and try to, so it doesn't have to learn it. I don't know.
Oh yeah. So you're saying maybe we can freeze this model and then learn like an offset to the model to some extent? Yeah. That's a good idea. All right, next time you could present on it, I guess, see if it works. Yeah, but that's an interesting idea. Yeah, I think. You had a question?
I think we're out of time.
Oh, we're out of time. Okay. So well, we could talk after.
Yeah.
All right. Thank you so much, you guys.
Thank you. All right. So we have two presentations now, 20 minutes each. First one is on computational design of target-specific linear peptide binders from transformer beta by Haowen and Zhao.
Great. Thank you very much. Can you hear me well?
Yes.
Great. Thank you very much for having me present at the conference. And I thank the last speaker for a wonderful talk on protein language model. And I would like to first apologize for not being able to present in person. I'm very happy to share our recent work on the computational design of peptides. In this talk, I will present our methodology for designing binding peptides for a selected target using machine learning methods. Peptides are these medium-sized molecules that are important in therapeutics, diagnosis, and many biological processes. And one of the most important applications of peptides is their use as therapeutic drugs. In the figure on the right, we can see that decade by decade, the number of FDA-approved peptide drugs has increased dramatically. And in fact, during the last 3 years, 17 peptides have received FDA approval, which accounts for almost half of the total approval from the previous decade. The first question, I guess, is why not just traditional experimental methods? Why are we even interested in computational methods? Binding peptides for target of interest have been discovered by rather well-established experimental techniques, such as chemical synthesis or display methods, which involve constructing a large peptide library for experimental screening. These methods have been very successful, but they can be time-consuming and costly. But perhaps more importantly, there are challenges such as the difficulty of targeting selected protein surface and dealing with many hard-to-target antigens, such as certain membrane protein receptors. Screening peptides for these antigens is still very hard, at least experimentally, and computational design has some promise at addressing some of these issues. A previous research which was validated experimentally designed peptides using fragments of protein-protein interactions. In their strategy, given a selected target sequence on the antigen, here I'm showing this in the red, their approach first searches for these interacting fragments in the collective fragment database and combines these fragments together to form a binding peptide, which is shown in the green. These fragments are combined using a set of rules to make sure fragments are compatible with each other, such as overlapping amino acid sequences and matching hydrogen bonding patterns. And if you ask, where do the fragments come from? In the circle here, I'm showing some examples. You can see these fragments simply come from interacting motifs within different protein structures. The design peptides could be used as peptide binders themselves, but in their specific case, they graft the peptide to an antibody by replacing a section of the CDR loop of an antibody with a binding peptide. But what is very exciting is that in the figure here on the right, this approach has been validated experimentally to produce very tight antibody binders in the nanomolar range. However, binding fragments of protein-protein interacting motifs has two limitations. First, fragments that interact with the target sequence or part of the target sequence must be available in the collective fragment database. Otherwise, this search and join method will not design any peptide binders. Second, perhaps more importantly, dependencies between amino acids in the design peptides were not considered when we only locally joining these fragments. So instead of combining fragments together, our solution is simple. We train a machine learning model to learn the protein-protein interactions from these interacting fragments, and we expect a well-trained machine learning model to be at least as effective as a fragment-based approach. Before diving into our methodology, I want to highlight that training a machine learning model on protein-protein interaction data is a highly attractive line of research. To design binding peptides, and regardless of the algorithm used, the data itself has to accurately describe these interactions. However, experimentally solved protein peptide binding complex data is very limited, and data is extremely limited in protein design in general. For example, a complex database called peptide binding database derived from protein data bank contains only approximately 13,000 solved complexes. This number includes cases where the same complex is associated with different PDB IDs, meaning the long redundant data set is likely to be much smaller. However, we have much more protein-protein interaction data within the protein structures and can be used as an alternative training data source. The rationale for doing this can be understood from two perspectives. From a biological perspective, the fundamental forces that drive protein-protein interactions within structures, such as hydrogen bonding and hydrophobic interactions, are the same at protein-peptide binding interfaces. From a machine learning point of view, collecting a database of interacting fragments can be viewed as a way of data labeling of a paired data set of binding pairs. So in the first step of our approach, we start by collecting these interacting motifs, not just from the protein data bank, but from the recent AlphaFold database, which contains predictive structures from AlphaFold 2. We kept only the very confident predictions to make sure that the collected database is of high quality. And here I'm showing 3 examples where 2 of the competency predictive structures were kept in our database. And same to our previous approach, we used a beta strand interaction, which is a stable secondary structure motif, and collected these interacting motifs from different protein structures. The paired amino acid sequences are then stored in our database. For each of the interacting pair, we assume one strand is a target-like sequence. Here it is shown in the red. And the other strand is a binder-like sequence. Here it is shown in the green. And by doing so, the peptide design task, which involves given the target sequence and predicting the binder sequence, becomes a more familiar machine learning task of building a sequence-to-sequence model or building a language translation model. And in the second step of our approach, we train a machine learning model on these interacting motifs, and we use perhaps the most popular architecture in NLP now, which has been widely used for language translation tasks, which is a transformer encoder decoder architecture to nerf on these paired sequences. For simplicity, we choose the original transformer architecture based on the attention mechanism as our model architecture, which can learn the dependencies between different amino acids. Training the transformer uses a self-supervised learning approach, a task called next token prediction. The idea is to show the model the target sequence, hide the next residue in the binder sequence, and then ask the model to predict what goes into that hidden position. And by training this model on these paired sequences, it learns to generate the binder sequences residue by residue. Once the model is trained, during the inference time, we select a target sequence on the antigen. Here it is shown in the red. The target sequence here is input into our model, and we sample different binders that might potentially interact with the target sequence from our transformer model. Before using this model to produce any binders, we want to understand if the model has learned any biologically meaningful information about general protein knowledge. Parameters of the model were randomly initialized before training, and after training, we first get the embedding layers of the transformer model, which transform each amino acid to its embedding representation. What is very interesting is when we project these representations down to 2 dimensions and label them by some key chemical properties, including charge, polarity, and hydrophobicity. In the figure on the left, we see a clear clustering pattern, and the pairwise similarity of these representations also correlates very well with a Blossom substitution score, which reflects the chemical similarities between different amino acids. We then take the average of these cross-attention maps in the cross-attention module and of the transformer decoder, and we observe that most attention weights are concentrated along the diagonals. This means when the model predicts the next amino acid of the binder, the model used actually most information from the facing residues along the target sequence. And this is also expected as key interactions such as hydrogen bonds are typically formed between 2 facing residues in an interacting pair. These are very encouraging, as by training on these sequences alone, the model has learned some biologically sensible patterns. And to understand whether the model captures the distribution of interacting peptides, we compare 3 sets of peptides. One is what we call the natural set, which is a sequence distribution of interacting peptides in our database. One is what we call generated set, which is a sequence distribution of peptides generated by our model. The third is a negative control set of randomly sampled peptides, and we project them together down to 2 dimensions. And here you can see that the overall sequence distribution of these generated peptides is very similar to the natural set and being very different from the random set. We also check the chemical properties of these generated peptides, including charge, hydrophobicity, and molecular weights of these 3 sets. And the figures are slightly crowded, but the figures showing these generated peptides
there is a camera onto there, but I don't know what they're showing. Could go and try and do sign language from there.
Right, and we want to speak to the speaker. Thank you.
Without knowing if I don't know if you can hear that. I'm emailing her.
All right. I guess I'll say hi. I think we might need to join the video though. I don't know.
Yeah. Or actually, if I go on to the, well, no, but he's not going to be on the YouTube. Right?
Sorry about this. I think there's something wrong with the microphone. Great.
Okay.
Great. I'll just jump very quickly to the final slides. Essentially, we perform some discrimination against some randomly shuffled peptides to test the model specificity, and we observed that the model has very good discrimination power when we have some negative controls. But we also observed that the generalization ability of the current architecture decreases as the target becomes sort of dissimilar from our training data. And we expect future machine learning system can train more models on our system. But I think to conclude, I want to say our peptide design strategy is based on training this machine learning model on these interacting motifs, and we demonstrate our model can design some chemically viable interacting peptides. But the specificity of the model is also shown with our negative controls. And we're taking some promising future directions, including carry out experimental validation of some of the peptides, either by testing the peptides and or graft these peptides onto antibody, as done in the previous research paper. And we are also exploring using other structural motifs, not just beta strand interactions, such as alpha helixes. And I think a very interesting extension of our work is to actually train this on protein peptide complex data, which has not been done in our paper. Great. I'm happy to take any questions, and sorry for the interruption.
Any questions?
Hi, nice talk. Have you benchmarked against diffusion or any structure-based methods for peptide design?
Great. So thanks for the question. We have not benchmarked in our paper, but we have some early exploration of some of these models when they are trained on very small data sets.
I think it's on his side.
Okay.
It's on his side, I think.
Yeah.
It's okay. We can.
Yeah, we can just move on.
All right. I think we'll move on. Hopefully, we can maybe chat some questions over to the presenter later. But that was a good talk. So we'll then move on to the next one. And this is the last talk of this session. We'll have a coffee break after that, and then the panel. So this one is on predicting cell cycle stage from 3D single nucleus trained images by Gangli.
Hello, everyone. Thank you for the introduction. My name is Gangli. I'm a postdoc in the Bill Noble's lab. Today, I'm going to talk about our work to infer cell cycle stage from single-cell nucleus stain images. And today, my talk is going to consist of 3 parts. First, I will introduce the existing tools for the cell cycle prediction, and then I will introduce our method to predict cell cycle from the Fuji cell lines. And ultimately, we want to extend our work to other cell lines or mouse tissues. So first, I want to give you a brief review on how cell cycle, what is cell cycle. In the cell cycle, each cell grows and divides, and it has been known that it can be classified to different stages. For example, in G1, cell grows and prepare for the DNA replication. And in the S phase, DNA are replicated roughly twice. And in the G2, and cell grows and prepare for the mitosis stages, and M represent the cell division occurs. And there's also a small stage called G0, which represents when cells stop dividing. And there are many tools have been developed to estimate cell cycle stages, particularly from the single-cell transcriptomics data. Here I list 6 methods, but there are many more of them. However, a lot of those predictions from several methods don't agree with each other. And this is one way to infer cell cycle stages, but we are going to focus on how to infer cell cycle stages from image modality. One method can be used to infer it from the pCNA images, and research group from the UNC Chapel Hill, they have been able to use pCNA to assign cell cycle stages to study human cell cycle structures. They identify this manifold where you can see the cell cycle structures here, and including the G1 synthesis cells on the tail of the left side. And they are able to classify cells to 3 categories, G0, G1, S, or G2. And this has been published in the Cell System 2 years ago. And we particularly focus on to infer cell cycle stages from the DNA stained fluorescence microscopy images. And DAPI is one of the most easily to use and easy to get signals from the images. And this has been done to use, in 2015 Nature protocol paper, they have established a method using Gaussian mixture modeling to first get the integrated DAPI intensity after the 2D maximum intensity projection. And they provided a commercial software called FCS Express to modeling the 3 class cell cycle stages and estimate the cell cycle labels for each single cell images. Here, I summarize all the existing tools to predicting cell cycle stages from the DAPI images. However, those existing tools have several limitations. For example, most of the existing tools focus on the 2D maximum projections, which throw away a lot of the 3D cellular image information. And those image data sets are not directly available. You need to send emails to them. 2 of them did not reply to our request. And also, it would be interesting to see if we can extend these models to other cell lines or tissues. So to address those limitations, we developed a machine learning framework called Cell Cycle Net, which we release both our data and models, and it has already been put on the public GitHub repo. And we try to use 3D image information directly to improve the cell cycle inference. And we evaluate on both epifluorescence and confocal microscopes. So now I'm going to introduce our method. Before that, I want to show you how do we provide the ground truth label for the cell lines we collected. We specifically utilize the Fuji-Zhou system to provide the ground truth cell cycle labels. Here on the left, it's a cell imaging movie. As you can see, it's measuring the HCDT1 and HGEMINI, 2 signals to provide the ground truth cell cycle label. Initially, the cells are in red color, classified as G1, and then in the later stages, they show more green signal. They are classified as SG2M. So following the recent paper on Narutomo et al., we are able to classify the Fuji cell line data to 2 categories, G1 cells versus SG2 cells, using the Fuji system. And we collected 2 benchmarking image datasets with Fuji-Zhou systems. And here are the distribution of G1 and SG2 dataset in the 2 most widely used microscopes, epifluorescence and confocal. And in both 2 datasets, we have a slightly more G1 cells. And next, I want to show you the data set we collected. Here is 2 morphology spaces. On the x-axis is the normalized integrated DAPI intensity, and on the y-axis is normalized volume. And as you can see, the green cells are SG2 cells. They are usually typically larger and brighter, and the purple cells here are the corresponding to G1 cells. We use Apricot, which adopts a submodular optimization algorithm to highlight the top 10 representative cells in this 2-dimensional morphology space. And here are those 10 representative cells we selected. So now I'm going to formally introduce our cell cycle net, which takes the 3D single cell images directly to the input, and we fine-tuned a UNet model to predict the binary classification label, whether cells belong to G1 or SG2. And specifically, we utilize this UNet encoder, which to learn the 3D cellular information. And we fine-tune an existing UNet, which have been trained on some cellular images. This have been published in 2011. And our baseline model is one of the previous tools, SVM model. This model takes two morphologic features, the 2D depth intensity and the total cell area as input, and train a spot vector machine model and output the cell cycle stages. And here I'm showing you one decision boundary using those two morphologic features as input to predict cell cycle stages. And one of the data set, epifluorescence data set, here I'm showing you the RC curves of our model, the blue curve, cell cycle net, compared against with red curve, SVM curve. As you can see from RC curve, our model significantly outperform SVM model in terms of the classification performance. We achieve 0.95 error under the RC curve. And on the confocal data set, we achieve a similar performance improvement with our fine-tuned cell cycle net. And here is a test data set accuracy comparison. I think you can see both two data sets, our model are significantly outperform the support vector machine model. And next, we investigate whether our prediction errors are uniformly distributed across those two morphologic features. Here it's a same scatter plot, but colored by whether the cell cycle net is making correct predictions versus incorrect predictions. As we can tell, those black dots are uniformly distributed in this 2D space. And similarly, trends are observed in the SVM predictions, but we can see when we compare the predictions of cell cycle net versus SVM, here blue dots are the ones where are making correct predictions and SVM are making incorrect predictions. This improvement seems also be uniformly distributed in this 2D space. And one of the questions we have is whether we can incorporating 3D features to enhance cell cycle estimation purely from their SVM model. As this intermediate experiments, here the original SVM pipelines go from the 3D depth image. They do the 2D maximum projection and 2D segmentation to get those two morphologic features and train SVM to get cell cycle stages. So we proposed an intermediate experiments is to do 3D segmentation and also similarly extract two morphologic features, but it's 3D integrate depth intensity and the volume of the cell images and train an SVM model to see how it performs. Interestingly, that we found that using 3D features with a purely SVM model significantly outperform using the 2D features for both data set. The blue curves represents the epifluorescence data set, red curves represent the confocal data set, and the solid lines are the model with the 3D morphologic features, dashed lines are the 2D morphologic features. And we also find ensembling those four morphologic features could further improve the performance. And here the blue bar is the SVM model with all the four morphologic features I mentioned before. And the rest four bar are just simply taking each morphologic feature to train SVM model. As you can see, the 3D integrated depth intensity is the most predictive single feature, and the cell area is less predictive compared with other features. And last but not least, we want to test the generalizability of our cell cycle net models. And we found that our cell cycle net model can benefit from learning across both data sets. Here, we are doing a training scheme where we are training on the confocal data set or training on both data set and test it on the epifluorescence data set. And the dashed curve represents the model trained on both data sets. As we can see, for the SVM, the red curves, the model trained on the other data set or both data set doesn't seem to change too much. But for our data set, if we can join training from both image data set, the model got significant improvement in this generalizability task. Similar trend if we train on the epifluorescence data set and apply to the confocal data set. So after I establish our cell cycle net method, next I want to show how we plan to extend our current framework to other cell lines or tissues. Specifically, we want to extend this to the tissue environment. Here is mouse organ images. Here we have the DAPI signals. It's easy to extract the DAPI signal. However, it's hard to get the cell cycle dynamics. And you will be able to get different different morphology of cells. And the next question, whether the model can accommodate for those different morphology features from different kinds of cells. And we are able to use a Fuji-2A cell cycle reporter model string for the ground truth. Here I'm showing you an example where we see in this mouse embryo images, there are certain regions are enriched for G2S cells. There are certain regions are enriched for the G1 cells. And we will generate those kind of images for the whole embryo scanning. And in summary, I think today I have shown you we collected those both epifluorescent and confocal image dataset with Fuji-2A labels. And our cell cycle net achieved the state-of-art performance. And we generate Fuji-2A mice and harvest embryos for imaging. Ultimately, we want to study the cell cycle dynamics for each cell type and tissue. And our code is available on GitHub. And our preprint is also available from this QR code. And with that, I want to thank my advisor, Bill Noble, and Brian Belleville's lab, Wall, Nico, Eva, Connor, and all the Noble Lab members. And I also want to let you know that our Noble Lab have 4 postdoc positions. If you are interested in the above-mentioned projects, you should talk to Bill Noble here in person. And I'm happy to take any question from here.
Great, thank you. Questions? None? Okay. All right. So everyone's ready for a break. Okay. We'll thank the speaker again. Thank you.
Thank you.
So we have now, what, a half an hour break? 20-minute break, and then we'll be back for the panel.
I'm also close to neutral as well.
Why don't you just play devil's advocate?
No, I can try, but I just feel like I wouldn't do a good job. I'll also do devil's advocate.
All right. Can you hear me if I use this? I think this is okay. Yeah, okay, great. So let's get started. I'm really excited to have our panel discussion on foundation models. So I'm James Zhou. I'm happy to be a moderator for this panel, but I'm really just going to try to keep people asking questions and encourage everyone here to speak up. So the context of this is that there's a lot of developments and excitements around this foundation model paradigm in biology, right? In a nutshell, right, the idea of trying to train larger models on large scale of data usually for self-supervised kind of tasks, right? And then using those models or representations for quite diverse different downstream applications, right? And we actually have seen quite a lot of nice talks that talked about foundation models earlier today from some of the spotlights and oral sessions. So what we want to do in this session is to hear from several experts in the field, Sergey, Anshul, Maria, Suyin, and Jian, around their thoughts on the promise, both the limitations and strengths of these foundation models, and where they think are the most promising directions going forward. All right, so I'll start off with a few questions, hopefully to get people started thinking, and then encourage you to think about questions so we can you can ask the panelists. So maybe I'll just start one with the question around, what is the current state of art for foundation models for genomics, right? Basically, how useful are the current models for genomics, And do they really solve some useful tasks, right? Maybe if Ansho and Maria wants to start, but other people can also feel free to chime in.
All right. So I think we have 3 basic types of models. There's the protein, the single cell, and the DNA. And the utility of them is widely different, right? I think we can all agree that protein models have been extremely successful. I use ESM. I don't even work in proteins. So I think that's been really amazing. So single cell, we had a couple of talks last year in exactly this conference, benchmarking those models. They seem to really not do very well. And I think that's a little bit surprising. I feel like you should be able to do better in that space, and maybe that's something we can rethink a little bit. And the DNA is kind of like the final frontier, and I think that's just extremely challenging, especially when you go to mammalian systems. This has been alluded to many, many times. It's just most of the DNA is not constrained. It is literally just random. So you could try to predict some coin flips instead, and spend some time doing that. So I think we just need to, obviously, like, there is some kind of future where they all work, but we may need to think a little bit more about how to go about it more efficiently.
Yeah, I don't have that much to add other than to say that I'm actually a fan of the self-supervised training objective. I think that's very powerful. And I think when you match that objective with the way you select data and the scale of it, the protein language models, I think, are a good example of that. You can end up with something very powerful. My view is that the single cell foundation models are just trained on the wrong data. I think observational RNA-seq is not the path to glory. Like, it's not going to learn really causal models, things like that. And so I think it's worth thinking about what are the key applications that different classes of foundation models could solve, and what are the right kinds of data and the amount of data that you need. The DNA language models are much closer to what we work on. And again, I think there's a lot of promise there. My biased personal view is that to really get them to work, they cannot be annotation free. They will not be completely unsupervised. There will have to be some assistance provided based on tricks to find potentially different classes of elements, and then bias the models in terms of how they're sampling, things like that. So I think to be more neutral, I think there's a lot of promise, at least on the DNA front. I think it's very early days, and it's going to all come down less to, I think, architecture engineering and more to how the problem is set up and how the data is curated and how sampling and so forth work. And that might be the case for single cell too. Maybe if they're trained on multimodal perturbation data sets, they will probably do much better than observational stuff.
I just want to add, I guess, we didn't really define foundation models, and I was really happy to see Informer and Borzoi proposed to be in the class of foundation models. I tend to agree with that. I think a lot of people don't, but I'm not really sure why. I mean, they're trained on lots of data, and those models are actually incredibly useful. So I think, yeah, if we broaden the class to include these models, which, okay, they're supervised, but I don't know, there's actually a really fundamental difference there.
Okay, great, thank you. I guess both Ansho and Maria mentioned sort of protein language models as sort of maybe the one shining example of the success stories. Right, so I just want to push on that a little bit more, and maybe it would be good to hear Sergey's thoughts on this, is that certainly they have been quite successful, but how far do you think we can push this pure language modeling paradigm even for proteins? Right? At some point, do you think we just need to bring in additional information like structure to really get these models to work?
I guess I have to answer that. Okay. I guess to echo some of the earlier points, I mean, I think self-supervision is definitely very powerful, but I think the key part is it's really easy for models to cheat. Like even the gremlin model that I talked about, like you could just learn an identity mapping. So I think it all comes down to how you mask things. And so you mask something, you try to recreate it. And so for example, the GLM2 model, like we find that it doesn't really do good at RNA structure prediction because you're essentially masking giant chunks. And so there's no reason for it to learn individual positions. And vice versa for EVO model, you're not masking entire codons, and so in that case, it has no incentive probably to learn things. So I think in proteins, I think it's really successful just because It's a really straightforward approach. Like, you just mask the amino acids, and you know those are the amino acids that actually interact with each other, and it's one-to-one. But with DNA, it might be more complicated. It might be more like, maybe it's not individual nucleotides you have to mask. Maybe you have to mask them in some smart way to prevent it from cheating and learning something too easy or too obvious. And so I think that's why the genome language models are still not there yet, just because they're not. But I guess the real question was proteins. And so to get back to proteins, I think there is a part to all of this where even masked language modeling is kind of an approximation of pseudo-likelihood, and pseudo-likelihood is also an approximation of likelihood. And we're kind of making this assumption that every position is almost independent of other guys. And so by definition, we're kind of forcing the model to only maybe learn pairwise interactions, because you're essentially just masking one thing at a time. And also our architecture is attention-based, which is also pairwise. So I feel like there might be more things to learn about proteins that the models can't capture because of the loss function and the architecture itself. Yeah, okay, I think I should stop rambling. Okay.
Do other people want to add to these points? I guess maybe a related question, right? As we're thinking about the value of these foundation models, is really thinking about the difference between these sort of more, let's say, self-supervised pre-training versus more training very task-specific models, right? And maybe this, on that point, I also love to hear from Jian and from Suyin. Is like, where do you see, which approach do you find more promising, especially in biology?
Okay, so I think that was the main theme or takeaway message of many of the talks we saw today. I mean, whether to use the foundation models at all, or should we fine-tune more, or should we just get rid of it and then just forget about it? Sounds like, hearing from this world-class, experts in genomics and proteomics, it sounds like, proteomics space is a little more successful than in genomics space. So I think the more relevant question is what to do. As a dedicated scientist in this, MLCB community, I feel that, the major, the strengths. I mean, we definitely see transformative potential of foundation models or pre-training, or self-supervised learning in many different fields, not just computational biology. So there are definitely, pros of this new technology. But like other, machine learning or AI technology, there are pros and cons. And we definitely see some concerning aspects in terms of these embeddings learned, pre-trained, don't have much information, and they are not very transparent or interpretable. So I think what we need to do is to understand clearly all this success and the failure modes of this foundation models, and we can do that with existing tools in AI or machine learning, but we could also, think about how to advance AI. We have seen a lot of, success stories in the field of computational biology where we actually advance, the state of the AI field, how that can, have a significant impact in biology. So as, many of you can imagine, my answer would be that, how about we focus on explainable AI techniques or, interpretability methods to actually, make these foundation models more transparent so that, for example, you know which training samples are responsible for a certain output we really, don't understand, or how to, understand this like black box embedding spaces based on whatever, some sensible, biologically meaningful aspect. So I think there's a lot of room, to improve, that, the principles and techniques in the way we diagnose or interpret these, multimodal foundation models.
I think also in the same directions about what to do. I think thinking about foundation models versus like test-specific models, and maybe you can also just generalize a little bit more from like foundation models that are purely pre-trained self-supervised model to just maybe a generalist kind of model that we train one model that can be applied to many downstream tasks versus developing very task-specific model, like just focusing on one specific kind of biological process, right? I think I don't really see that. I think there are values in both kind of directions, right? I do expect that we will see development on both directions. I think it is just like for us, for people who develop methods, I think it is sometimes hard to just cover both the breadth and the depth, right? It's hard to just tailor everything. If you are modeling 10,000 different things, it's hard to tailor, introduce your biological prior and all those understanding to all of them. So you might just use one architecture that can fit all of them, right? But whereas when you are studying one specific system, you might try a lot more and try to incorporate a lot more about biology that you understand into that. I think in an ideal world, we can I think have task-specific model for everything, right? And they all work well with each other, and they can communicate with each other. And I think that would probably be the best from my point of view, right? But I think we will not get there in one day. I think we probably will have those generalist and specialist models developed sort of in parallel, but maybe they will converge more and more toward each other, where the specialist model can talk to each other and share information with each other, and the generalist model might be more specialized toward more specific tasks, right? And yeah, I think also kind of implicitly conveyed my opinion that maybe you want to build in some biological prior, right? Instead of just let the model learn everything. Like I think a lot of things about foundation model came from the experience from large language model, right? So the scale is all you need. Attention is all you need. So I think when data is, I think the most limiting factor, you just want the model to be big enough and powerful enough to utilize all of those data that's available. I don't know in biology whether we have the same sentiment that the data, we have too much data that we cannot use it by the current model. That's maybe also a question for all of you. I think, I don't know the answer, but I definitely feel like I think in the data that I'm familiar with, I still see, I think, overfitting all the time. I think it's not in a regime that we can just scale up everything and just expect it to work.
Yeah, can I just add one thing? So,
yeah.
and you know, James, you might know a lot about this too, but I think even in the language model space, if you're seriously critical about how these models perform and how they're trained, it's whack-a-mole, right? Like you train these models, and then the amount of engineering that goes into not just the design of the model and the engineering of the training, but then the engineering of the data set, and then the reinforcement learning on top of that, and then all the other filters it's literally whack-a-mole. You don't know what they're exactly doing, and every time you find a mole, you whack it, right? And then some other mole pops up. Like, is that how we want to do biology? I don't know. Like, and in natural language, at least we understand the language. So like, we can see the mole. Like in biology, a mole will pop up, and you wouldn't know it's a mole. It just looked like, oh, this is cool bio, right? And it's like I think even more dangerous to have models. I mean, dangerous not in like a fatal physical way, but in terms of like scientifically to have models that are really unreliable in terms of what they encode and not really fully understanding how they do it, right? So anyway, that's my view, but I know you've done a lot of work on biases and things like that on this too, so yeah.
Great, great comments. People should start to think about questions that you want to ask the panelists. But I just want to actually quickly follow up on something that's both Zhou and Jian mentioned, which is the quality of the data, right? And also the scaling of the data, right? Because a lot of the sort of the underlying thesis, right, of foundation model is that somehow if you keep scaling the data and compute, maybe you get some emergent behavior, so you get better performance. But how much of that do we believe is true, especially in biological applications, especially if the underlying data, as we know in biology, is often tends to have all these artifacts and confounders and potential biases?
Maybe I'll start a little bit. So I think in the models that we built, I think for the genomic sequence, a very obvious constraint is how big our genome is. I think so for the model that we built for the human genome, we're really limited by how many base pairs are there in the genome, and also how many genes are there in the genome, right, which is much smaller number. And I think it is one of the reasons why it is so hard to get the gene expression prediction very accurately. You just don't have that much, that high number of data points, right? So I don't think we have depleted the current possibilities yet, but maybe there will be one day that it's just important to get a lot more data. If I can dream, I would want to maybe like synthesize lots of DNA sequences and put them into the chromosome. Like we have long DNA sequences that are designed that are maybe like, we can put up as much as 100-fold of how human genome size is and collect all those data, then that would be great, right? That might solve the problem. But of course, there are lots of challenges here. So both technically, like how do you actually synthesize and put that amount of sequencing in the genome is still challenging, and also it's also a computational problem. So what kind of sequence do you want to put into the genome? It's probably not random sequence, but beyond that, what do you do? I think it's, yeah, I think to me it's, yeah, at this stage, I can only dream, but yeah.
Yeah, I mean, I have two thoughts on biases. I think one is in the unsupervised setting, as we've discussed multiple times. There are biases just in terms of the sheer prevalence of different classes of elements and how they're distributed across the genome. And of course, that also varies across species. So there's selection acting very heavily on a species. The genome is already cleaned for a lot of garbage DNA that's sitting around. But there are, like, mammalian genomes are full of what appears to be potentially garbage, right? So that's just simply in terms of biases of how you train the models in an unsupervised way. And then there are biases coming from the experiments that we use for supervising itself. And in that case, I actually think it almost flips on its head. That is, since the genome has so much of effectively neutral, non-functional DNA, you get a free experiment that measures what signals look like from different measurement technologies on random DNA, like that is not really functional. And I think one thing we stumbled across is if you train powerful neural networks on that background, you can in fact learn biases extremely well. So in other domains like natural language, the biases are built into the data you train on, but the genome does provide this nice compartmentalization of functional DNA and random background, and every experiment you do effectively gives you readouts on both types of sequence. Then maybe there is a more effective way of separating those biases out, at least to correct biases in experiments. Now, I don't know still how to correct biases in the underlying sampling of the genome, but at least that's maybe one advantage.
Yeah.
Yeah, so in large language models for text, they found that certain types of data are much more useful than others. For example, like Llama 3 was trained, I think like 50% of its training was on code, which is really interesting. And they found that that was extremely useful for the general task of intelligence and downstream tasks from there. I was wondering what you would see as like, so yeah, like basically the really high, what is the analogy in biological sequence space where what are the really high value sequences you're interested in, and what are the sequences that you want to filter out of your training?
I mean, I guess it's just what we were saying a little earlier, like I guess there's parts of the genome that maybe doesn't have as much structure or as much selection. And so if you train on that, you might not learn anything useful, I guess. Is that I think, the argument Anshul was trying to make? Yeah, for proteins, I guess they're usually pretty useful, unless there's like disordered regions. And then it's actually disordered regions to have a hard time learning anything there. But it's not clear if it's not learning well because it's just not, like the objective that we're using that mixed language modeling is the correct objective to learn things about those regions. like if you imagine things are moving around, maybe instead of masking, you want your perturbation should be also motion that you correct on the other side. And so maybe just the wrong objective. But yeah, otherwise I can't think of any other analogy that. Does anybody else have a?
Do you want to go next?
So I think Anshu alluded to the fact that a lot of mammalian DNA seems to be kind of just junk and it doesn't do anything. And like one example I think that comes to mind is that there's all these repetitive elements. And human genome are just like tons of them, and mostly they're tolerated, right? Like that's how we think about it. They're tolerated because they're inserted in some position where they just don't cause any damage. But actually, it turns out that they're like in some cases they are functional, possibly like through some evolutionary accident. Like apparently this is why we don't have a tail, right? It's because of like an allo insertion. This is a very recent paper I think from last year. And so I think this question of like what is junk and what is like you really like. If you just ignore the repetitive elements, you would never be able to learn about this tail, right, phenomenon. So I think it's kind of challenging.
I think one thing is to potentially think of using these models to help design experiments. I think to me that is the most proximal immediate utility of them. Like they're obviously learning some interesting biology and they help us understand that more, but I do feel like they also really identify regions of the space that are undersampled, that clearly the models aren't good at. And luckily, in biology at least, we can generate our own data. So like, I mean, not maybe people in this room, but in general, we can. So it might help to actually push that paradigm much faster, as much as we're pushing model building, right?
So I have a very basic thought. So we've been defining foundation models based on size of training data, number of parameters, all of this. Is this even the right way to think of a foundation model? Like, shouldn't we be thinking about, is it telling us something foundational about the underlying biology? So let's say thinking about single cell models, like cell type classification or cell type annotation is a interesting, but also to my immunology colleagues, a very boring task. They can use flow cytometry, multi-panel flow cytometry to just tell you exactly what cell subset it is. So shouldn't we be assessing whether a model is truly foundational based on a hard downstream task, irrespective of whether it's a billion parameters or not?
I think that's a good point. Yeah, I mean, I guess for proteins, I would say like a foundational model is something that would work on anything. Like it would understand the physics from first principles, maybe figure out how proteins fold. Maybe instead of foundational, we could call it like a knowledgeable model. So I mean, it's just sort of summarizing all the knowledge that's accessible, like be it human language or in the case of proteins, like everything evolution has ever done to protein sequences. It's just summarizing all that information for you. And in some ways, if you're like an evolutionary biologist, that's great for you because that's actually what you want. You don't actually want something that nature hasn't actually sampled. You actually want to stay within and understand those constraints really well. So I guess maybe it depends on the question that you're asking. So for some people, it's foundational. For some people, it's not foundational. If you're a physicist versus a biologist or evolution biologist.
All right, maybe I can also add to the confusion around foundational models, or I'll start with a question. In your day-to-day, how much do you think a new foundational model will change, or how much benefit will you get from that versus improving data curation, improving the pipeline based on your task, et cetera? In my perception, a foundational model is a model that gives us a salient representation of the data, irrespective of the model size. But that's just a representation. It still depends on what we use it for and what we train it on.
Yeah, I mean, I'm fully in your camp. I mean, I think you have to first define the problem statements and then design whatever kind of model you want in that space and then evaluate accordingly, right? And also amass the data in a way that helps you accomplish that. I again think like the protein language models have been a very serendipitous success. I don't think, I mean, maybe I'm wrong, maybe you know the history better, but it may not have been obvious at the time that they would be this generalizable, right? Maybe to some extent, given their relationship to previous simpler approaches like Gremlin and things like that. But it has, I think, triggered a movement where the same strategy would just apply elsewhere. And it's not clear if the data design or the problem statements are a good fit for that strategy at all.
I guess it depends how you define generalization, because I guess I would argue it's not generalizing. And it's essentially just learning things. I mean, maybe learning things more efficiently, maybe more end-to-end, but I guess it depends what you mean by generalization, I think. Because, like, for example, somebody would say something generalizes because it's capturing the constraints, but not necessarily memorizing the individual sequences. Like, you could say, okay, I've learned the conservation coevolution for this family. That's generalizing for that family, but it's not generalizing to other families, right? Or, and memorizing would, on the other hand, would be like, oh, I memorized all the sequences without learning the underlying, I guess, the statistics or pairwise statistics about them.
Yeah, I guess I was coming at from a fine-tuning / probing scenario, right? Where there's plenty of evidence that like zero-shot, few-shot on protein representations work surprisingly well. So they're learning something fundamental for the problems that are relevant in the protein space. So in the single-cell space, what are those relevant problems? Is cell labeling the most relevant problem, or is it everything else that we did generate single-cell data for? The question is, does it solve those better than the trivial problem, right? In the protein space, you can solve many relatively non-trivial problems by piggybacking off the representation. So that's what I meant by, I guess generalization is the wrong word, but maybe useful representation, yeah.
I think my question was, I think, on the same line as the last question. So about that, okay, I think we are spending so much effort, like in terms of resource, into training these language models. I think a GPU 256 core takes like $250,000 for 3 months if you use it for 3 months. But we don't do like, more experiments for something like ATAC-seq and just create higher quality data. So I want to just give one example is that we have been trying to account for personalized genomes, like, into models like Informer, but many efforts have been in vain so far. Maybe we are seeing some of the efforts that are coming up recently. We are yet to verify, okay, how well they compare against training to reference genome. Now, what do you think, like, why all the efforts so far has been just in vain? Like, why is it that, okay, whenever, like, you train on the reference genome, you just achieve the best performance compared to like accounting for individual genotypes? Is it because that, okay, the data sets that we create, like for different cell lines from different individuals, that, okay, the noise in the data that are so high that, okay, it's masking the effects of individual genotypes, and maybe we need to increase the quality of the data so that we have those, like, effect of those individual variants, and then if we train the models on those, then maybe we'll be able to learn them. So,
I think the multiple genomes is a really interesting question, because obviously, like as Jian said, with the single genome, we're very data limited. There's only so many genes. I can't like get more genes for my genome. I can get more genomes. That's But yeah, I mean, with different individual, right, it comes with a bunch of like all the variation is coming. Some of it is genetic, some of it is non-genetic, some of it is genetic but is in trans, and there's no way you're going to capture that with the simple models that we have. But I think there is these kind of more productive avenues, like F1 hybrids, that people actually use quite successfully in the space.
And what is your thought on, like, annotating, let's say, something like random genome to create a baseline model, and then create new synthetic DNA to map, like, more and more of the cis-regulatory landscape, like,
I guess, I don't know, maybe people know more about synthetic DNA. It's quite complex, right, because you have to, ideally, you want to put in synthetic DNA, you want it to integrate, you want to maybe integrate in a specific location and really sample all the possible combinations and really look at some, like I want to make a change and enhance it kind of quite far away from a gene and see what happens and really do this at scale. And I think that will happen eventually, and I'm not sure how quickly. Maybe somebody else can comment on the technological progress there.
Okay. My question might be a bit too futuristic, but I'll ask anyways. Big companies like OpenAI, Facebook, Google have started discussing about AGI, artificial general intelligence. As computational biologists, when do we expect to start discussing the AGI of biology? Thank you.
Everyone is looking at me. Can you define AGI in this context? What do you mean by AGI in computational biology?
Like I said, AGI in the context of biology, more like a this is something I envision sometimes when I'm alone in the room, like a model that could understand the whole human body, the genes, everything about a human.
I think it's a very long, long way to go from now.
Yeah.
I mean, I know you just said, futuristic view, and not sure if I directly answer your question, but hearing all this, questions and then all these concerns, and I feel like it's very problem dependent. Whether the foundation model is successful, whether we need to train better or gather data, cleaner data, all these questions, it's really problem dependent, and there is no way to even many of these questions, no way to actually address these questions. So I feel like we really need the principles and then really rigorous machine learning techniques to verify the foundation models are actually good. And they're usable for any particular, task, either genomics or transcriptomics or proteomics, some, standardized principles for that. I just get feeling that we really, need, there should be a whole area. For example, we don't even know, how fine-tuning should be done, right? Is there any, theoretical guarantees or theoretical, theorems that, guides us in terms of, which part of the network should be fine-tuned, or, what level of fine-tuning is going to be enough for addressing this kind of biological question? We don't know, right? We don't have solid machine learning, principles. I think it might be good, for us to think about, like a set of questions, tangible, machine learning questions to actually, advance the field through foundation models. That's, I think, a pathway to this AGI in biology.
I just want to add one thing. I think you're going to ask like when are we going to have AGI in chemistry, which is a much simpler problem, and I think we're like not close there. So, but I think maybe that will happen first.
Yeah, I think a more proximal goal is, again, to just use all this technology to just increase the efficiency of the scientific process. So like right now, we do kind of like brute force / intuition kind of strategy for doing science. And in the end, we're experimentally limited, right? So if you could be better at suggesting the next best step that we can take, and then the success rates of our downstream experiments, which are really the bottleneck and the time-consuming piece, if that becomes 10,000 times more efficient, AGI, right? That's like the human-computer AGI for science. That's to me is the ultimate goal, right?
I wanted to go back to the generalization question a little bit. So my takeaway from the benchmarking talks this morning, as well as the benchmarking papers that came out recently, is that many of these large language models on DNA are overfitting, right? Whether that's memorizing pieces of the genome or overfitting in other ways. So, and the standard machine learning solution to this is cross-validation. So you could imagine a world where instead of training one model and maybe testing one or leave one out, you train an ensemble of models, each one leaves out a chromosome or something, and then you report only the test the prediction on the test case set, right? So why is this not standard practice?
So it's maybe easy to do it on one genome. As soon as you go to 2 genomes, 3 genomes, multi-species, what is actually independent and what's not becomes extremely fuzzy, right? In fact, one of the applications of these models is to understand evolutionary relationships between sequences. So like, should you hold out things that are in Hamming distance, or should it be in embedding distance, or should it be in alignment distance? It becomes very difficult to construct a single set of cross-validation schemes that really test the models. So I guess the solution is maybe to create many, many different tests with increasing difficulty to see, how the models generalize. But I agree with you overall.
Yeah, I would say in the protein world, we often don't really have to do that just because we know there's a whole bunch of proteins out there that are orphans that don't have any neighbors, and so if it's nowhere similar to your training set, or there's only a few examples of those training sets, like those curves that I showed during my talk, it's essentially just saying how far away are you from the training set, or how many examples are there in the training set, and so. But that being said, people do leave out entire families, and usually they don't do well on those. But I guess the question is, like, do you want to store that information, that family, or not?
I'm thinking primarily of supervised models like Informer or similar classes of models.
Yeah, I support more than a single test fold for many reasons. But I think most of the models do at least have a validation and test fold, but it's usually one separation. And it's worth testing these out in more adversarial ways.
I mean, what I'm seeing is the comparisons of models based on training scores, right? And benchmarking models based on in-training data. Is that not accurate representation?
No, I think at least in most cases, the evaluation schemes are sound in that way. Most of the leakage is either cryptic, like some people talked about pre-training leakage, right? Or other kinds of like homology that you don't realize exists in the training set. So it's not intentional, and it's not like bad ML. It's just like structure of the data is difficult.
Hi. So large self-supervised models give us a number of new tools to play with. The ones that you hear about the most often are generation and transfer learning. But in natural language processing, the idea of algorithmic improvements based on the vectorization of data has been really prolific. So like semantic search. You don't see as much of this in biology. And so I'm wondering if you see opportunities for algorithmic improvements based on large self-supervised models in biology.
I think there's one case that's really taken off is just using protein embeddings for similarity instead of doing some kind of homology detection and alignment based. And that's been incredibly powerful and really simplifies kind of a very annoying task.
Hi. I have actually a specific scenario of using those large language models or foundation models. So let's say we've generated some functional data on top of DNA sequences or protein sequences, but we don't have a lot of data. And we want to train a model on that. One way we can do is to input those sequences into the foundation model, ask it to generate a lot of predictions, or ask it to output the embeddings and train a small model on the embeddings or the predictions on other features. And comparing this, generating a small model on embeddings to generating a different model or distinct model directly on the sequence data, which way would you go, and which way do you think is better, given that the large language model or foundation model has learned something and the embeddings are meaningful?
I guess I would say one technique that's been getting really popular is to not fine-tune models, not necessarily to train on representations, but to try to train adapters to the model. So the idea there is often, I guess the assumption is that your model has sort of learned the entire space, but maybe it's not aligned specifically to your problem. And so you could essentially shift some maybe internal biases in the model for your problem. And so there, you prevent the model from forgetting everything, but you sort of bias it towards your desired task. And for those kind of things, so these are techniques often referred to as low-rank adapters, and the idea there is that you don't need as much data because you're not necessarily retraining the model, you're just shifting, I guess, the internal representations in the model. In our field, it's becoming more and more popular, and people are finding that it definitely prevents like overfitting and all that kind of stuff. So that would be something to try.
Hi, I wanted to ask about, so we've been talking about getting more genomes or more patients for making the DNA language maybe better. We have a lot of proteins, so that maybe that's why those models work well, not just. But and we've talked about why for the it was difficult also for the DNA language models because we didn't know how to get to the masking well compared to the proteins. So in that context, is it possible? We haven't talked about the single-cell language models except saying that they don't work super well, maybe. But we can get a lot more cells as well. And is there an opportunity for thinking about the single-cell data as a way to align those two type of models, given that DNA language models and protein language models, given that you can think of a cell as a bag of protein and a huge sequence, I guess. And it's loose thinking, but the key would be that maybe the ATAC-seq and RNA-seq, if we have some multimodal data, could kind of help, just align those things together. So it's still pretty naive, but like, do you see that there would be a certain assay that would help kind of align those two emerging language models, and that finally could make single-cell language models more useful?
You want to say something? So I agree. I think actually sequence might be the unifying theme there. And how exactly to do it is unclear, and whether we should be learning all of them simultaneously or separately and then fusing them in some ways. But for example, I definitely feel like the current single-cell foundation models learn decent cell embeddings, at least within the space of cells they're trained on. I don't know if they generalize out. There are claims, but that's... So given that information, maybe, for example, when you're training sequence models, you can provide additional tokens, conditioning specific elements active in specific cell states through these cell embeddings, right? That would be like one example. But I think there are many interesting creative ways to link these types of models together. But I think all of it is conditional on whether the base embeddings that they are learning are good enough. And then if we're training them all together, it becomes even more difficult to understand how to control learning of each of those components. But I think it's a great idea. I mean, eventually maybe you'll figure out how to do it, but it's certainly the holy grail, I guess. That would be the mega foundation model of sorts.
Let me add, I was almost thinking, Roman, you can, that can be your next R01. It sounds a very innovative approach, and then I think, aligning, different modalities through foundation model can be a really, I haven't come across any specific paper on that, but I think it can be a really good future project. And I think one of the strengths of the foundation model is that it provides a tool to bring multiple modalities together into one model. So then we can now then think about what modalities, what are the missing modalities here, which is actually one of the questions James provided before this panel. So what missing modalities can we think about in biology? And I think that when we think about like multi-model foundation model, there should be anchors, right? I mean, you will gather data sets from multiple different sources covering different modalities, and the question is then, what is the most common modality to use as anchor? So that's something we can all think about.
Yeah, I guess if I was to add something to that, I would say biodiversity is something that's very high in sequence space. Like people sequence everything out there. I guess that's what InstaDeep is doing. But I guess the problem I feel like maybe in single cell stuff is people often just capture humans and I mean not capture humans but get their cells. to me, it feels like an analogy of that would be, hey, say, hey, let's get every single mammal out there and do the same exact experiments, or let's look at every single cell, right? And that's where it becomes really, really powerful, because now you can leverage all that evolutionary selection that's been going on forever, or at least for a few billion years. And then the idea is like you can start to learn these dependencies. Otherwise, all you could do is probably just learn some clusters of things, but you really need to see a lot of like multiple changes through lots of history of time to be able to, at least this is what we learned from on protein data. It's like that's where the big breakthrough came from, was being able to take all the biodiversity. And the question is, like, for other fields, where do we get that same kind of biodiversity of data? And I think that's the key to getting them to come to the same level as what we see the success in protein world.
Thank you.
Yeah, I had a sort of related question or comment, which is, I mean, alluding to Angel's point about maybe one of the things that the DNA language models could really benefit from would be annotations. And if you look in SRA at all the like ATAC-seq and all of the epigenomic data, like it's like 90% of it is human or mouse. I think it might be more even. So how much do you think that could benefit, especially the DNA language models? And it's interesting, I think, that it is something that the protein models have really benefited from, is having this cross-species stuff. Obviously, we have a lot of genomes, but they have this issue of so much of it is not functional. And yeah, how much should we be trying to get our experimental collaborators to go get a bunch of like species and do their single cell attack or whatever?
Yeah, to me, for non-coding DNA, I do think, going back to, I think, Rafi's comment, like synthetic data might be a better option. Just because, like, the evolutionary process in protein coding DNA are pretty well understood, and they are structured. And non-coding space, it's really unstructured and really kind of a coin flip kind of experiment, right? So like, you could collect a whole bunch of genomes and get very little in return. On the other hand, the rules in non-coding DNA are much easier than in protein space, right? The explicit rules. You kind of already kind of largely know what the invariances are. So why not generate synthetic DNA and measure things in the mammalian genomes you care about, but go completely out of distribution from what you see in those genomes. I think where I might disagree with Carl and others is you can synthesize random DNA, or you can like synthesize in a rational way. And I would prefer the rational way, because I think that's where you want to target the out of distribution stuff more. But anyway, that might be another strategy rather than sampling genomes.
Thanks. While I have the mic, I would recommend everyone read The Opportunities and Risks of Foundation Models, where Stanford a few years ago just invented the term. It's only a little bit problematic as a paper. My question, though, was how y'all feel about computational biology in kind of relationship to AI. Do you see it as just another AI field, right? You have vision, you have language, you have speech, you have genomes. Is that something that scares you? Do you not like that? Would it be bad if we just became another AI field? But how do y'all personally feel about that?
I'm not sure if I follow what you're trying to get at here. Do you mean, like, are you worried everybody's going to be doing AI in the field instead of other things? Well, I mean, at least for me, like, I feel like we are like using these new deep learning models, but it's always about, the end goal isn't just to get a better prediction. The end goal is to try to understand what new biology, what new physics these models are learning. So it's always, I feel like the next step is then to break the model, see what are they actually approximating. Like at the end of the day, all these models are just some function approximators. And so the goal is to figure out what function that they're approximating. So
I mean, I think it's good to have a model that captures everything and then later on break it up and see what it's learning. But I'm not sure if that's answering your question. Maybe I misunderstood it.
It's a positive thing if everyone next year submitted foundation models.
Well, if they have money, they should do it, so then we can use it. But maybe we should all collaborate instead of training the same thing. That could be probably also a good thing to do.
All right, so I think there's a lot more interest and questions from the audience, but in the interest of time, I think we can have some of those discussions during the poster sessions and also for the reception. But let's thank the panelists again for very interesting insights.
Hi everyone, I'm Charles Gad and I'm a postdoctoral researcher from Oxford University. I'm here today to present my work on multi-scale analysis of somatic copy number alterations. And so, briefly, somatic copy number alterations quantify the change in copies of DNA segments. And these changes can occur across many scales. So we have at the coarser scale changes that may affect the entire genome through whole genome doubling, to scales events that may occur only in a small fraction of the genome with vocal aberrations. A multi-scale analysis attempts to identify the events at each of these scales and the interactions between them. So on the left, we can have learner representation based in the top left of the figure based on the coarsest of scales, and as we go down to the bottom right, we can learn representations based on small focal events. And this is important because learning these types of representations may lead us to driver mutation, discovery of driver mutations, biomarker discovery, tumor evolutionary dynamics, or to detect intratumor heterogeneity. So we introduce a method called Wave LSTM. Wave LSTM is a wavelet-based long short-term memory encoder architecture, and Wave LSTM learns separate representations for each scale. It combines each of these representations using a self-attention mechanism into a single attentive, multi-scale attentive embedding. And we demonstrate Wave LSTM on two tasks. The first is the task of identifying clonal structures. And we demonstrate that Wave LSTM is able to recover the clonal structures obtained through state-of-the-art bioinformatics approaches. But on top of this, it's also able to discover novel subclones. Secondly, we demonstrate Wave LSTM on the task of predicting survival outcomes in a cohort of cancer patients. And we demonstrate that these representations are useful in improving survival outcome prediction. The preprint of the paper is already available, and you can find it using this QR code. And if you have any questions, feel free to talk to me at poster ID 28.
Hello, everyone. I'm Remy Liu. I'm very excited for this opportunity to briefly advertise our work, context-specific network embeddings via contextualized graph attention. This is a joint work with Haowen, Kayla Johnson, and Arjun Krishnan. There are a lot of human diseases, and many of them can be explained by the dysfunctioning of one or several proteins in the human genome. And several previous work, including ours, have shown that using protein interaction network embeddings is actually a pretty effective way of uncovering proteins that are associated with a particular disease. However, there is still a lot unknown about how the protein association works under different cellular contexts, such as tissue and cell types. And in this work, we aim to shed light on this problem of protein disease associations in different contexts. The idea of our approach is very straightforward. Instead of preserving the network topology of the context-naive protein interaction networks, we want to preserve the context-specific protein interaction subnetworks. For example, here are different subnetworks associated for these different contexts. And to achieve this, we designed a method called cone, the idea that effectively and efficiently achieves this goal of preserving the network topology of different contexts. We do this by using a shared graph attention neural network that can be contextualized via a learnable context-specific embeddings. The training goal is to preserve the network topology of these context-specific networks. So to quickly show you some key results, in fact, we are able to achieve better prediction for disease-associated proteins when compared to when using context-naive method. And furthermore, our methods can shed light onto what are the biological contexts that are relevant for particular diseases. Here are just some simple examples. I'm happy to tell you more about it when you're in the poster session. So for example, familial bicuspid aortic valve, we found that using the cone method, we found that heart is one of the top associated contexts, which is pretty naive. On the other hand, we also found some very interesting cases. For example, the hypochromic microcytic anemia, the method found out that small intestine could be one of the most relevant contexts. And we found out that this is also pretty reasonable. So if you're interested, come talk to us in poster number 12.
Don't see. Forget what you saw.
Hi everyone, I'm Wojtek, and my post is about QuickBinds, a lightweight and interpretable molecular docking model that we developed in the group of Professor Akroishi at Columbia University. Molecular docking is a computational technique that predicts the structure of the ligand bound to its protein target. It's oftentimes used in early-stage drug discovery to optimize the drug affinity. The affinity of a drug candidate to its protein target based on the structure of the complex. And in principle, the structure can also be determined experimentally, but that's much more expensive and much more complicated than doing that on a computer. And if docking is supplemented by an estimate of the binding affinity, it can also be used to virtually screen large compound spaces for drug candidates, thereby further reducing the cost on increasing the speed of drug discovery. There are traditional methods, there are methods for this already out there, but the problem with current methods is that they achieve good predictions, but at the cost of long runtimes, which in the case of physics-based methods is caused by sampling many different binding locations, many different binding poses, and in the case of recent machine learning models by increasingly larger models and shifting towards generative modeling and co-folding approaches, so predicting the structure of both the protein and the ligand from scratch. Instead, we developed QuickBinds, which is a lightweight method for rigid blind docking aimed at virtual screening applications by trading accuracy for speed. In terms of architecture, it's based on the AlphaFold2 architecture, which also makes it very topical based on the recent updates to AlphaFold2 for co-folding AlphaFold3. We benchmark QuickBinds on two data sets, the PD-bind test set and the postbusters benchmark. These are plots of the success rate, so the fraction of correctly predicted complexes versus model runtime, either explicitly or by sorting the models according to their runtime. And we can see that, as intended, QuickBind is orders of magnitude faster than traditional docking methods, than the current state-of-the-art docking method, DiffDock, and recent co-folding tools. But it still outperforms most other docking methods except for DiffDock and co-folding methods such as AlphaFold3. Given that a natural use case for QuickBind is virtual screening, so we developed a simple top model for binding affinity prediction such that it can be immediately used for virtual screening applications. And that's particularly important because most other docking and co-folding tools do not actually predict the binding affinity, so it cannot be used for virtual screening at all. So in a nutshell, QuickBind optimizes runtime speed while retaining competitive accuracy and is therefore a compelling option for high-throughput virtual screening. And if you want to learn more about QuickBind's architecture, performance, model interpretability, or customizability, come to my poster.
You have a PDF version too. Yeah.
Hi, my name is Masatoshi. Today, the goal of this work is a computational design, like for biological sequences. Especially, we focus on bridging model-based optimization and also generative modeling. So what is model-based optimization? It's a very simple approach. So first, like we make a sequence model from X to Y, for example, protein to like the affinity, or like DNA to like cell type specificity. And then we can optimize that with directed evolution or like the first order method. So that is a traditional approach. And another recent popular approach is also generating modeling approach. So we make a language model or diffusion model for sequences, and then like we can generate basic sequences. So the question here is that reward model, so model-based optimization, it excels in generating sequences with high functionality, and also generative modeling excels in generating sequences with more natural sequences. So here, the goal is to construct a new method that can enjoy the best of both worlds. And to achieve that, we propose a new RL-based fine-tuning approach for biological sequences. So here, let's assume that we already have a good generative model trained on large data sets. And then also like we have access to some reward model from sequence to functionality. And in our approach, gradually, like interacting with reward model, we fine-tune like generative model using reinforcement learning. And then, for example, we can generate like DNA sequences with high cell type specificity. And there are several challenges. So, for example, like in this kind of stuff is researched a lot in computer vision. But compared to this literature, there are two challenges. And the first challenge is that since we are dealing with more discrete objective, like basically sequences, so how to incorporate this like discrete diffusion model in our context is a challenge. And another challenge is that the coverage of the functional data is very limited. So like we can easily generate basically out of distribution samples if we just use a naive approach. So in the interest of time, I don't talk about how we deal with these challenges. But if we are interested, let's chat at the poster session. Thank you very much.
Hi, everyone. I'm Sabina. I'm a postdoc at Dana-Farber, and I'm going to be presenting my poster, which is entitled Precision Prognostics in Pediatric Neuro-Oncology using DNA methylation arrays and neural networks. So to motivate this problem, I want to stress how important it is to accurately predict the outcome, especially in pediatric patients. This is because we'd like to manage the trade-off between undertreatment, which can result in recurrence as well as death, as well as overtreatment, which can result in lifelong debilitating side effects. So to address this problem, we've collected data from patients with medulloblastoma as well as ependymoma, and we have this DNA methylation array data, as well as the associated survival outcomes, which is sensor data. And from this data, we can also obtain copy number variations. So we feed this into a sparse neural network, which then produces patient-specific survival probabilities over the course of 10 years. So I mentioned the sparsity of the neural network, which also enables it to be more interpretable. So we can determine for each patient which genes were highly predictive of outcome. And this has enabled us to also uncover new biomarkers. And lastly, we adapted the strategy using a causal inference framework to estimate individual treatment effects from patients which were randomized within a clinical control trial. And we noticed that there was significant heterogeneity in treatment response, which would otherwise be missed when looking at aggregated data. If you're interested in talking more, I'm looking forward to seeing you at my poster.
Good day, everyone. I'll be presenting the last spotlight for today. I'm Oladimeji Macaulay. Oh, that's one more. Okay. I'm Oladimeji Macaulay, and I'll be presenting MedGravNet, which leverages multi-relational, GNN, and text knowledge for biomedical prediction. It's really important to understand the interactions between biological entities in order for you to understand the association of these factors to diseases. In order to address this, we introduced MedGravNet, which is a multi-relational graph neural network which infers relationship between 4 biological entities, which are gene, drug, phenotypes, that is the side effect, and the diseases. In order to compile the data, we got it from several databases. And for the model architecture, the nodes were initialized using embedding from a language model. And then we trained the GCM for the link prediction. MedGravNet was able to outperform single relational graph and multimodal fusion, which will be shown in the poster. Also, we also tried to verify the generality of this model by implementing several assessment methods, which is trying to train on a set of disease gene association and then predict disease gene not included in the data set, that is the one from another database. Also, we also tried to see if we could predict risk genes for rare disease. And how did we model this? We modeled this by removing all known information of a particular disease from the graph, which is used to imitate a real-life scenario of a rare disease, and the model was able to perform well. Also, side effect prediction. We removed the side effect data entirely from the graph and tried to use the model to predict it, which the model performed very well. And for more information about this, I'll be at poster 18. Thank you.
Hi everyone, I'm Jessica. I'm a postdoc in the Q Lab at Cold Spring Harbor, and I'm here to plug our work addressing uncertainty in machine learning for biology. In recent years, deep neural networks have become a very popular and powerful tool for studying genomic sequence function, which is why we're all here today. But it's not always clear how well they generalize to out-of-distribution sequences, or DNA sequences that differ significantly from what the model saw during training. So it'd be nice if we had a way to quantify how certain our models are about their inferences, as this would give us a way to improve the quality and reliability of the biological conclusions that we draw from the application of these tools. One way to address this issue is with deep ensembles, which involve the use of multiple deep neural networks for a single prediction task. And typically, the predictions from the different models in the ensemble are combined to yield a single prediction, which has been shown to have improved performance over individual models. And additionally, the degree of disagreement across the individual predictions reflects the model's uncertainty for the given input. And this quantity represents epistemic uncertainty, which is one of the 2 primary sources of predictive uncertainty. Now, while deep ensembles offer notable advantages over individual models, they also suffer from several limitations arising from their dependence on multiple models. And so to address this, we leverage a method known as ensemble distribution distillation, wherein we essentially generate new target labels for the data using the ensemble's predictions, which are then used to train a distilled model that mitigates the disadvantages of deep ensembles while also preserving their benefits with just a single model. And more specifically, we achieve this by assigning the average and the standard deviation of the individual predictions from the models in the ensemble as new target labels for the data, which we then use to train a distilled model wherein the output layer is modified to include a new output head for epistemic uncertainty, which is learned from the ensemble standard deviation and trained alongside the original prediction task. And we applied this method to different genomic sequence models across various functional prediction tasks, and we were able to demonstrate that our distilled models do indeed yield improvements in performance over individual models, and additionally are able to predict epistemic uncertainty quite well. Having said that, epistemic uncertainty alone doesn't offer a comprehensive view of predictive uncertainty. Ensemble distillation doesn't capture aleatoric uncertainty, which arises from inherent noise or uncertainty in the data that the model learns from. So in the case of genomic sequence data, that could be like technical or genetic variation. So we estimated aleatoric uncertainty by training our models on the variability across biological replicates in the data, and then applying distillation to these models to yield uncertainty-aware models that can estimate both aleatoric and epistemic uncertainty. And when we evaluated these uncertainty estimates and compared them to those from several other popular methods for uncertainty quantification, we found that the total uncertainty estimated by our distilled models were quite well calibrated, and that we could additionally ensure further statistical guarantees when applying conformal prediction to these uncertainty estimates. So for more details on everything I've just shared in the Spotlight talk and more, make sure to check out my poster at this afternoon's poster session. I'm poster number 60. Thank you.
All right, that's the end of the Spotlight session. So now we have the poster session, which is in the Allen Atrium. So if you go downstairs, cross the little road, there's the Allen Center building, and then just go inside. You should be able to see the poster boards. And see you guys all there in 5 minutes.